A very good morning to everyone present here. I take this privilege to warmly welcome all our delegates and participants on third day of INDO-US workshop organized by JSS Medical College. We will now begin with our first session of the day on basic epidemiology concepts by Dr. Purnima Madhivanan. I invite Dr. Raghavendra Rao, Associate Professor, Department of Microbiology, and Dr. Mahesh P., Professor, Department of Respiratory Medicine, JSS Medical College, to chair the session on basic epidemiology concepts.
Yeah, a very good morning to all of you. I'm sure you have had 2 good days of learning, and we have one of the leading epi persons from India and US sharing her thoughts with us today. I'm sure I don't need to introduce Purnima. You have been in touch with her for the last 2 days. So without much ado, I will now request Purnima to go ahead with her talk.
Thank you, Mahesh. Good morning, everybody. There you go. Now I hear you. So you're all fed, caffeinated, ready for another exciting session. Today we're going to it's a culmination of all the things we talked about for the last 2 days. You learned about study designs, you learned about concepts, you learned about terminologies on infectious diseases. Today, we're going to put it all together to do an investigation. So what do we mean when we say an outbreak investigation? So in the time that I have, I'm going to discuss ways in which outbreaks are recognized. We'll discuss the role of a laboratory in epidemiologic investigations and surveillance. Given a scenario, we'll create a plan as to how we can investigate an outbreak. And then given the data, we're going to categorize cases using a case definition. And then we're going to figure out how do you produce a line listing and how do you create a epi curve. I think you're familiar with almost all the terms that I have talked to you so far. And the main one is case definition. Do you all remember case definition? Okay. So a case definition for an infection could be, there's a person who's showing up with vomiting and diarrhea. And when we say diarrhea, we're being even more specific and saying 3 or more loose stools. And it was happening among the members and staff of the T20 cricket team. And the onset was September 19th in the morning or any time after that. Okay? It's still a little vague, not too specific, just enough to know where it is happening, person, place, and time. So now we're going to get more specific from here. So who recognizes outbreaks? Well, the main people who can say something is wrong or something is going on are the clinicians, because they are the ones that people first go to if they're sick. Then, of course, you have the infection control practitioners, and then you have the non-medical staff. Maybe the children are not going to school, or they didn't come to the daycare. So they're saying, what's going on? Why are these kids not showing up? So they could be some of the people who can recognize. And of course, you have the contacts or the extended family of people, or it could have just come from a news report. Suddenly you say in this village, there was a jatra and then everybody is throwing up. So it might just be a news clipping in a local television channel. Or it could come from surveillance. So any of these different places can give you some hints or clues as to, is there an outbreak happening or is there an outbreak going to happen? So I'm going to talk to you about, I used to live in Miami before I moved to Arizona. I moved to Arizona just before the pandemic started. So I don't know Arizona that well. I used to live in the state of Florida, which is in the southern part of the United States. And in the southernmost tip is a city called Miami. I don't know how many of you are familiar with Miami, We don't think of Miami as United States. We think of Miami as the closest city to the United States. So it's highly diverse, and most places from Southern America, the first port of entry for people is at Miami. And Miami is also a place where if any of you want to go on a cruise, you're going to show up in Miami and board the ships in the Miami port. So it's a highly mobile population, highly diverse. So in the first week of February in 1999, they had 5 suspected cases of typhoid fever. And that was reported to the health department with the onset between January 1st and January 28th. Okay? Now, the question is this unusual? How do you decide whether this is unusual? Somebody take a shot at it. They saw 5 cases of typhoid. I can't move. If you don't tell, I cannot move to the next slide. Yes, sir.
Where is the source of infection? If the file is in different places, that's something different. But if the source of infection is cell, class of 5 people, the body, that may be something.
So you're saying unless we know the source of these cases, it's not unusual?
Part of the same source.
Yes, but this is a very simple yes-no question. Is this unusual? Yes, no. Yes. Okay, now the next question. Why?
So not unusual, they are not reported.
Exactly. Excellent. That's it. The fact that they're asking you the question, is this unusual, means it is unusual, right? Otherwise, we won't be sitting and talking about it. Because 5 cases of typhoid in Mysore, we don't even think about it. It's just another thing, right? So, how do you say if something is unusual? So here's a global map of typhoid cases around the world. Now take a look, and this is the United States. And when you look at the incidence rate, what do we mean by incidence rate? Number of new cases, right? Look at the incidence rate in the United States. It's 0 to less than 15 cases per 100,000, right? So in a situation like this, to find 5 cases, and I'm talking about a place right here. This is the state of Florida, and I am at the southernmost tip of Florida. So we had 5 cases of typhoid in this setting. Now compare it to India. Look at India, right? So if you see cases in a place which is light like this, that means something is not right. Okay? So that is your first clue. You know the basic epidemiology. You know whether something is common or not in those communities, and then suddenly you're beginning to see something emerge. So that gives you a clue whether this is usual or not. So in the US, we have about 400 cases a year. Now, when we come to the state of Florida, we have about 15 cases a year. And when we come down to Miami, we have 3 to 10 cases a year. Now compare it to the 5 cases in 28 days, right? That's how you start looking at the graphs. And most are associated with foreign travel. I told you where we are located. We are one of the port cities. There's a lot of movement. So here are those cases. Now we're looking at a graph. On the x-axis is the time, and on the y-axis is the number of cases. Now, if you go to 1997 or 1998, take a look at the number of cases. What do you see? 0 cases most times, right? Now, look at January of 1997, no cases. January of 1998, 1 case. January of 1999, 5 cases. So this looks unusual. So in terms of the outbreak itself, again, it's all about epi. That's why it's so important. Person, place, and time. So you have to go back in time and look at what was the epi profile of this infection. So in 1973, there were more than 100 cases of typhoid, and most of them, the source of infection was contaminated water. Right? Drinking water system was implicated in almost all of those cases. Now, when we fast forward 1993 to 1994, they had 17 cases, but there was no source implicated. They couldn't figure out. There was no common themes. Epi is about patterns, right? I told you, epidemiology is about patterns, recognizing patterns. You don't see any pattern there. There was no source implicated. So then that's when you decide, okay, we need to find out what's going on. Why is this happening? So that's when you start to investigate an outbreak. So the reason you do that is it has to be ongoing. You want to immediately have some control measures installed right away so that no new cases come up. And you want to identify the reason for those 5 cases and see how you can stop more people getting infected. So these are the public health measures, and public health departments have to rush into this. Then you also do an investigation to gain new information. You want to understand, has this agent become more virulent? Has it become more infectious? So you want to understand how the agent is changing or not changing. How is it transmitting to other susceptible hosts? You want to also understand, how can you control it? How can you prevent it? And then you have to plan for the future as well, so that no new cases arise in the future. And most of all, you don't want the public outcry, because it's at that point that the politicians get involved, everybody and their grandmother get involved. And then it becomes a drama. Opposition party will say, this party didn't do anything, and this party will say, we are doing everything, but these party people are not helping us. So it becomes a huge drama and takes away from the focus of trying to save lives. So you don't want to get to that situation. So you want to address the public concerns. So you want to be in front of the media before it becomes a problem, and you want to communicate. That's why it's very important for all of you to learn how to communicate. No using jargon. We know you know your stuff. We know you know all your techniques. But as a layperson, I don't care. I need you to tell me in simple English what you're trying to explain. So you have to learn how to communicate to different audience. And when you're talking to public, you never use complex words. You keep it simple. You have to be able to explain it to your grandmother. If you cannot explain what you're doing to your grandmother, then that means you're not learning how to communicate. So each of you need to try and practice explaining to your grandmother or the lady who comes to sweep your house what it is that you're working on. And she has to understand it. If you cannot do that, that means you're not learning how to communicate. So it's very important to address the public concerns in a manner that you're not agitating them and scaring them, but you're calming them. And then you are building an ally. Then they will help you. And when you become their friend, they're more likely to share more information with you. They say, let's go check in that village also. Why you are seeing so many cases? Otherwise, they won't tell you. They'll say, oh, these people are coming to put us in a jail. So they're not going to tell you. So you have to become allies. You have to become friends with the community. Build those relationships so that they will take you into their confidence and they will trust you that you're doing the right thing. And the only way they can do that is if they understand what you're doing. So in order for them to understand, you have to simplify into a language that they can understand you. And then you fulfill all the statutory mandates that are required when you see an outbreak. And of course, you have the legal issues that we have to address. So these are all some of the reasons why one needs to investigate an outbreak. And it gives an excellent opportunity for the next generation of researchers and scientists and public health people to learn how to do some of these things. So in terms of this particular outbreak that I was telling you about, the 5 cases of typhoid, the reason we needed to investigate that outbreak was it was ongoing. So we needed to identify and eliminate the source of that infection. And we needed to follow up with all the contacts to see, are there more infections arising in these populations? And then we want to fulfill all the statutory mandates. So I've been talking about typhoid. So we need to know what it means, right? What is typhoid? It's essentially a bacterial infection caused by Salmonella typhi. And the most common signs means what you see, and symptoms is what you feel. So you want to understand what are the signs and symptoms of typhoid fever. So if somebody has prolonged fever, or they're not hungry, they don't feel like they want to eat, they feel very tired, they feel fatigue, they have pain in their stomach, and then they can't pass motion or they are passing excess number of motions, right? Either it is constipation or it is loose motion. And their heart rate slows down a lot. There's relative bradycardia. And then they start developing a rash on the body, which is a rose-colored rash. And you have non-productive cough. So these are some of the signs and symptoms of diagnosing typhoid fever. So what's the incubation period? We heard that word yesterday, right? Incubation period is from the time that a person gets the infection to the time they start showing symptoms. That period could be anywhere between 1 to 3 weeks. The reservoir, the reason why this is unique is it's not through any vectors. Remember, we talked about vectors. There are no vectors for this. The only source is a human. So the organism gets into the body and it stays in the body and it does all its magic in your body. There's no other animals or birds that it will go to. The most common risk factors are foreign travel and consumption of contaminated food and water. Okay? How do you diagnose typhoid fever? Well, the most common way is to be able to isolate the bacteria from the blood or the stools, urine, or bone marrow. And the complications, if it goes untreated, is it can lead to ulceration and perforation of your large intestine. It can lead to inflammation of your liver, your gallbladder, your lungs, your covering of your heart, the covering of your brain. It can lead to inflammation of all of these parts. And if you treat it, it's completely a normal thing. You can give antibiotics and person will recover 100%, at least 99.9%. But if left untreated, 1 in 10 people will die. So that's the seriousness of typhoid. So you want to identify the case, you want to make sure that you diagnose it, and you want to treat it immediately. So in order to do all that, you have to go ahead and start the investigation. So how do you start it? First of all, you have to confirm that there is an outbreak, right? You have to understand, you said this is unusual. So it's unusual, so we need to do something. So now you have to go and confirm the diagnosis of those 5 people. Do they really have typhoid, or is it they're confusing it for some other diarrheal disease? So the laboratory confirmation, the case definition we talked about yesterday for listeriosis, you have to have all of those things come together. You have to have a clinical diagnosis, a laboratory confirmed diagnosis to say, yes, this is typhoid or Salmonella typhi infection. And then you have to establish the background rate. You just saw the graph. We saw the background rate. It was usually one case over a period of 2 years. And suddenly now this year there were 5 cases. So you know your background rate was very low. So you have to have those kind of data in order to identify if this is an outbreak. Then the first thing you want to do is, it's like if you have a hole in your roof and it's raining. You don't go and say, well, let's see how much water is going to come. I'm going to measure the buckets of water. First thing you want to do is seal it, right? Seal it with whatever, maybe a tarpaul or a plastic, something. Then you can decide whether you have to repair the tile or you want to put new concrete. That's secondary, but you want to first seal it immediately. That's what you want to do. Implement immediate initial control measures to see how we can stop this. Then you establish a case definition, because that will become the premise on which you are going to do your entire investigation. Based on that case definition, now you're going to go and find all the cases. That's why we call ourselves shoe leather epidemiologists. That means we are not sitting in front of the computer. Our soles of our shoes get worn out because you're going, walking, checking everything. So if you look at the EIS officers or EIS training, the Epidemiologic Intelligence Service at the CDC, the logo is a shoe with a hole. That's what EIS officers' logo is. So traditional classic epidemiology is all about going into the community and checking things out. So you have to go and find cases to establish the source. Then you have to collect comprehensive data from these patients about all the possible exposures. Did you travel? Or did somebody in your house travel? Or did you go to a picnic? Or did you go to a party? Was there a wedding? Did you go attend a wedding where you went and ate? You have to go ask them all the potential ways that they might have picked up this infection. Then, based on that triangle I showed you yesterday, now you have to start thinking about placing all that information in the context of the person, place, and time. And that's the classic epidemiologic information and in the context of environment. Then you determine who is at risk of becoming ill. So now you need to not only find the cases, you also have to find the potential contacts of the cases to see if they might pick up the infection and they might become a case themselves. So you have to go and find out about all of these people. Then, with all of this information, you're going to set up a hypothesis. And once you have a hypothesis, then you go and test the hypothesis. Unless you have a hypothesis, you can't test the hypothesis. And unless you have a hypothesis that is testable, you can't test it. They're connected. That's why you have to be able to say your hypothesis in a manner that it is testable. So you develop the hypothesis, then you test the hypothesis. Again, ensure that you're implementing all the control measures, and then you write your report. So these are essentially all of the steps of investigating an outbreak. And much of this information that you collect is going to become part of the surveillance data. We talked about surveillance yesterday, right? This becomes part of the surveillance data. And surveillance data, again, gives you clues as to whether there is an outbreak happening or not. So talking about case definition, the most important basis on which you will investigate an outbreak, you have to be able to come up with a very, very specific case definition. So you have to have your necessary inclusion and exclusion criteria. You have to talk about how you're going to define it in terms of clinical diagnosis, in terms of laboratory diagnosis, what is the population you're looking at, what's the time period you're looking at, and what is the place you're going to be investigating this. So for us, it was in Miami between July 1st and August 30th. We wanted to look at the nursing home residents. It's just an example, not related to the outbreak I'm talking to you about. This is just an example of how you would think about it. Then you have to identify whether these are probable cases or are they confirmed cases, right? How would you differentiate that? How would you say something is definitely a case, or how would you say something is probably a case? That's a confirmed case. Excellent. So if you have one or the other and you don't have the whole thing, then you can say it's probably typhoid. But if you have both, like she said, you have the laboratory confirmation and you have the clinical confirmation between the two of them with the magic word and, it's a definite case, right? So you have to make sure that you have all of those definitions. And many times, as the investigation is progressing, sometimes you might have to tweak your definitions a little bit. But you have to make sure you're documenting that. Documentation is very important for that reason. So our initial case definition was a case of illness in the Miami-Dade County with symptoms of typhoid fever with isolation of the bacteria from blood, stool, or other clinical specimen since December of 1999. Okay, so we have the clinical piece and we have the laboratory piece. And we have given the time period and the location. So then you start your case finding. How are you going to find out where all these people are? We know of 5 people. But can there be more? How do you find out? So you pick up the phone and start talking to health departments. You send out messages to all the hospitals. Say, hey, it's possible that you might start seeing more diarrheal disease cases. Or you might tell the laboratories, the public health laboratories, please let us know if you see a case of typhoid right away, inform us. So you're going to start, this is called active case finding. Remember active and passive? You're going out there and asking them, let us know if something is happening. So active case finding is very important in outbreak investigations. And then you, of course, you reach out through the media. You put out little snippets saying, oh, do you have somebody in your family who's having diarrhea? Do you have somebody in your family who has fever? Do you know somebody who has this? If so, please contact us. 188, whatever the number, right? So they put it out to the public so that the public also can start helping them with identifying. And for all the cases that we have found, we go and talk to them and try to find all their networks. Talked about networks yesterday, remember? So you have to go and check all of their networks to see anybody in their network is also sick or not. Then you will use a survey form to collect all the information about these people. Who are these people? So all the characteristics of them. How old are they? Are they men? Are they women? Are they children? Are they living in a Pca house or are they living in a slum? So you need to know everything about them because you want to identify all the particular sources that could have potentially led to the infection. So you want to understand all their risk factors. I told you for us, it is foreign travel is the main risk factor. So we want to find out, did you travel? Or did somebody in your family travel? Or did you go to somebody's house who had traveled? So you want to find out all the potential ways that they might have picked up the infection. Then you'll test their networks, their close contacts, their household members, and then you continue to monitor these cases very closely. Then you start to map out all of these people. So there are three ways we can orient ourselves in trying to understand the person, place, and time. So one of the ways is a map. The map I've put there is a classic map of a cholera outbreak that happened in the UK, in London. There was an outbreak of cholera, and we call John Snow the father of epidemiology. This man, he actually went, he drew out the whole community, he mapped out, went house to house and found out how many of them had cholera in that house. So if you notice all these little black dots here, all those are cases, and each one is mapped out by the house. And then you start seeing if there's a distribution. Are there any clusters in any areas? And based on just mapping all the cases, he found out that the source was one water pump where people go to collect water. One water pump. All they did was they removed the handle of that pump and the cases stopped. Even to this day, if you go, you will see that water pump in London. And there is a pub right next to it. So you go take a picture with the pump, then you go in and have your beer. So the Broad Street pump was the source for cholera, and that's how they map it out. So you go and map out all the cases. Another way to do it would be to use line listing. That means every case you have, you describe it in detail. In this case, we have the case numbers. When did the symptoms arise? What was the symptoms? Did they have diarrhea? Did they have vomiting? Did they have fever? Did they have laboratory confirmed diagnosis? And who were these people? Male, female, age, where did they live? So this kind of the table that you create is called line listing. And then you graph it out. And this is how you create the curves, the epidemiologic curves. So here now you're putting it out along the x-axis with time, your y-axis with the number of cases, and then you put out the epi-curves. So these are the three classic ways of presenting the information you have in an outbreak. So for our 5 cases, this was the line listing. We had 5 cases diagnosed between January 8th to January 28th. The ages range from 6 years all the way to 40 years. There were men and women who were affected, but they were all of one race. They were all of one race. They were all of Hispanic ethnicity. They were not Caucasians, not white people. They were not African American. They were not black people. They were not Asians, not brown people, but they were Hispanics, another set of brown people. Hispanics are from the region of Hispaniola or Latin America. Okay, so they were all, that should give us a clue already, right? Because the age is telling it's all across the spectrum. It's not just men or it's not just women, so it's both. But the fact that they were all Hispanic is something going on. Is there something associated with being Hispanic? You start to think about potential hypotheses for this, right? And then where did they live? They're all living pretty close to each other. Hialeah, Opelika, and Westgate, they're all in the same geographical region in Miami. But none of them had traveled. You remember one of the main risk factors we said for typhoid was travel? But none of them had traveled. Now, based on this, what do you think it could be? You have to start thinking, right? What could it potentially be? Because you have to think, what is your next steps? Where do you want to go and investigate? What are you going to do? Food sources. But that's for everybody. right? So why just this group? Sorry? So somebody says maybe Hispanics are more susceptible to typhoid. But why? We said the main source is contaminated food, right? Is there something specific that this group eats that maybe others don't eat? Could that be a potential idea? Right? So you have to start thinking along those lines. And here is the epi curve. We saw this, so now you have an idea that it is unusual, right? Now the next thing we do is now we go and do the laboratory investigation, and now we use the specific molecular methods we talked about, right? So all of them were tested with the PFGE investigation to see were they all coming from the same source or different sources, right? We did find that 4 of the 6 had same patterns. Now you're really wondering what's happening, right? And that's the 4 that had identical patterns on the PFGE. Further investigation, now you have to start generating hypotheses. So on Feb 10th, all laboratory confirmations were done, initial control measures were initiated, there was enhanced surveillance set up with hotlines to hospitals, and then there was press releases for public education on making sure, please be careful, there's typhoid, make sure that your food sources and your water you're drinking is safe. By Feb 15th, now we had 10 more cases. And 3 of them mentioned something called mame shakes. Mame is like a sapota, you know? It's delicious, unique. We used to go all the way to the southern tip of Everglades to a stand, a fruit stand called Robert is Here. He was so popular that people from all over the country would come to go to Robert's fruit stand to have a mame shake because he was so popular. 3 of the 7 people had said they'd had mame shake. So suddenly now you have a clue. Could it be the mame shake? How do you find out? Right? But you have some clue to start with. Now the next thing is, why only those few? So now the thing is, can that bacteria survive in different environments? Somebody asked me that yesterday. So now the question is frozen mame a plausible vehicle? Was that package contaminated in the first place? And then it was when they were processing of that frozen mame, the bacteria got settled in. And now wherever those contaminated packets were being used could be leading to these cases, right? So biologically frozen mame production, Salmonella survives very well in freezing temperatures. And many times when you're using the pulp, you're not cooking it. Because it's a smoothie, right? It's a fruit smoothie, so you're not going to cook it. You're going to take it, put it into your mixie, put more ice and enjoy. So you're ingesting it wholeheartedly, right? So that's how the mame, frozen mame comes into the grocery stores. So 2 of the 15, all case patients contacted, all but 1 had consumed the mame shake. So now we definitely have a clue. So now we're going to start generating hypotheses based on this information. So our hypothesis was drinking a mame shake or eating mame is associated with typhoid. Now you have the exposure and you have the outcome, right? And the way the hypothesis is written, it can be tested. So now we have a testable hypothesis. Then we go and test it. You're going to go and do a case control study. Now, why are we using a case control study? Why? Why are we using a case control study? When do you use a case control study? When you have a rare disease, right? Rare disease. You cannot use a case control study for typhoid fever in India, right? Because it's not a rare disease in India. But in the United States, I showed you the graph, it's a rare disease. So you can use a case control study. So all those cases, typhoid fever, were your cases. And then you match people to those cases to create your controls from the same base population. So now you have your cases and you have your control. Now you go and talk to all of them and find out, did you have mummy shake? Did you have mummy shake? Did you have mummy shake? So you go and ask all of them about all the potential risk factors for typhoid fever. Then you create that 2x2 table that I showed you. Then you will calculate something called odds of exposure in the deceased, odds of exposure in the non-deceased. Then you take the ratio of the two, you get your odds ratio. Okay? That's how you test your hypothesis. Simple. And all of this you're going to do based on the data you have collected using your questionnaires and by administering your questionnaires. What's really important in case control studies, I told you, you can get biases. Remember biases? Because he's a case, I'm going to say, Kaleem, try and remember. He'll say, I don't know, I'm not. Kaleem, yesterday you had mummy shake. Maybe not, possible, but think, you might have. Because he's a case, I am beating the information out of him. And because she's a control, did you have mummy shake? She's not even responding. So I'll say missing information, done. That is the problem with case control studies. So you don't want the interviewer to know who is a case and who's a control, so that they will be standardized in how they ask the questions to everybody. The results, now you will go and create those 2x2 tables for every single exposure that you are assessing. So you want to check for water sources, fresh vegetables, fresh fruits, juices, shakes, milk products. You will find out the odds ratios for every single one of these exposures. That's how you find out. And here, when we did all of that, we found no association for any of these except for mummy shakes. So this is your odds of the exposure in the cases, odds of the exposure in the non-cases. So this is your odds ratio, and here is your 95% confidence interval, right? Because odds ratio is on a multiplicative scale, if you have your numerator and denominator being the same, what's the number you get? 1. When you have an odds ratio of 1, which means there's no difference between the two groups, right? So if your 95% confidence interval includes a 1, that means there's no association, right? So for banana shakes, for mango shakes, it included a 1. But for the mummy shake, it did not include a 1. And your odds ratio was 8.8. That means the cases had 8 times increased odds of having had a mummy shake as compared to controls. That's how you find out whether something was a risk factor. Then, you remember I told you you have to be very careful about using the word causal. You always say association, but you don't say causal. But in outbreak, you need to talk about causation. So now you have some specific criteria you can use to say an exposure causes an outcome. And some of those main concepts was the strength of association, consistency, specificity, temporality, and coherence. So we have to be able to address all of these criteria in order to say that mame shake caused typhoid fever. And then based on that, now you have to go and do your control measures. So we informed the Department of Agriculture and Consumer Services to stop sales in all the supermarkets. We made sure that all the sales in the restaurants were stopped for this mame shakes, and then we put out a press release. When I say we, it's not me. I'll show you who actually did this. I'm just presenting their information. But you have to do all of this as soon as you found out 8.8 odds ratio for mame shake. So we know there's something going on. You have to inform the FDA. FDA issued a warning. Frozen mame, there's only certain companies that produce it. So the FDA will run after those companies to say, hey, stop production. Please recall all the product that you have sold because we don't know which of those are contaminated. Then the CDC calls all the states with large Hispanic communities, but there were no other associated cases found in other Hispanic communities. And then you report the patterns on PulseNet, and there were no matches found. Then they had to go and trace back all the samples. So they collected all the samples that were available from the homes and the restaurants and the supermarkets. There were only 3 brands that manufactured frozen mame. So they went after all the 3 brands, but all the patients had identified one of those 3 brands. So now it's narrowed down further. It's not all, we just need to go after this one brand. So you contact those distributors, those companies. Most of them had come from Guatemala. The local growers were contacted, and no frozen mame was produced locally in Miami. So the product was recalled, samples were tested. They found large amounts of fecal coliform in unopened packages for 2 brands. But there was no Salmonella typhi identified in them. They had other coliforms. So FDA announced a voluntary recall of both the brands. And then you can see the number of cases now for February has also risen. These were the additional cases that I told you about. So basically, it landed up that there were 16 cases of typhoid. 15 of those isolates were tested. They identified the patterns. We were able to epidemiologically link mame to typhoid fever, right? And now, since most of the cases were in Miami-Dade County, now the question was, is there something unique to how this mame product is being distributed in that community? So now you start thinking about it. Could there probably be more cases? If we remember, we talked about surveillance and underreporting. Is it possible that somebody is self-medicating? Because most Hispanics from other countries are like us Indians. We take our packet of antibiotics wherever we go and we self-medicate. We don't go to a doctor. So maybe we have missed cases, right? And the importance of understanding the community. That's why it's so important to understand the cultures that you're working in, understanding the communities you work in. At PHRI, we do not allow lab folks to sit in the lab. They are some of the best trained lab folks that I have seen. But at PHRI, they're not allowed to be in the lab by themselves. They have to go into the community. They have to understand the dynamics of the community. They need to know where those samples are coming from. How are they being collected? How are they being transported? It's very easy for Kaleem to say, hey, this piece of blood is hemolyzed, go get another one. But for the community person has to travel 3 hours to go and find that person and draw blood again. He would not appreciate that if he didn't go and see what people have to do to collect that sample. So it's very important for you all who do laboratory work to understand your communities. Now we have better diagnostics than PFGE. Now we're doing whole genome sequencing. But this was 1999 that I'm talking to you about. It's also important to understand how different agencies work. How do they work among themselves, different departments, and how do they work between agencies? You have to understand the logistics of it. We have to understand globalization of food supply. All the mame that caused typhoid fever came from Guatemala, Central America. It came into United States. So the food that you're eating may not necessarily be grown in your backyard. It may be coming from very, very far off, what we call food miles. The more food miles your food has, the more problematic it is. That's why we say, eat local, eat seasonally available stuff. Don't try to eat fancy stuff, which has to come thousands of miles away because of these issues. And you want to understand what are the best way to investigate and how do you diagnose. Maybe there are newer techniques, laboratory techniques you can use. Now, like we said, we've moved on from PFGE and now we use whole genome sequencing because we get better information, more information, richer information. So these are all the people who were involved in that one outbreak investigation. We had the Office of Epidemiology and Disease Control. We had Healthy Start, the school programs. We had environmental health folks. We had the regional laboratory. We had the HIV/AIDS unit. We had public information people. We had administration. We had the Department of Health. We had practitioners of infection control. We had Department of Agriculture, Department of Business, FDA, CDC. All of these people were involved in that one investigation that I just talked to you about. So this is just an example of an epidemiologic curve of a hepatitis A outbreak that happened in 1978. Again, x-axis is the time, y-axis is the cases. You can see there was one index case, which was actually a food handler, and look at how that spread. A typical example of what I talked to you all about Typhoid Mary yesterday, right? It's very similar. And here is another Hep A in another state. This was happening in a dorm room or a dormitory or a hostel. Again, a staff member and students. So that's it, folks. That is it of an outbreak investigation. Any questions?
Thank you, Madam, for another very crystal clear presentation. I think Madam explained very clearly first how to identify an outbreak, starting from how to identify outbreak to what is the importance of knowing an outbreak and various steps and the importance of communication, documentation, everything. And I'm sure all the audience here know that how important is it to investigate an outbreak once you confirm that an outbreak, so that it will help you to identify the source. Thereby, you can start your establishing your control measures and preventive measures so that the small outbreak you can prevent it to become an epidemic or a pandemic. I'm sure all of us are really in a position to know that how important is investigation, because we're all going through that bad phase of pandemic. So this could have been prevented if there was an early investigation into it. Okay. So, anything you want to say?
Any thoughts, any questions from the participants? Okay, there are no more questions. I think we'll close the session. Thank you, Purnima, for a very lucid talk.
Thank you for your insightful information, ma'am. I request Dr. Akila Prashant to hand over the mementos as token of appreciation to the chairpersons. Now I request Dr. Kerry Cooper to take over the session on practices of molecular epidemiology. I invite Dr. Akila Prashant, Associate Professor, Department of Biochemistry, JSS Medical College, and Dr. Prashant Alami, Chief Scientist, Molecular Biology and Biotechnology, CFTR, to chair the section on practice of molecular epidemiology.
So good morning, everyone. Yesterday we have heard Dr. Kerry Cooper when he took us through the technologies of next generation sequencing, from the basics to the advanced technologies and different platforms what we can use. And then later he took us through the bioinformatic pipelines too, and how we can analyze the data what has been generated from the whole genome sequencing from the microbiome. So today he'll give us some real world examples. So over to you, Dr. K.
Thank you. So as was just mentioned, we've kind of built up, the technology, how you analyze it, and now we'll look at some of the stuff we've done in my laboratory using a lot of these technologies to answer a number of different molecular epidemiology questions. So today, I'll go through and present a couple of examples of applications of whole genome sequencing for molecular epidemiology using real-world data. One will be a clinical case where we were looking at human cases, and the other will be more using molecular epidemiology to try and track what's going on potentially from contamination and looking at more directly at the sources and how things are moving around. So I'll get into each of these. I'll go through the different analysis we used on the data, so you'll kind of see the output data. I'll walk you a little bit through how we got there. And then finally, the interpretation from this data analysis and how it pertains to the initial molecular epidemiology question that we were asking when we started these studies. So the first one we'll go into, this will be the one that's more of a clinical base. I'll talk a little bit about Campylobacter because I'm sure not everybody in here is familiar with it, but it's considered a sporadic. So we just heard about outbreaks. This one is a pathogen that is considered to cause sporadic cases. So it doesn't typically or isn't typically associated with outbreaks. However, with the advent of next generation sequencing, we're starting to learn and we're starting to see in a number of different countries that maybe there are more outbreaks that are just being missed. So we just heard all about tracking an outbreak, but there's also the chances of missing some of these outbreaks if it's not reported. I mean, you see all the work that goes into determining an outbreak and all that. So you can see how it would be very easy to miss these outbreaks. So we went into this to look to see, are there Campylobacter outbreaks that are being underreported or being missed entirely? And then does population demographics alter the clinical manifestations of these Campylobacter infections? So does it alter, whether a person develops fever, whether a person develops, diarrhea, some of the other symptoms that are associated with these diseases? So to kind of give you a little background, Campylobacter, it's a small, thin, curved, gram-negative rod. So you can see the EM photo here. On the right, there are 32 species that are within the genus. 4 of these species are commonly associated with pathogens as human. The 2 major ones being Campylobacter jejuni, that's the most major, that's about 80% of the infections. Campylobacter coli is the next. There's Upsilensis and a few others that are associated with disease. But we're constantly using next generation sequencing. My group, in collaboration with a group in Peru, just published a paper where we think we found a new species that's causing disease, particularly in young kids under 2 years of age. So it's constantly a growing kind of moving target when it comes to this. They're modal. This is a big key. They move around, they zip in, and they get into the intestinal tract, and they kind of burrow down into the intestinal tract and cause diarrhea that way. So motility is super critical for this pathogen. Microaerophilic, so they like just a little bit of oxygen, not too much, but they can grow anaerobically like you see in the intestinal tract. They have enhanced growth at 42 degrees versus 37. So they prefer a warmer temperature. So actually their preference and their reservoir are avian or birds. So in the US, poultry, actually in a lot of the world, poultry is a major source of Campylobacter. But birds in general are a big reservoir because they're happier at 42 degrees. But they will grow at 37 degrees. So if they get into us or into a mammal, they're pretty happy at that body temperature, not as happy as they are if they're in an avian source, which will be key. And that's why I'm kind of emphasizing this for the second study that we'll talk about. So they're widely distributed though in warm-blooded animals. We find them all the time, not only in poultry, but in cattle, pigs. You know, you'll see in the second study, I mean, we looked at coyotes, which if you're not familiar are canine species, raccoons, a wide variety of different mammal species around the US. Contaminated food, water, and raw milk are the most common sources of it, particularly foods that neutralize gastric acids are key to transmitting Campylobacter. So the front line for intestinal diseases is your stomach acid. Campylobacter is not crazy about it, doesn't survive really well. So any of these things that help neutralize that help lower the infective dose of Campylobacter. Fecal-oral transmission, it can occur from person to person, but it's pretty rare, even for food handlers to transmit it via disease. It's most likely contamination of the food. Oops, lock up here. Oh, there we go. So globally, it's estimated that there's 500 million cases of Campylobacter. So it is the #1 bacterial enteric infection in the world. It's been heavily linked recently with growth stunting in a lot of countries in kids 1 to 2 or less than 2 years of age, because they're getting it and we're finding out Campylobacter's everywhere. They're constantly exposed to it. They constantly have diarrhea. Of course, then they're not getting their nutrients. So this is a major problem. Effective dose can be as low as 500 bacteria, so you don't need a lot in order to get sick. Incubation periods, 2 to 5 days, typically. It also is associated with a number of complications, and this is where it raises some concerns. It can be reactive arthritis. The big one with it is Guillain-Barré syndrome. If you're not familiar with this is a temporary paralysis. It starts typically in the arms and can descend to the legs, where upwards of 3 to 6 months, people lose the use of their arms and their legs. Fortunately, it typically reverses itself after that period, but you can imagine what it's like to have 6 months of not using your arms and legs. The other one that it's been associated with is post-infectious irritable bowel syndrome. So this is one we look at in my lab too, because this can last upwards of 10 years afterwards. Duration is usually self-limiting, so most people without any type of antibiotic treatment are going to be fine. They'll recover. Usually within a week, upwards of 10 days at the most. But there are cases where people need antibiotics in order to help recover. All right, so for this first study, the methods we used, so we used the study site, Denver, Colorado in the US, which is not, it's this little spot here that drops in right here. So Denver is the capital of the state of Colorado in the US. And these are, the different highlighted in green are what are called food-net sites. So these are CDC sites that they set up to monitor foodborne disease. And they do a lot more epidemiological investigations, a lot more laboratory work in those states to kind of monitor around the United States, because obviously they can't monitor everything that's going on. So these sites are set up to monitor heavily, kind of get an idea of what's going on around the US when it comes to foodborne. So Denver was our site that we chose, and we chose this because it's a food-net site. We obtained all the Campylobacter isolates from the Colorado Department of Public Health and Environment from 2017. So the one thing, and you'll see I said not confirmed as Campylobacter. So they sent us all these strains. They sent us 64 strains. So not all Campylobacter cases get cultured in the United States. A lot of them, they use other molecular tools like PCR and ELISAs as immunoassays to detect it. So these were 64 cases where we had an actual culture that had been confirmed. Not all of them at the time that they sent them to us were Campylobacter. So we then turned around, we extracted DNA sequence and sequenced them using an aluminum iSeq. And you'll see we used 59 of them because 5 of them turned out to not be Campylobacter. There was 2 salmonellas, 2 E. coli's, and another not even an enteric pathogen. So those were thrown out of the study. And then we used a variety of bioinformatic analysis in order to look at these. So again, our main goal was to look and see, okay, how do these strains relate to each other? None of these were supposed to be from outbreaks. So what we wanted to know was, do any of them cluster together, suggesting that they actually were a common source? So this is a minimum spanning tree, and this is based, so the first thing we did, and I won't show you the analysis, but we did the core genome. So we took all of the genomes and we compared it together and we came up with the core set of genes that were shared among all those. So similar to the core MLST. In this case, we used a SNP analysis on it instead of because it's a little bit more detailed. So it's kind of a between SNP analysis and core MLST. And so you can see here, and they're colored differently. So part of what we were looking at, as I mentioned, was looking at clinical manifestations. So those that are colored blue had just what was the patient said, just diarrhea. Those that are colored red had bloody diarrhea. So we were trying to look to see, okay, is there a difference? Are we seeing, are they clustering together based on not only their relationship, but are we seeing a big cluster of groups based on, the different types of more severe diarrhea? And then those that are white, they didn't have a reported whether it was bloody diarrhea or regular diarrhea. So you can see a couple of things with this minimum spanning tree. You can see a couple of clusters. So these in here, in here, in here, these in here are all suggesting that they are part of a cluster. The other thing you can see is that you're not seeing all the red in one tight group. We're not seeing all the blue in one tight group, which suggests that, okay, there isn't anything very distinctive that says, oh, this is going to cause a bloody diarrhea or this is going to cause regular diarrhea. But we'll get into that a little bit more. So looking at it, we then did a phylogenetic analysis of this, again, using that core genome. And so you can see this, but obviously I knew you wouldn't be able to read this. So I pulled a couple of key things out here. That group of, going back here, that group of 5 right down here is this group right here. So phylogenetically, these all are exactly identical to each other. So, and then there were, again, we identified a total of 8 clusters with at least 2 identical strains in them. So then what we did is we went a little further and said, okay, what is the core genome MLST? Do those sequence types match? 3 of those clusters had identical core MLST sequence types. So then we went back to the epidemiological data and said, okay, are any of these from the same family? Because it's possible families all eat obviously the same meals, same foods. So those aren't considered an outbreak. We're looking for those that were between families where they're eating, they could have eaten a common source, but they're going to be from at different times and things like that. And we did find 2 of these clusters came from unrelated persons. So that identified, and this group of 5 was one of them. There's another group of 2 in there. So the other thing that is known when it comes to Campylobacter is that for every 1 case that's identified and you get a culture, 29 cases go unreported. So you take a group of this where there's 5 strains, multiply that by 29. This was probably an outbreak of almost 150 people that got sick. Just the mass majority of them didn't report the disease. They just kind of wrote it out and got better. So it suggests that there are actually some large outbreaks going on of Campylobacter, at least particularly in the Denver area, but would suggest elsewhere, and they're just being underreported. The other interesting thing is the top one that we saw. So some of these, as we were going through them, we were looking at the colony morphology of the bacteria themselves, and we noticed that some of them had different, what looked like, appeared to be different Campylobacter strains based on the colony morphology of them. So we pulled those out and we sequenced those 2. So they came from the same patient, but they were 2 different. Most of them turned out to be the same strain, but we did have one group that came from a man. And so this 316 and this 317, which are in different clusters here, came from the same patient. So this suggests is that this patient actually had 2 different strains of Campylobacter. So he had a polyclonal infection, which we're starting to find more and more now, actually, with Campylobacter, that a lot of people have polyclonal infections if you dig in deep enough with them. So this was the first time we'd kind of seen this. So it just kind of shows you some of the things you can start to, the questions you can start to look at. Sometimes we didn't go in looking for polyclonal, but this kind of raised another question that we're now investigating further from this. So this just is a diagram of the core and accessory genome. So this is what was used for that analysis. So the blue little lines, each one of those represents that strain. So along the X there is the strain name. And then as it moves up, it's just different gene families. This is actually an HTML that I took a screenshot of that gets generated by this analysis. So you can actually click on each of those blue bars and it'll tell you what that specific gene is. So each blue bar that's there says that that genome, that strain in its genome has that gene. So the bottom along there where everybody has it, that's the core genome. And then you can kind of quickly look and see the differences between these and all these accessory genomes here. So kind of this along here is kind of considered the core genome. This, and I don't know if you can see it, but there's a little yellow line that's drawn across there. Anything below that's considered core. Anything above that's considered accessory. And so you can kind of get a general idea. It's almost kind of like, it is a DNA fingerprint, but it's almost kind of like, you know, pulse field gel and all that to kind of see how they kind of relate. You can see how some of these are very similar and share these. This group over here they are different than the rest, and you kind of see how their core genome dips. And they're actually part of this group right here. These are actually coles. So the rest of these were Campylobacter jejunus. These were a different species, well, all of them causing disease. But so that makes sense that we're seeing a little bit. They do share a lot of genes, but obviously you can see there's a big dip in the core between these. So looking at some of the, and digging in a little bit to the epidemiological data, as I mentioned, one of the key things we wanted to look at was, you know, using the epidemiological data to kind of drive the genomic analysis and look to see, this is the kind of the first time someone had tried to do this and see, okay, can we figure out, okay, if all these people had bloody diarrhea, you know, is there some type of factor that's involved in that and such. So again, this goes to, so this is a Pca plot and this is based on the core genome. So this, it does a blast search and it compares every gene from each of the genomes across each other against each of them. And then it plots it on here based on a couple of different factors. And then you can see again, so the red is bloody diarrhea. The blue it's a little bit different on this one. So you can put multiple factors. So the circles are bloody diarrhea. The triangles are regular diarrhea. The red are people that had fever. So if it's a red circle, it's going to be somebody who had fever and bloody diarrhea. Blue is no fever. And then the genome size, the larger the symbol, the bigger the genome size for that particular strain from that patient. So what you can see, we were hoping that, again, these would cluster and you'd see all the same type of symbols next to each other, same type of colors, and you're not seeing that, which suggests that we're not seeing any distinctive factors from the genomic side that says, oh, this Campylobacter strain is always going to cause fever and bloody diarrhea in a patient. That isn't being suggested here by what we're seeing, which was disappointing to me because I was hoping we'd have this cool figure that would show that, but got to go with the science. And the way it also works is if you're not familiar with this, the closer they are, the closer they're related. So like those little group way up there, they're very distantly related versus those that are closer together. So then again, we started looking at some of the epidemiological data and taking a closer look at some of the genomic analysis. So we combining genomic factors. So we looked at genome size, virulence, the number of virulence factors in the genome, antibiotic resistance, and different mobile genetic elements like bacteriophage, integrated elements, and plasmids, and compared that against epidemiological factors using age. You know, was there something associated with, you know, young kids getting it versus elderly? Diarrheal manifestations, you've already seen examples of that with bloody versus regular. Travel, international travel versus travel within the United States. Fever, again, we've already kind of looked at that. Antibiotic usage. So as I mentioned, antibiotics are not typically assigned for Campylobacter infections. So we look to see, okay, is there a difference if a person was given antibiotics? Like, was that infection, typically you're going to assume it's more severe. So was that, there's something about the strains that way. And then looking at gender, males versus females, was there any difference between those that got, because females tend to have a higher rate, but a lot of that has to do with variance in the diets. So looking, just going through, we didn't find a lot, but there were a few that we did find. And so one that we did find was, well, in this case, there was no difference looking at genome size and differences in bloody versus regular diarrhea. So the reason we looked at genome size, because genome size, the bigger the genome, the more genes it's going to carry. So that would suggest if we saw something with bloody diarrhea where the genome size was significantly larger, that would suggest that it is carrying extra genes. And then we can go in and look closer and say, okay, what genes extra are they carrying? So in this case, we didn't see that. So same with antibiotic genes, the number of antibiotic resistance genes. There wasn't any statistically significant, which my p-values are covered by me on this, but but there wasn't any major differences between the number of antibiotic genes. In this case, I just put up some examples of it based on international travel. But there was a statistically significant difference for the number of virulence genes and those that were assigned or given prescribed antibiotics. The weird thing is, so you'd assume that you're going to have more virulence genes, that the strain's going to be more virulent, you're going to need antibiotics. What we actually saw is the reverse. Those that didn't get antibiotics had more virulence genes. I haven't figured out why that is yet, but that's the way it worked out, that it was. So it may be that there are specific genes that are more critical and require, then can drive antibiotic. It could just be that the doctors in the US were just trying to get rid of the patients and wrote them an antibiotic and got rid of them. So we're still trying to make a little sense out of all that. So we also looked at mobile genetic elements. There wasn't a lot of differences when it came to mobile genetic elements, but in cases with bacteria, for the non-bacterial genomics people in here, mobile genetic elements are how you move antibiotic resistance genes, you move virulence factors. So these move around a lot. They can change the bacteria and its ability to produce disease, colonize new sources very easily. So that's why we were looking at that. In this case, we were looking at the age of the patients. There wasn't any difference. But then when we started looking at specific mobile genetic elements, we did start to see a pattern. So in this case, this was one. So this is CGIE1, which is an integrated element. So it's a large, what they call a genomic island. So it's a big piece of DNA that can move very easily. And so when it does, it moves like 40 or 50 genes with it between these different strains. And so what we saw was this island was more common in those that had bloody diarrhea versus those that didn't. So that suggests that maybe there's something in this island that could be causing patients to have bloody diarrhea. So we're going through it now and looking at. So that's where my lab then jumps in and we start doing more hardcore microbiology and looking and doing some of the pathogenesis stuff. We also found, the colors disappear on that one. Island 3, so there's 4 islands, genomic islands in Campylobacter. Island 3 was found with international travel. So those that had traveled internationally had a significantly, you can see the p-value 0.002, higher chance of having island 3 in it compared to those that had not traveled. So again, we're looking to see, is this something that maybe is more common in other countries? And we do know the countries that the people travel to. So we're looking at some of the genomes of Campylobacter from each of those countries to see if that's something there. And same with island 4, with the age of the patients, there was kids seemed to get infected with and develop disease with those that were associated with island 4. This one I have no clue as to, and we're trying to figure out what this can mean when it comes to the age. So sometimes you get data and you're still trying to figure out why that pattern occurred. And this is the case with this one. So the other thing that we've done is called a genome-wide associated study. And so each of these dots represents, in this case, it's a Kamer. So there's a software program called Pca. It cuts up the entire genome into these little pieces called Kamers. So they're about 20 nucleotides long. And so it just does that across. And then what happens is you can go in and you can say, okay, all these strains were associated with someone who got, in this case, someone who got antibiotics versus those that did not. Or, all these strains were associated with bloody diarrhea versus those that did not. Each of these dots represents a Kamer that was identified uniquely to whatever group you were looking, in this case, antibiotic use. So then this is the genome across the top, and you can start to see these patterns, and you look for these clusters in here, and you can look at these genes, and these clusters all are statistically significantly, these Kamers are statistically significantly more associated with strains that came from people that were assigned antibiotics And you can then look at those genes and see, okay, what's in there. In this case, it actually turned out to be island 4 is part of that cluster that I just highlighted, but you can see there's several other clusters within there. We're still sifting through all this data. So antibiotic usage turned out to be kind of an interesting one when it came to genomics. The one I was really interested in is the bloody versus the watery, because that's something we've been studying, and I've been studying it since I was a postdoc. And it kind of turned out to be disappointing. I mean, we saw a little bit, as you've kind of seen, but when we did the same analysis here, you can see, the number of dots there really showing you some strong areas. Here was the bloody diarrhea. You can see that we didn't get much. So it's possible that there's just not much difference, and it may be more host associated, the microbiome and stuff. So these are things that we're looking into too in my laboratory to see if there are differences in the host. It's just easier if it had been in the bacteria because they're easier to study. So it doesn't always work out the way you want, but this is a similar type thing where we just broke it up into Kamers, bloody diarrhea versus watery diarrhea. And you can see there was one little cluster in there. And that's island 3, actually close to it. So we're sifting through all this data. So conclusions from this first study was that we did identify several unidentified outbreak clusters from Campylobacter present in Denver. We found a polyclonal infection in a patient. The number of virulence genes was suggested to have a role in patients being prescribed antibiotics, with it being the opposite of what you would naturally think, that it went that if they were prescribed antibiotics, it actually had more virulence genes than those that didn't. There were specific mobile genetic elements of Campylobacter jejuni associated with patient demographics and or clinical outcomes, and specific gene clusters associated with patient demographics with the Kamer analysis. So shifting gears, so this is the one where we were looking more at movement and kind of source tracking in the environment and among, so in this case, we were looking at Campylobacter strains movement between avian sources, which I said is kind of the common reservoir, versus mammalian sources versus environmental sources. And then how do these strains from different sources relate to clinical strains? So, are we seeing, are the strains moving? Basically, the question we're asking is, are the strains moving between birds and mammals in the environment? And if not, which ones are closely associated with those that cause disease? So the methods we use, these were tracked from Salinas, California, which is nicknamed the salad bowl of America. A vast majority of the vegetables grown in America are grown in Salinas, California. So they were out there tracking Campylobacter and a number of different foodborne pathogens. In this case, from 2011 to 2013, they were sampling water samples. They were sampling produce samples, although produce is not something Campylobacter typically is associated with. It can cause disease. But they were also trapping animals. So they were trapping birds, sampling them, letting them go. Trapping, as I mentioned, they were getting cattle samples because there are also cattle that roam around in this area. But also then trapping feral pigs, raccoons, coyotes. There was a number of different birds. So any of the wildlife so U.S. Game and Fish was involved in helping with this. Anything that they could trap, they would sample and then culture. So in total, we had 62 strains that were collected from this 2-year period. 34 of them were from an avian source, 22 from a mammalian source, and 6 from an environmental source. And all the environmental sources are water sources. Again, we extracted the DNA, sequenced it with a MiSeq, and did bioinformatic analysis. So this is a phylogenetic tree based on 100 core genes. So it's a little bit different than the analysis where the other one we did, there was about 1,000 genes that we used in this one. This one we're using about 10 times less. But it still tells you the similar type picture. I've got it here. So this one, those that are in the box, those are all the avian species. So you can see right away that the avian species and everybody else are the mammalian and the environmental samples. So it shows that the strains that are in the birds are very different than the strains that are in the mammalian and the environmental sources. And you can't see it real clear, but a lot of these in here too were some clinical strains that we picked up we just used genomes that were publicly available. And you can see all the clinical strains also clustered with the mammalian and the water sources, suggesting that not only did the wild birds have their own strains, but they were not strains that were being passed on to humans and causing disease using this analysis. So looking a little closer, we saw some other interesting things. So this is a genomic comparison. The colors in the rings, each ring in here represents one of the genomes. So everything over here, this kind of shows you represents the core genes that are present. So these are shared among most of the strains. And then you've got the accessory genes. So it's a little bit different way of looking at the similar one where I had the square graph. Then each of the colors on here represents a different source. So you can see we had clinical sources are the dark blue, water sources are light blue, coyote, Canadian goose, feral pig, goats, raccoons, cattle, starlings, blackbirds, and American crows were all the wild birds that were in there. Each of those has their own colors. And so this gives you a quick, you can start to look at it, and you can, again, this generates an interactive, and you can click on each of those and see what the specific genes are. So some of these you can see, but you can use these also to identify clusters that may be present in one of these strains or not. The other thing that this does is this breakdown here. So this is a average nucleotide identity. And so this compares each of the strains against the other strains and compares the percentage of the average nucleotide identity across the entire genome. So those that are very closely related will share a lot, and they'll be darker red. So what you can see here is a number of different, you can compare. So these are a lot of the mammalian and clinical strains. So again, you see that they're closely related to. This is a cluster of the wild birds. The other interesting one is this is a cluster of wild birds from starlings. And if you look, they're not closely related to any other strains in there, which is why the whiter it is, it disappears. And further analysis, I'm not going to go into details of this, but what we believe is that the ones coming from starlings are actually a new subspecies of Campylobacter. Based on, they also have, for the biochemists in the room, the Antner-Duderoff pathway for glycolysis, which Campylobacter normally doesn't have. But all of these strains have that. So that also is a suggestion that there's something very unique about these birds, and it's only seen in starlings so far. So we're not sure why, but that's the fun of the investigations. So we're digging into that in more detail. So a couple more here, looking at virulence factors again by source. So you can see here that, again, the birds were not associated with clinical disease, and this is kind of supported by the number of virulence genes that are typically seen in birds. You can see they were statistically significantly different than those that were associated with clinical disease. They're in the light pink. Environment and mammals also were statistically significant differences across those, comparing between them. But there was even within these mammal and environment, there was a difference in the virulence factors compared to the clinical strains. So again, this is just looking at pathogenic protein families. Here you see a strong association of all these different proteins that are associated. So they're not necessarily virulence factors, but they're associated with pathogenic bacteria. So certain pathways and things that are associated with helping a pathogen, survive in a host and things. And this is where you see a major difference where clinical, environmental, mammal, very strong, that they're all very similar levels, whereas birds, you see this giant crash suggesting, and again, birds are the natural reservoir. Campylobacter is happy in there, doesn't need a lot of pathogenic proteins to try and survive in that environment, whereas if it gets into a mammal or the environment, it's more likely and can pick up more of this, and that will cause it more likely if a person's exposed to this to get disease. And then looking at these mobile elements again. So these are, again, looking at a couple of plasmids. So PVIR, PTET is a tetracycline resistance plasmid. So we did see a little bit of a difference between clinical and birds and carrying PTET, which kind of makes sense because they're not exposed to tetracycline if they're in birds. But again, looking at Island 3, again, turned out to be one of the interesting ones. in that the birds most commonly carried island 3 versus those clinical strains and some of these others. So again, that suggests that island 3 may have a significant role in surviving in the avian host and not as much in causing disease or surviving in the mammalian host. So conclusions, Campylobacter strains associated with wild birds were different than those that were associated with mammalian or animal or mammalian and environmental samples, and also were not associated with clinical disease. Certain strains of Campylobacter jejuni associated with wild birds appear to be potentially a new subspecies from starlings. And those associated with birds had less virulence factors than other strains, which again ties together that they're not producing disease, that they're happy and just to survive within the avian host. And there were specific mobile genetic elements that were more associated with wild birds than other sources, particularly island 3. And this may be a critical component to surviving within the avian host that is not commonly seen when it gets into these other mammalian. So with that, I'll take any questions. I know I just threw a lot at you guys that you're like, what is he talking about? So I'll be happy to answer any questions to try and clarify.
Sir, why was Campylobacter jejuni chosen? Why not any other bacteria? Is it that common in Colorado?
Yeah, well, so it goes back and forth with Salmonella as the #1 foodborne. It was chosen because it's what my lab favors. I mean, we work with Salmonella too, but we just had an opportunity, partially because we wanted to look at, there were starting to be more and more evidence. Denmark had done a big study suggesting that there were actually more outbreaks than were. So we had this opportunity with Colorado, and we wanted to look and see, okay, is it actually that true in the US too?
Because we come across Salmonella and Shigella more often than these kind of outbreaks.
Yeah.
So that's why.
Yeah, we're doing some additional things. I didn't want to kill you guys with boredom on all these things. But we're doing some similar things with Salmonella, looking at, outbreaks and things and tracking along those lines.
And
Any
each
other?
strain probably would have different type of antibiotic resistant genes. So would you suggest some limit, the number of samples that we have to analyze to actually come to a conclusion that these many number of resistant genes or these many number of virulence factors are present in that particular bacteria?
The exact number, it varies from organism to organism. But you can put it in and run statistics and see if there's enough to get a strong statistical, analysis when it comes to it. All those bar plots were just done with R, and then the statistics run in RStudio. So, usually you want at least kind of 10 to 20 of each, but again, it's going to depend, you're not going to know unless you've done antibiotic phenotypes or something on them, what the gene profile is when you sequence them. So if you get lucky, they kind of break down. You need at least 10 per group to do a comparison and get pretty good statistics between it.
Hi, Kiri. So do you think to what extent the probability of getting false positives with some E. coli infection or other salmonella infection, because seropositivity, they look for some antibodies to O antigen or flagellin like that.
Sorry, I didn't hear the last part.
So to what extent you get false positives for like E. coli or salmonella or Campylobacter jejuni, because all of them are flagellated pathogens.
Right. So there can be quite a bit of false positives, particularly they're finding now that they've gone, they're moving in the US, they're moving, as I mentioned, to what they call CIDT, which is culture independent diagnostic techniques. So, so that's PCR, ELISAs, and those tend to have more false positives. Studies have shown that they, in some cases, particularly the ELISA tests, are, if I remember it, it had like a 25% false positivity rate. The advantage it has is that you can screen one sample in one shot for multiple pathogens. So you can look for all that. So compared to then having to culture for each of them. So yeah, there is quite a bit of rate of false. I mean, even when we cultured, as I mentioned, they didn't confirm that they were Campylobacter. They just said, oh, these look like Campylobacter, but 2 of them were E. coli, 2 of them were Salmonella. So even those that they cultured, technically they weren't Campylobacter. So they were false positive Campylobacters that were cultured. So, so yeah, it's still an issue. You know, I think it'll always be an issue that we get false positives.
Thanks.
Sir, I have one question here. So like you told Campylobacter jejuni, so it is antibiotic associated diarrhea, like it leads to the antibiotic associated diarrhea. So in avian populations, like what type of samples you have like collected? First thing. Second thing is like how we can use real-time PCR before going for the sequencing.
Mm-hmm. So it's not associated with antibiotics. It's just that you may get prescribed antibiotics if you're on Campylobacter. With the avian, it's just a fecal sample, so they carry it in their gut. So it's just a fecal sample. So in this case, they would trap the birds. They would basically do a cloacal swab and then release the birds. And then we would use that to culture. So, and then real-time, you asked real-time PCR, repeat the real-time PCR. Is it a good idea to do real-time PCR before you sequence? Yeah, it's definitely, I mean, you definitely, you know, if you're trying to do something along these lines, and it goes back to the false positives, you at least can narrow it down and try and minimize your false positives before you culture and do all that stuff, before you do the sequencing. That's a good way to do it. If you really want to go crazy, you can do shotgun metagenomics where you wouldn't do anything. You just take the whole sample and do that. But that adds additional cost, time, energy, analysis, and things like that. So we're actually working on that for Campylobacter diagnostics. That's part of what we're also working on in Peru. And can we use this? Because what we're finding, particularly in kids, young kids in Peru, is that they carry, in some cases, 3, 4, 5 different species of Campylobacter, even different strains. So they have, so shotgun metagenomics or something along those lines is the only way you're going to be able to identify that, at least now. Then we can build panels to do more, you know, RT-PCR and things like that to then screen for all these things. So it really just depends on your goal of the project. But if you're going for something like this, you definitely would want to screen with something like RT-PCR, then culture, then do the sequencing.
And what are other potential like samples apart from what you told stool samples for this jejuni? So what are other samples we can take for the, you know, like detection?
So what other kind of samples can you collect?
Other type of samples.
So in this case, you know, we had water samples that were collected. I mean, most of it's going to be for, at least for Campylobacter, it's going to be fecal samples. On different species of Campylobacter, you can do oral swabs. Upsilensis is, and I'm blanking on the other species. Those are carried in the mouth of people for Upsilensis, and I'm blanking on the other species, but it's carried in dogs. They carry it in their mouths. And so, so those are, you know, oral swabs, water samples, and fecal are the 3 when it comes to Campylobacter that you can sample.
And you have discussed about some proteins, pathogenic proteins and all, like among, you know, like different proteins, and it's a different, you know, statistics. So
yeah,
what proteins, and like it has been isolated from the fecal samples?
So in this case, we were using the genomics to then translate to the protein sequence and go from that. So we weren't doing any actual protein work. It's more that we're using the DNA. We can say, okay, this is this protein. Use it to translate it to the protein sequence and compare that way. So yeah, so we weren't actually doing any hardcore protein work from it. I stay away from my protein. I'm not, by any means, that's not my expertise.
Can probiotics be used to combat this?
That's an excellent question. We don't know. It hasn't been done yet. but it's something that is being investigated. In fact, I know that in Peru, my collaborators there, I'm not part of that, but they are looking at it, particularly in these kids, like I said, and that part of the study we're doing is following these kids from basically birth all the way through the first 2 years of their life. And we're like 6 months into the study, and these kids have had, in some cases, 3 or 4 different Campylobacter infections in just 6 months. So that's one thing that they are also looking at is, can we use probiotics, particularly in these kids, give them, block it so that we're preventing it and they can grow and it prevents the growth stunting. So hopefully yes is the answer, but we're still looking into it. Any other questions? Everybody else is shell-shocked from all the, yeah.
Thank you. Very nice presentation. With reference to the food microbiology, Campylobacter, Helicobacter comes in a similar group. But when we see Helicobacter, mostly in the stomach infection, this is in the colon, right? In large intestine.
Sorry, can you repeat the last part?
No, with reference to the colonization or infection of Campylobacter, it is mostly in the colon region.
Yes.
Yeah. But initially, Campylobacter and Helicobacter, they were classified in the similar group.
Yes.
Now, how they differs with reference to the virulence or the pathogenicity?
How do they difference in their virulence between them? So one of the big difference in Helicobacter is its ability to survive, obviously, in the stomach.
Yes.
So it has all these urease genes that allow it to buffer basically it creates its own little microenvironment in there by creating these ureases that buffer the pH of the stomach, and that allows it to survive. It burrows into the stomach lining a little bit, which is why then it eventually over long term can result in cancer. Campylobacter, so it doesn't go quite deep, Helicobacter, whereas Campylobacter, when it invades, can go into deep tissue. It usually isn't associated with sepsis, so it won't go deep in and get into the bloodstream, but it will go into deeper tissue, into the muscle and things like that. So it will go a little bit deeper. So that's one of the major differences, is that Campylobacter doesn't really have those ureases. Is probably the biggest difference. Like I said, whereas Helicobacter is perfectly fine in the stomach acid, Campylobacter is the complete opposite when it comes to that.
And do you see the genome plasticity, what we usually see in the Helicobacter?
Yeah, there's a lot of genome plasticity in Campylobacter. Okay. It's very, very flexible. So
how is this genomic islands of virulent genes in avian sample and then sample from the clinical origin?
Yeah, so the genomic islands, from a virulence perspective, the short answer is we don't know. These were identified maybe 6 or 8 years ago, maybe a little longer than that. And they haven't been investigated to see, okay, is there something specific? That's why I'm trying to use all this, because I'm interested, because I think there's a lot going on there with those islands, but I'm trying to use it to narrow down, because each one of these is anywhere from 30 to 60 kb. So you're talking about one will have 30 genes, one has 60, a couple of them are about 50. So you've got hundreds of genes to look at to really characterize it. It's quite, so I'm trying to get it down to that narrow and hopefully hit a home run and find something that's in there. There's definitely something in there. But what it is, we don't know. The one thing we do know is that it contains a bunch of DNases that help prevent, well, one thing is island 4 makes it a pain. If it has island 4, you can't knock genes out because it produces all this DNA, so that when you try and induce the mutation, it chews up the DNA before it can get in. But other than that, we haven't learned much. So.
And what determines the host specificity, especially in the
host specificity?
Yes.
Yeah, that's another one that we're looking at, because like I said, we find it, in all these different environments. But it appears, we're starting to see that there are, differences between birds and that. We have another study we're trying to get done and published where we're actually sampling bears. So black bears in the US, they carry Campylobacter too. Most of them have normal jejunum, but we actually found a subset of bears that are carrying what appear to be a new subspecies of Campylobacter that are only found in bears. So, and they too have the enterodeutoroth pathway. similar to the ones that we've seen in Starling. So we're starting to see as we're sampling these that there is something to host specificity. What it is, we're just beginning to understand with it and look into it. But it's something we are, like my group and a number of others, are starting to actively look at too.
And most of the antibiotic resistance which you see in a clinical sample, is it because of the antibiotic usage or otherwise also a Campylobacter has the natural resistance genes or what you can say?
So it's suggested that the antibiotic resistance is due to the poultry industry. And that the fact there are studies, and we're doing a study in Peru, part of that study we're looking at those clinical isolates that have high resistance compared and where are they coming from. Because in Peru, they get their poultry sources from two different sources. They can have backyard where they raise them themselves, or they can get them from an industrial, larger facility. So if they're in the city that we're studying in, it tends to come industrial. If they're more outside the city in the suburbs, it's more agricultural, and so they raise their own. And what we're seeing is it's the industrial where they shove handfuls of antibiotics into the chickens as they're raising them. That has a higher risk, a significantly higher risk of Campylobacter, the clinical isolates having a high resistance, suggesting that it's all these antibiotics that are being pumped into these chickens. Similar things have been seen in the US and in Europe where it's been studied. So it's all comes down to agriculture. In the US, we're terrible with it. I mean, they abuse antibiotics left and right in the agriculture industry. And it's become this hot button political issue. It should have been banned years ago, but it hasn't. So it's really definitely, with Campylobacter for sure, it's definitely the agriculture and the poultry and just the sheer volumes that they're using there.
Cultivation is mostly anaerobic nature.
Sorry, can you repeat it?
It's anaerobic bacteria.
Yeah, it's an anaerobic bacteria. Well, it's a microaerophilic bacteria. So just a little bit of oxygen to keep it happy, but it can grow anaerobically.
And mostly the transmission is from the poultry product only.
From the what?
Poultry product.
Yeah. Poultry is the #1 source in most cases.
What are otherwise the standard, because in US, most of this meat products consumption is not that cooked like what we eat here.
Yeah.
What are otherwise the measures which you take to prevent Campylobacter infection?
Yeah, that's it. Obviously we're not doing a very good job.
USDA's measures are
USDA monitors it. You know, but I mean, it's there. So they wash the chicken carcasses in a chlorine solution. We've done studies where it does reduce it, but it doesn't eliminate Campylobacter. So, you know, because it's there in such high loads when they slaughter the chickens and all that. And so it's a challenge. They've even tried where they process the poultry differently. So if a particular house of poultry is free of Campylobacter, process that first so that there's no cross-contamination. It helps, but it doesn't eliminate the problem. You know, it really, I mean, we've done, I mean, Campylobacter loves chickens. I mean, it loves birds, and it will colonize it like a wildfire. I mean, we've done studies in my lab where we take one chicken in a room, and they're in separate pens. So like a room like this size, and we'll have pens all over. We'll give one chicken Campylobacter. They're all free before. And 2 days later, every chicken in that room will have Campylobacter. So it moves like crazy. So we haven't come up with a solution. They've been working on a vaccine to try and prevent it, but nobody's even close, you know, because it's I mean, it doesn't cause disease in the birds, so they don't raise a very strong immune response. So, you know, those are the challenges. So.
Yeah, wonderful lecture. Anything from you? Any other clarification from the participants? Those who has to basically take care of such kind of infection.
See, I can't force you with lunch today, so.
Poultry products are very famous in India. Poultry meat is very cheaper.
Yeah.
Though I think this question is not valid in JSS. But similarly, the antibiotic usage, which is actually my laboratory is working on antibiotic resistance in lactic acid bacteria.
Yeah.
This question we will answer separately, how probiotics can be used. So from that angle, the information which is given by Dr. Kerry Cooper with reference to the Campylobacter and AMR, distribution in a different ecosystem, host-specific gene locus, the different diseases, the bloody diarrhea, watery diarrhea, and fever. Again, very common, even in Indian context. The virulence, which I was more concerned with the genome plasticity. As you have explained, and Helicobacter has such a, labile genome plasticity, which we see. And how the strain specific difference. And then the last two questions which you have shown, that how from the avian to the environment and then clinical sample, the distribution with reference to the virulence, the antibiotic resistance, and the pathogenicity also. All these areas you have covered. I'm sure that participants got benefited with your lecture. So once again, on behalf of the organizers, so I'm actually, my session starts from the 10:15. I'm a little bit the early guest here. I thank you profusely for your nice presentation. Thank you.
Thank you for insightful information, sir. Thank you, Dr. Akila Prashant and Dr. Prakash Alami, to sharing this session as chairperson. We will now have a tea break for 10 minutes and let's gather again for the next session.
Good.
We will now begin with next session on modular exercise. I invite Dr. Rashmi P. Mahale, Associate Professor, Department of Microbiology, JSS Medical College, and Dr. Prakash Alami, Chief Scientist, Molecular Biology and Biotechnology, CFTRI, to coordinate this session on modular exercise.
Good morning, everybody. I'm Dr. Rashmi Mahale from JSS Medical College. So the last 2 days, you have learned the basics of epidemiological techniques. You have also learned the practicality and usage of this epidemiological techniques which have been taught to you for the last 2 days. So now we will just see how we can translate this or how we can put this into action, the knowledge which we had for the 2 days, how we can implement that knowledge into practical scenarios. So for that, we are going to have a modular exercise. Okay. So now this, we'll just see what the exercise is. So this exercise is to solve a problem which has occurred in a hospital. So now we'll see what exactly is this problem which has occurred in a hospital. This is a hospital setting. So on June 13th, a patient was transferred from a hospital A to the ICU of National Institute of Health Hospital, and he was discharged on July 15th. So here what you need to keep in mind is the patient was admitted on June 13th, and it was not his first admission. He was transferred from hospital A to the present hospital, that is NIH hospital in Bethesda. Okay, and he was discharged on 15th July. So he had a very long duration of stay in the hospital. So patient was found to be colonized with carbapenem resistant Klebsiella pneumoniae. So when they investigated the patient, they saw that the patient was colonized with carbapenem resistant Klebsiella pneumoniae. Now Klebsiella pneumoniae, as you all know, is a pathogen which can cause variety of infections in humans. So they found that it belonged to the genotype or sequence type 258. So this is the most common genotype which was found across USA, and that was one which they have isolated from the patient. Okay, so now patient was immediately, so now the infection control team of the hospital was sent word, and they said that the patient is colonized with this carbapenem resistant Klebsiella pneumoniae, and they initiated a contact precaution. Okay, contact precaution, barrier precaution, which included contact isolation. So this patient, they did not want it to be a source of infection to others, so they did a contact isolation. They isolated the patient, wherein the visitors or the staff who had to tend to the patient had to wear gloves and gowns before they could enter the patient's room. So this was what was done to prevent an outbreak. So in spite of all this, a second patient also was Klebsiella pneumoniae was also isolated from a second patient on August 5th. So the first patient had already discharged from the hospital, but a second was isolated from another patient on August 5th from the tracheal secretion. Okay, so now we had two patients. One patient was already discharged, but he already had a organism which was of concern to the hospital. So we had two, and we wanted to know whether this was a start of an outbreak. Okay, we wanted to know. And another thing which they noticed was patient A and patient B, or the first patient and the second patient, were never housed together. So they were never had any contact, or they never had any interaction with each other. So now why this Klebsiella pneumoniae, KPC Klebsiella pneumoniae had so much of concern in the hospital? So these Klebsiella pneumoniae are gram-negative organisms which are associated with a variety of infections in the hospital setting, and they are also a part, they are called as the enteric organisms. They're also a part of the gastrointestinal. There can be a normal commensal in the gastrointestinal tract. So they colonize. So when they are part of the normal flora, they also can colonize other patients who are there. So if you have a resistant strain, that can be transmitted to another patient, and it can result in transmission of the resistant strain to other patients. So this colonization can occur in various niches in the patient. It can be the oropharyngeal secretions, it can be the tracheal secretions, it can be the gastrointestinal tract, or it can be the rectal mucosal secretions. So all these are the sites where this organism can be lodged as a colonizer. So this may cause severe infections. As I said, variety of infections can cause pneumonia, it can cause bloodstream infections, and a variety of other infections also. It can be a surgical site infection, it can be a wound infection, so they can be associated with any type of infections. So it is responsible for roughly 15% of gram-negative bacterial infections that occur in the ICUs of the hospital. So that is the burden what we have. So now, so what? There are so many other enteric organisms, they also can be colonized. So why are we so much bothered about this? Carbapenem resistant Klebsiella pneumoniae. So why are we so much resistant? So carbapenem is an antibiotic which is usually used as a reserve drug in the hospital. So it is one of the higher antibiotics which are used in the hospital. So we have this particular organism producing an enzyme which is going to inactivate the activity of carbapenem, which is a high-level antibiotic which is used in the hospital. So that was of concern for us, because if the organisms start getting resistant to carbapenems, then we are not left with much options for treatment of the patient. So we have left with only might be tigecycline, colistin, so higher antibiotics left out. So we do not want such a scenario to occur, because even the last resort which we have in the hospital for treating infections might be used irrationally, and there might be development of resistance to even those antibiotics. So we do not want such a scenario to occur in the hospital. So we are very worried about a carbapenem resistant organism being present in the hospital, isolated from a patient from the hospital. So now we don't want that to happen. So this carbapenem resistant Klebsiella pneumoniae, in the US, it is mostly secreted by a gene which is called the carbapenemase enzyme, is secreted because of the BLA-KPC gene. If the organism possesses this gene, then the organism is going to be resistant to carbapenem antibiotics. So 70% of the infections are caused by one genotype, that is sequence type 258. So this is the sequence type which is most commonly associated with carbapenemase production in the US. So now we'll come back to the problem. So we had a carbapenem resistant Klebsiella pneumoniae which was isolated in our hospital. So we had a problem which is going to further, so we had two cases. First, from the patient who was transferred from other hospital. Next, a patient who was admitted in our hospital, and he also developed the we isolated carbapenem resistant Klebsiella pneumoniae from that particular patient. So now, was it the end? No, it did not end there. So next few weeks, we saw that there was one new case of either colonization or infection. So the infection control team in the hospital also tried to search for colonization of the organism in probably healthcare workers or the hospital environment, so that appropriate actions could have been taken to contain an outbreak. Okay, so now they saw that every 1 week there was a new case of either active infection or colonization with carbapenemase producing Klebsiella pneumoniae in the hospital. So this lasted, the first case was in June, and the last case what they saw was in December 2011. Okay, so from December to June to 2011 to January 2012, they had around 17 cases excluding the first case, which was the parent case. So total of 18 cases were isolated with carbapenemase producing Klebsiella pneumoniae. So out of those 17 patients, 10 patients died in the hospital. So now death of 10 patients, out of the 10 deaths, 6 patients died because of infection. So infection was due to carbapenemase producing Klebsiella pneumoniae. So this was of grave concern to the hospital. So now do we consider this an outbreak? So we'll divide this group into two halves. So now this, you have to just tell me, now do you consider this as a outbreak? So madam, morning itself she has told what an outbreak is, isn't it? So now do you consider this as an outbreak, or do you want some more information from me to consider this as an outbreak? What do you suggest? What do you think it is? Is it an outbreak? It is an hospital acquired infection, but is it an outbreak? Okay, so anything here from this side? Do you want to consider this as an outbreak? What information you want? Source, you want to find a source of infection. Okay, then we had one patient, and then we had 17 more cases of carbapenemase producing Klebsiella pneumoniae. So do you want to consider this as an outbreak or not? Signs and symptoms when they are either infected or colonized. We are afraid of both. We don't want the patients to be colonized also. We don't want the patients to be infected also. We don't want our healthcare workers to be colonized. We don't want our hospital environment also to be contaminated with this particular resistant organism. So it is of very concern, lot of concern to the hospital staff. So now we'll go on to see what are the requirements for you to say that this is an outbreak. So this may be an outbreak. We are not very sure still. So what else do we require? What other information do I require to say that this is an outbreak? So now if my hospital has always been having infections due to Klebsiella pneumoniae, which is carbapenemase producing, then I will not have much concern about it because it is already there in the hospital. So we have to review the background rates of carbapenemase producing Klebsiella pneumoniae in that particular hospital. So if my hospital has got this infection already in the hospital prevailing since long, then occurrence of this, I will not accept it as a outbreak. Okay, so we need to review the background rates. So that one information is required for us to say whether this is an outbreak or not. Next, we need to, as she said, we need to trace locations, patient locations in the hospital. So we need to have a source from where, if it is an outbreak, then it has to have originated from some place. So we need to know from where the patient, the organism has been started, it's initiated, the outbreak has initiated. Next, what we need to do is to know how it is transmitted across the hospital from one patient to 17 patients, 18 patients. We need to type the organisms and to know how the transmission link is happening. So now, review background rates of this resistant organism in that particular hospital has revealed that it has never been isolated before in the hospital. So if it has never been isolated in the hospital, it means that it is an outbreak. It is an outbreak, right? So in this this is an outbreak. So now is it enough if we know that this is an outbreak, or do we need to do something more? Mode of transmission, yeah, we need to know. So for that, why do we need to know? It is in the hospital. From where it is originating inside the hospital is our concern, isn't it? So now we want to know. So there were 17 patients who were infected with this particular Klebsiella pneumoniae. So now we want to know whether it was from one particular patient, whether it has spread to the rest of them, or there are multiple sites from where we have isolated, from where this organism has been transmitted to various other patients. So is this an outbreak caused by a single circulating strain of Klebsiella pneumoniae, which is carbapenemase producing, or are these multiple isolations of different strains of that particular strain from unrelated patients in the hospital? So you have one patient 1 coming from another hospital, patient 2 coming from another hospital. You want to know whether it is like that, or is it originating from one patient? So if it is coming from various sites within the hospital, then our outlook of how to contain the spread of the infection or the spread of the strain will be totally different from when it is originating from a single source. So all of the KPC strains, KP isolates were genotyped by, what is this, MLST, multi-locus sequence typing, and were found to belong to S2 258. So in the previous slide, I told you sequence type 258 was the most common sequence type which was associated in US, okay, for production of carbapenemase. So this was the most common genotype. So it was somewhere from within, okay. So the investigators assumed this cluster of Klebsiella pneumoniae isolates represented an outbreak, and because it had this belonged to the same genotype, by doing a MLST, they came to know that the genotype was the same, sequence type was the same, okay. So is it enough for the hospital to stop at this? Yeah, all of the strains are of the same sequence type. Is it enough? So now we had 17 cases where we did not have even a single case. So we are worried. So should we stop at this time, or should we proceed further investigate this to know the transmission link of the outbreak which has happened? So we need to further investigate, isn't it? So now why we need to investigate? To contain, that is the main aim. We need to contain the spread of the strain. So we should investigate this because there might be a point source. So there might be one person who is harboring the strain, okay? It might be a patient, it might be a healthcare worker, it might be a medical device equipment, it might be contaminated medication, it might be IV solution, or it might be present in the hospital environment. The bed railings and surfaces, other surfaces in the hospital might be contaminated with this particular strain. So we need to know whether it is a point source. So if it is a point source, if it is from a healthcare worker, probably we can shift the healthcare worker from patient interaction so that we do not further spread the organism from disseminating inside the hospital. So if it is from a patient, we can cohort the patient, like contact isolation, what I told you before. He'll be put in a separate room, okay? Or if it is a medical device, then it has to go to extensive cleaning, okay? And then if it is contaminated medications, it has to be discarded. If it is IV solution, again, it has to be discarded. If it is environment, again, the hospital surfaces has to be cleaned thoroughly, might be with the most appropriate disinfectant available. So now exposure to a point source might still be occurring. So now we know that it is there, we need to contain. If we do not do that, the outbreak will continue to happen. So we need to stop that, isn't it? So exposure to a point source might still be happening. So it is very important for us to find the point source of this particular isolate. So there might be a laboratory contaminant. So the laboratory itself might be contaminated. So all this are from culture, isn't it? Culture of the tracheal aspirate, culture of the blood, culture of other environmental samples. From there we are going to get this. So the laboratory, microbiology laboratory is going to tell us that there is a Klebsiella pneumoniae which is carbapenem resistant. So now might be the organism is there as a contaminant in the laboratory. So we have to rule out that. So it might then, if it is so, it might be a pseudo outbreak. So might be the lab is reporting not correctly. It's not correct. The correct way of reporting is not happening from the laboratory. So there is a false alarm of pseudo outbreak. So should be differentiated from point source, from multiple isolations, from unrelated patients, because the way of containment is going to differ. Okay, now how do we contain? So I said containment is very important. So how do we contain or control the outbreak? So this, as I told you previously, cohorting of the patient. So the patients have to be separated from the rest of the patients. So if it is possible, we can put them into a separate isolation room. If not, in the same ward, we can cohort them to one side. And we have to make sure that strict barrier precautions, contact precautions are adhered on to by the healthcare workers who are tending to the patient, and strict hand hygiene compliance should be there for the patients and also the relatives who are coming to talk to the patient. Then to identify asymptomatically colonized patients. So now we know. So there are some healthcare workers who are tending to the patients who have got infected or colonized with this particular strain of organism. So the organism might have transferred to the healthcare worker. So now asymptomatic colonization has to be screening of the healthcare worker and other patients has to be done. Okay, so that if they are found to be colonized, proper actions has to be taken. Okay, now coming back to our problem. So what would the main objective of our investigation of this controlled outbreak at this point be? So what will be our aim? What do you want to do now? What do you want to do? What tests do you want to do? Now we have done MLST, we have found that it is from the same serotype, the sequence type. So now how are we going to, what else do we need to do to know the transmission link? So the aim of the investigation was to establish transmission link. So that is very important to know the transmission links between patients, and also to search the environment for the contamination with that particular strain of Klebsiella pneumoniae, which is carbapenem resistance. So now performing new investigation. So MLST molecular we have already approached, and we now know that the sequence type is same. So now we need something new to give us more information about the transmission link. So new investigations are put forward to establish the transmission links. So identify possible environmental sources and map patients. So along with the new investigation which we are going to inculcate to investigate this outbreak, we also have to have a possible, we have to screen the asymptomatic colonizers, and we have to screen the environment, and we also have to map patient locations in hospital and overlap of hospital stay. So like she said, the patients might be overlapping. So the patient 1 might be besides patient 2. So if they are from the same ward or from the same ICU, so then you can say, yeah, the transmission has occurred here. Okay. So along with that, we need to do a mapping of location of the patients. So now by doing all this, we need an epidemiological curve for first isolation of KPC. So we'll see what this curve in this particular investigation was. So here, the first isolation was on 12th of June. The last isolation was on 18th December 2011. Clear? So this is the epidemiological curve, and here so in between you had 18 so this is the first isolate, and after that we had 17 isolates of carbapenemase producing Klebsiella pneumoniae. So this is the epidemiological curve. So now this is mapping of the patient. So where were the patients? So these were all the various locations, ICU, then might be the patients all with this particular strain colonization or infections were cohorted in one particular area, the cohort area, then ward A, ward B, so various wards. These were the various sites where they were present. But here these black marked lines, what you're seeing, they are the places where the organism was first isolated from the patient. Okay, so these were the first patient on day 1 or probably day 2, they screened him and they saw that he had carbapenemase producing Klebsiella pneumoniae. So here on a different date. So like that, this is the outbreak curve wherein the positivity of culture was noticed in these various 17 patients. So now understanding this transmission events between patients on the basis of epidemiological data proved extremely difficult. So we have everything. So we have the timeline, we have the patient mapping. So now can we make out anything in this? We cannot, right? We do not know where exactly what is happening. So what do we need to do? There was an extensive, it was difficult, though we had this basic epidemiological data, it was difficult for us to come to a conclusion where the outbreak started, from whom, was it from a one person, was it from multi, it was not possible for us to clearly define. So what we have to do, we need to have another method of typing. So we need to have another method of typings which will answer all our queries. So now how to assess the direction and chain of transmission events. So how we are going to conclude from where it started, how it transmitted from one patient to another. So how is it going to be done? Any idea? What can be done further? So from yesterday you've been listening to molecular techniques, isn't it? So what can be used to know the relatedness between various isolates? What test or what technique can you use to know the various relatedness between the isolates? So if you say isolate A, isolate B are closely related, then they are from the same source, isn't it? They are almost similar. So which is the technique which will tell you that they are related? Huh? Pulse field gel electrophoresis. Anything else? Whole genome sequencing. So whole genome sequencing is the test which is going to answer all, which is going to map our transmission events in the outbreak. Okay, now we have decided we are going to do a whole genome sequencing to come to a conclusion of how this this outbreak has happened. Okay, so now the questions which we need to answer before we undertake this investigation is, how many isolates are you going to include? How many isolates you want to type? Because typing is a whole genome sequencing is a expensive procedure, isn't it? So we need to know how many isolates you need to type. How to decide who was the first index patient and how to decide which isolate was acquired next in the transmission chain. So this is the whole genome sequencing is going to answer all these questions. The first one is how many isolates we need to type. So now how many isolates we need to type? We have one case. So from that patients we have to type, isn't it? So to know from origin of that particular, we know to know the genome of that particular, all the sequence type of that particular isolate, we have to do from the patient. So anything else you want to do? What about the rest of the patients? You want to? And one more thing, what I told you in the first slide, what you have to remember is the first patient from whom the resistant isolate was isolated had a prolonged hospital stay, isn't it? He might have been colonized previously. Only in this hospital we have screened him and we have found him to have this particular strain, isn't it? So now what can we do? What should we do? Is it enough for us to just screen, have that one isolate which has to be whole genome sequenced, or we need to have more isolates from the, should we screen the patient from various sites to know whether there is an heterogeneous, might be mutations keep happening in microorganisms, isn't it? So is it something that is happening in that particular patient? Because he has been in the hospital, in and out of the hospital for a very long time, isn't it? So now, is it required to do that, or is it sufficient to do only one isolate from the parent patient? It is required to do multiple samples. You need to screen the patient from multiple sites so that you to know whether there is an heterogeneous colonization by the patient. Okay, so now that's what has to be done. And then the rest of the isolations, so this was the first, so we consider this as a parent, ancestral, parent type. So now we have to see whether the other things are going to be related to this particular sequence type. So if it is related, then everything is in place. Okay. So how many times? Type at least one isolate from each patient. Afterwards, the first patient we want to do more screening because he had a prolonged hospital stay and there might be development of heterogeneous resistance, mutations might be occurring in the patient. So now how to decide who was the index patient? So see if an isolate has a genome sequence already deposited in a public database to compare. So now that's what they have done when they did the MLST. So they have compared it and they saw that the sequence type was similar to sequence type 258. Isn't it? So we have to first, when we isolate, we have to see, does it belong to anywhere near any particular genome sequence which is already deposited in the public database. Okay. So next, how to decide if it is so, then it becomes the ancestor of our particular isolate. So how to decide which isolate was acquired next in the transmission chain? So now this decision will depend upon looking for mutations. So you look for mutations from the isolate. Okay, mutations. They can be single nucleotide polymorphism or single nucleotide variants. Okay. So now this will tend to accumulate even otherwise also in the patient. So that's why we need to look for the mutations. Okay, which has happened. So from me, it is going to go to another patient. So in my strain, it was, might be I had genome A. Okay, so mutation A. So now it goes to another person B. So he'll have A, along with that A, he'll have genome B. Clear? So now there is this thing has to happen. So isolates with more SNP SNPs tend to appear sequentially. So sequentially, I had A, the next one will have A and B, the third one will have A, B and C, fourth one will have A, B and C. Probably one might get deleted in the course of time, but we should be vigilant to notice that there is sequential changes in the genome of the isolate. So the subsequent sequences should carry the same mutations found in the strains that precede them. So I had mutation A, the next one had mutation A, along with mutation A, he had mutation B. The third one will have mutation B plus mutation C. It's like that. So that will tell us, first, I was the one who acquired or was colonized with the isolate. Next was the patient B, where he had A and B mutations. Did you understand? Is it clear? Yes or no? Okay. Now, typing strains by whole genome sequencing. So now here in this particular outbreak investigation, they have taken the index patient, that is the parent patient, what I said. So from him, as I told, because he had a long hospital stay, they have screened him and they have isolated 7 isolates of Klebsiella pneumoniae, which was carbapenemase producing. Clear? 7 isolates were selected. So these isolates were cultured from 4 different body sites over 4-week stay in the hospital. So he stayed for 4 weeks in this particular hospital. So over the course of his stay in the hospital, they have screened various sites. Okay, 4 body sites they have screened and they have isolated 7 Klebsiella pneumoniae. Now, because the patient had been colonized for several months, so this is what I said, there can be genetic heterogeneity. The patient is one, but in different sites he has different strains or different Klebsiella pneumoniae with different types of mutations. There might be slight mutations in the patient. So that's called as genetic heterogeneity. So they saw that there was genetic heterogeneity in the patient, index patient, from various sites. These isolates was examined to reconstruct the putative transmission events more accurately. So now this is the patient. Now we have 17 more patients who are infected with or colonized with that particular Klebsiella pneumoniae. Now we know that if we had not done this whole genome sequencing and if we had not tried to screen the patient for various other sites, we would have only ST258. Okay, we would have only that, and if we did a sequencing of the isolate from patient B, it would have mutated and we would have said, oh, this is totally different from this strain. So might be it is not at all related to each other, isn't it? So what they have done is they have screened. So now they have found that within that patient, there are various genetic heterogeneity. So there are slight variations in the isolate which has been isolated from the same patient. So if I have mutation A, I also have mutation B in another site, organism isolated from another site. So if a patient has A, B and C, so we should not be, where did the B go? So a link is missing. So it is not missing, it is there in the parent patient itself. Okay. So 7 nucleotide variants were detected in the patient, index patient. So now these are the 7 isolates. Okay. So now 1, 2, 3, 4 were similar. They did not have any much nucleotide polymorphism. Whereas in this and this, where you have this yellow asterisk, you see that there is polymorphism. So there were 7, 1, 2, 3, 4, 5, 6 and 7. 7 polymorphism were detected in that particular patient and different sites, organism isolated from different sites. So now in the parent patient, they have screened various sites. So the various sites, one of the site in urine, groin, throat. Okay. So these were the common sites, bowel. So these were the common sites which were screened for the presence of the colonization of this particular isolate. So now they saw that the groin isolate also contained one SNP that that was not seen in the bowel isolate. So now, which is the groin isolate? So this is the bowel isolate. So you have 3 genotype, means SNPs, and then this is the groin isolate. It also has the same one, but it has one different sequence. Okay, this was detected by doing a whole genome sequencing. So we saw that within the same patient, there was slight difference. So genetic heterogeneity was identified by the within the isolates recovered from the index patient. So this is just showing you that there was difference in the sequence. So this demonstrated the importance of culturing various sites and sequencing isolates from multiple body sites. So you will know that even this one also is from the patient, index patient only. This isolate also is from index patient only. Now this also is from the index patient. So though there is slight difference. Okay. So the next step was to perform whole genome sequencing of a single. Now we have done the index patient. We have screened him multiple sites. Okay. Now next we are going to screen only the isolate. We are going to do a sequencing for only the one isolate from the rest of the patients who have got this colonization or infection due to Klebsiella pneumoniae, which is carbapenemase producing. So now genomic comparison revealed a total of 41 SNPs amongst the 18 outbreak patient isolates. So there were 18 outbreak isolates, only 41 SNPs were observed. So this is, that is, the isolates varied in 41 sites distributed over their 6 million base pair genome. Okay, so that was the variation which we could find because we did a whole genome sequencing. Okay, so now when the whole genome sequencing of all the isolates which were isolated from all the 18 patients was done, along with the multiple screening from the patient, index patient, so they could come to a conclusion of clustering the strains into 3 clusters. So cluster 1, cluster 2 and cluster 3. So now you see that within cluster 1 almost it is similar. So they have lot of similarity, that's why they have been grouped into cluster 1. So next we have cluster 2, here the similarity is here. Okay, so now again that's why they were grouped into cluster 2. Next we had one isolate from patient 8. So here we had little similarity with 1, and then we had no similarity with 2, and then we had an extra mutation or extra gene sequence which had been occurring in this particular strain. So this was given a different cluster. It was designated to be belonging to a different cluster. Okay. So members of the cluster 1 and 3 share 3 variants. So here 1 and 3, they are sharing 3 variants. So 1, 2 and 3. 3 variants they are sharing. Okay. In patients, variants present in patient 1's bowel and groin isolates. So this is the groin and bowel isolates. So they are similar to it. Okay. So next, whereas the isolates in cluster 2 share 3 variants present in patient 1's throat isolate. So this one, they are sharing only 2. Okay, they are sharing 2 clusters which are, they share 2 similarity between the throat sample of the patient 1. There is throat sample of patient 1. So this is the throat sample of patient 1. So this is the cluster 2. Okay, they are sharing 2 similarity. Okay, so these data therefore suggest genetically distinct isolates from patient 1 were transmitted to other patients. So what we are incurring from here, we have genotype sequenced, or whole genome sequencing was done to all isolates, and we have seen that they vary slightly from each other. So though they are varying from each other, we still attribute it to the parent one, because in the parent one we have done multiple screening and we have seen what is the sequence type in those various isolates. So the data indicate that not only is patient 1 linked to the outbreak, but also 3 independent transmissions. So transmission has occurred from the throat, the transmission could have occurred from the bowel, and transmission has occurred from groin from the first patient. That's what we could make out by doing a whole genome sequencing. Is it clear? So we have seen similarity between the cases from the parent or the index patients isolate, which was isolated from groin, which was isolated from the throat, which was isolated from the bowel sample, that is bronchoalveolar lavage fluid. So that is what we could identify. Otherwise, probably if we had not done, we would still be searching for the origin of the variants which we have detected here. So all of the Klebsiella pneumonia cases likely originated with the index patient rather than by independently introduced bacteria. So that's the advantage of doing this whole genome sequencing. So though they were little different from each other, still we could attribute it to index patient because there was similarity in the various other isolates which was isolated from that particular patient. So now, along with this, they have done a survey of they have screened the environment and medical equipment. So once they have seen that, they have isolated the same organism, which was carbapenem resistant, from ventilator. Okay, so ventilator here. So here you are seeing. So you have one ventilator organism, which is isolated from ventilator. So that was also grouped into cluster 1. Okay, but it was unlikely the cause for the transmission to other individuals because this has a different sequence in this particular site, which is not seen in any of the isolates. So that was not attributed to the source of infection for the rest of the patients. Is it clear? So ventilator had been thoroughly cleaned after being used on patient 6. So this ventilator was used on patient 6, and afterwards when they screened or when they did when they took a swab from this ventilator, they found this organism, but they had cleaned it before they used it on other patients. But this was not a source for other patients because the isolate was totally different genotypically compared to the one which was isolated from the rest of the patients. So did the ventilator nevertheless serve as a source of infection to the rest of the patients? It did not, because it was different by the whole genome sequence analysis of the isolate from the ventilator. So I told you it differed from the rest of the isolates. None of that particular mutation or particular sequence type has not been seen in any of the isolates from the others. So the ventilator isolate had one variation distinguishing it from the patient 6 original isolate. So patient 6 had an isolate which was not there in the ventilator, confirming that the isolate likely survived the cleaning process and that it was not the source of infection to other patients. So now what do we infer? So the inference of most likely transmission route. So that is what was our aim at the start of this outbreak investigation. So distinguish among scenarios that were equally likely based on genetic data. So the investigators used patient stay overlap in hospital wards and proposed the most probable transmission sequence as the one requiring minimal length of undetected silent colonization. That means other than doing all these sequencing, we also have to screen the asymptomatic colonization which can be happening in the hospital. Okay, so now coming to the pros and cons of the different sequences which we have used. So here we have used whole genome sequencing. We had one case and then we had so many other cases, 17 other cases. So now if we had only the multi-sequence MLST, then we would be roaming around blindly. We're not coming to a conclusion, the epidemiological link. So the black arrows are showing inconclusive searching for the source and transmission link, whereas the red arrows are showing the epidemiological link once the whole genome sequences had been done. Okay, so suggested transmission pathway. So this is our index patient. So before 2, the patient 3 had been infected. Then we had 4, 8, and then there was the rest of the patients. So this was the one with the black, red shows the epidemiological link between the in the transmission chain of the outbreak. So with this, I conclude the advantages of using this whole genome sequencing as a tool of epidemiological typing to know the source of an outbreak. So this whole thing has been taken from an article which was published in 2012. So tracking a hospital outbreak of carbapenem-resistant Klebsiella pneumoniae with whole genome sequencing. So this was the article from which the whole exercise for this module was done. Okay, so if you have any questions. So this was just basically done to know the importance of whole genome sequencing in tracing an outbreak, to know the transmission link. See, implication for treatment might not be much. Okay, the within the same patient you have different niches having different sequence type. That is because mutations keep happening in all organisms. It is a process. It is happening in all organisms. So now, because his stay was there in the hospital, probably initially in hospital A, he had acquired this resistant strain. So there was a prolonged, as they multiply, they mutate. So that is the rule of law. So they multiply, rule of nature, that they undergo mutations. So now for treatment, it does not make much difference, but epidemiologically, it is significant to know what are the different mutations which are happening and do they have any consequence on patient treatment. There might be some, at some point, there might be some mutation occurring which might result in resistance to something else. Might be now I said now we have, for this carbapenem resistant organism, we have colistin and tigecycline, polymyxin B as the treatment. So during mutation, probably they become resistant to polymyxin. So we have to keep seeing. So that will help us when we are doing. Otherwise, it is happening in all isolates.
Madam, these mutations are random or?
They are random mutations.
But then if it is random mutation, how it will affect with reference to the virulency or the?
There might be, it might affect the virulence also. Some isolates, if the mutation is occurring in the ones which are required for the survival of the organism, or if they are required for the virulence of the organism, then the organism might become avirulent or might be not able to survive at all. They might die out.
What is the drug you prescribe now for the carbapenem resistant isolates?
We usually recommend tigecycline, colistin or polymyxin B.
But colistin resistance
is seen, sir. That is because of irrational use of antibiotics.
How is carbapenem resistance now is severe in India? Because one of the link which you have missed perhaps is the NDM1. You know?
Other than NDM, we have other types also which are producing carbapenem.
Which basically detected in Acintobacter and then Klebsiella and several other.
No, so even in this one you have lot of resistance.
And I think from Indian context that can be a very good study because we have a large number of patient population and hospital visitors. During COVID time only visitors were not allowed. He was the only person who was visiting me when I was infected last year. But otherwise, our relatives actually likes to visit the patient with a fruit baskets and flowers and many other things. Is it?
Pune. Okay. Yeah. There might be, there all chances are there that they might mutate.
So the multiplication and the replication, how it works, how it happens. But here it is, I think, the antibiotic pressure, which is more of concern. And pneumonia is one of the major problem in the clinical setting. I think not much questions from the participants. Dr. Rashmi tried to take the participant on task for this modular exercise. She asked only this side of participant, no response from this side. But otherwise, I think with reference to the overall this lecture, quite informative. It would have been really nice. I think case study would have been from the Indian context somewhere. Maybe next time you can
yes
make this one and this ST2F258 particular strain. I think data and then history record of the patients is also another very important issue, which I think our nurses that tries to keep this record, but then still I think when it reaches the doctor, I think it misplaces or gets converted to some other information. And as far as in the clinical setting or the treatment is concerned, I think microbiology is the last subject to be viewed. But then all this infection stuff comes from the microbiology, right? And most of this pathogen actually again comes from the animals, veterinary field. I think from that angle, I think next session will be very interesting. Both chair, co-chair, speaker is also here. They are waiting for 11:15. If no more clarification from the participants, I take this opportunity to thank Dr. Rashmi Mahale for
thank you, sir.
For her very nice
Thank you for insightful information, ma'am. I request Dr. Prasanna Kumar to hand over mementos as token of appreciation to the chairpersons.
I thank the organizers for giving me this opportunity. Within a fraction of a second, I will be going now from here. Come on.
ವಾಕ್ ಮಾಡ್ಬೋದಾ? ಇಲ್ಲ ನಿಂತ್ಕೊಂಡ್ರೆ ಮಾಡ್ಬೋದು. I can walk. ಕ್ಯಾಮೆರಾ ಮೇಲೆ ಕಾರಾಗಿ ಇಟ್ಟಿದೆ. Thank you.
Now I request Dr. Matam Vijay Kumar to take over the session on practice of molecular epidemiology. I invite Dr. Prasanna Kumar, Associate Professor, Department of Biochemistry, JSS Medical College, and Dr. Ravindra PV, Associate Professor, Department of Biochemistry, CFTRL, to chair the session on practice of molecular epidemiology. I request the chairpersons to introduce the speaker, Dr. Matam Vijay Kumar, to the audience.
Good morning, everyone. This is going to be another enlightening presentation, which is most important in the context of interactions between the diet, gut microbiome, and the metabolic disorders, particularly in the context of immune system, innate immunity particularly. So I take the honor of introducing the speaker to the session. Matan Vijay Kumar is tenured professor in the Department of Physiology and Pharmacology, Medical Microbiology and Immunology, University of Toledo, College of Medicine and Life Sciences. He received his doctorate degree from CFTRI Mysore in 2001, then went on to complete two postdoctoral trainings from Georgetown University, Washington, DC, and Emory University, Atlanta. Dr. Matan has been the recipient of many awards and honors during his research career, like Outstanding Faculty Mentor Award from Penn State Postdoc Society, Dean's Award for Research Excellence, New Investigator for 2019, University of Toledo College of Medicine and Life Sciences, to name a few. He has also authored and co-authored in several peer-reviewed journals, including Nature Communication, Journal of Clinical Investigation, Cell Metabolism, Gut, etc. Currently, he is a member of several prestigious organizations like American Association of Immunologists, American Society for Nutrition, Society for Mucosal Immunology, etc. His research laboratory focuses on host gut microbiota interactions, whose deregulation may often precipitate into metabolic and chronic diseases such as obesity, type 2 diabetes, and liver diseases. His lab has successfully demonstrated that the loss of innate immune response, innate immune function in TLR or TLR5 deficient mice resulted in gut dysbiosis, leading to the development of spontaneous colitis and hallmark features of metabolic syndrome, including insulin resistance, hyperglycemia, hyperlipidemia, and hepatic steatosis. His discovery of the link between the gut microbiota and metabolic syndrome has earned him more than 1,050 citations so far. His ongoing studies seeks to understand what dietary practices could result in gut microbiota dysbiosis and under which the circumstances our gut bacteria serve as a friend or foe. With this introduction of our speaker, and I hand over the mic to the speaker, please.
Yeah.
Thank you, sir.
Yeah. Thank you, chairs. First of all, I thank each and every member of this organizing committee for the invitation. I just came yesterday I think Monday morning. I'm still in the form of jet lag. I could not sleep for last 3 days. If anything happens, okay, I will blame on my jet lag. Okay. So my talk is on, no, I think he opened wrong one. No. JSSJAD, I think I told you to remove it, right? JSS. You delete that one. Okay, sorry about that. Yeah.
So my talk is on gut microbiota in health and disease, how the host innate immune system can play a role in aggravating the disease. So I see some women faces are sad or some with question mark, because sometimes the media persons, they are biased. Okay, microbes make the man. Okay, microbes make the woman also. Okay. So I am not sure how many of you are aware of gut microbiota. So I prepared lots of introduction, so you will better appreciate my results. So basically, my talk is quite different from what you have heard already. It is a kind of a multidisciplinary. You can see biochemistry, microbiology, immunology, nutrition, physiology, everything. It is a mixture of everything. Okay. Not working. Okay. So far, bacteria belonging to 52 phyla have been identified on the earth, but only 5 to 7 phyla can colonize our GIT. GIT stands for gastrointestinal tract. Okay. And where they can form very stable and diverse community and form a productive ecosystem by helping each other. So one interesting part is the proteobacteria, which we heard already, Salmonella, Campylobacter, E. coli, they all belongs to proteobacteria. They dominate on the earth, but they are minor communities in our gut. Okay, if you see the length of our gastrointestinal tract, because this is one of the system which exposed to external environment constantly in the form of water or food. Okay, so we have almost 4.5 to 5 meters of our gastrointestinal tract, which is exposed to a lot of some toxic molecules or some antigens which are very allergic in nature. Okay, so this is the length. And you can imagine what could be the length of this GI tract in the herbivores and omnivores or carnivores. Okay, so I don't think you will be surprised if you look at the carnivore, they don't need very lengthy intestines, right? And the omnivores, they have medium length, whereas the herbivores, they have longest gastrointestinal tract, because these are the ones they depend on plant-derived complex carbohydrates. So they have to pass through this long GI tract so that it will get enough time to break down and also for the absorption. So all the mammalian intestines, including humans, which are lined by a single layer of epithelial cells. Okay, these are the epithelial cells, very typical colonocytes, and which are separated by a mucus here. Okay, and these are all the fecal matter. So don't think that GI tract is just tube. It is not just tube. It has a lot of division of labor happens there. Okay, it is not just for digestion and absorption. So at each region, for example, in the duodenum, only the iron absorption takes place. Okay, like that, there is a clear-cut division of labor. So it is the largest surface area is almost like a tennis court, which is a home for 4 into 10 to the power 13 zillions of bacteria belonging to 500 to 1,000 distinct species. The predominant phyla are Bacteroidetes and Firmicutes. In fact, we carry more bacterial genes than our own genes. So the person sitting next to is more bacteria than the human nature. Okay. So here we are talking only about the bacteria. We are not at all talking about the viruses or fungi or archaea and eukaryotes, because we do not have that machinery. But still, the people have done some virome in the intestine and also fungome or microbiome. Okay, but it is not many papers are on that. So the functions of this microbiota is comparable to that of liver. As you know, the liver is the major metabolic organ. Okay, they can influence bone density and also the lens lipid composition, which are very distant from the gut intestine. So gut microbiota is densest at the distal end of the gastrointestinal tract. Okay, in the colon, they are heavily colonized with all these bacteria. And previously it was called as microflora. That word is no more used in the literature. It is replaced with microbiota. Okay. So why do we care about gut microbiota? They were there for thousands of years. So now all of a sudden there is a rapid change or interest on this gut microbiota. The reason is we have developed a very sensitive instrumentation or technology to identify them by using a 16S ribosomal RNA sequencing. Okay, they use different platforms to do the sequencing of all those bacteria. So this slide is a typical textbook information. So gut microbiota helps in a number of ways. They help in the development of or maturation of gut-associated lymphatic system. They increase our food deficiency ratio, and they also source for vitamin K, biotin, and B12. Of course, this is absolutely true in the case of rodents, which are coprophagic. But I don't think they supply vitamin K for because we are not coprophagic, right? And they also are known to protect against enteropathogens. So this is data from our lab. It is from the mouse. So you look at the size of the cecum. Germ-full means conventional. In the microbiota language, we use the word conventional means regular or germ-full mice. Whereas germ-free, it is almost 5 times bigger than the conventional mice. So you can recapitulate this phenotype by treating them with broad spectrum antibiotic in 10 days. All the bacteria will be wiped out and the cecum becomes enlarged. Okay, this is a typical characteristic feature of a germ-free rat or germ-free mice. I have never heard about germ-free humans. Okay, maybe in future there may be germ-free humans. So another feature or importance for gut microbiota is the germ-free rats are neutropenic. Okay, they have very low numbers of neutrophils. So if you know about the immunology, how important this cell type is, okay? Like if cancer patients who are under chemotherapy, they die because of the neutropenic nature. They are susceptible to various bacterial infections. So once we colonize them with the bacteria, see the neutrophils goes from 10% to 38%. So the reason I'm showing this slide is how important they are, okay? So that means you don't need the live bacteria to go into the circulation and stimulate the bone marrow to make neutrophils. So this indicates that some gut microbiota derived product or gut microbiota derived metabolite can signal the bone marrow to make more neutrophils, okay? This is another example. So this is the conventional germ-full mice feces. There is a lots of IgA, that is immunoglobulin A, secretory IgA. Whereas in the germ-free, there is no IgA, almost undetectable levels. Once you put back the bacteria by co-housing or by putting some fecal feces into their, by oral gavas, you can bring back their IgA levels in the feces. Okay, these are the very typical features of germ-free mice. So how you can manipulate by sharing with microbiota from other animals. So gut is smarter than you think. So it is an anaerobic bioreactor because the bacteria express more than 3,000 types of glucuronidases. They can chew anything you can eat. Okay, and it is also regarded as a second brain because 95% of the serotonin, it is a second major neurotransmitter, is found in our bowels. How? Because the microbiota stimulates the tryptophan to enter into serotonin pathway. Okay, so that's why we often use the word gut feelings, but we never say kidney feeling or liver feeling. Okay, because we have a lot of serotonin in our intestines. And also it is a major immune organ. 70% of the immune system is directed against the gut. In fact, the intestine is one of the largest reservoir of macrophages. Okay, and also more than 30 gut hormones have derived from the intestines. Like those are called as incretins like ghrelin, GLP-1, GLP-2. Okay. I'm just giving a snapshot. If I talk about each slide, 1 hour is I need more than 1 hour. Okay, so gut microbiota is also helpful for maintaining the chemical library of gut and also systemic circulation. So in vitro silico techniques have identified 3,449 metabolic reactions predicted to occur in the gut, out of which 37% were uniquely performed by these bacteria or microbiota, because the host has no machinery to carry out the fermentation or generating secondary bile acids. Via bile salt hydrolase, this is uniquely expressed in certain types of bacteria. And they also generate indoles, which act as ligands for aryl hydrocarbon receptor, which is very much critical for gut homeostasis. Okay, so they can also promote or demote metabolic diseases. The best example I can give is the cisplatin or irinotecan. Do you know that drug? It is one of the most widely used anti-cancer drug. But unfortunately, after a few days, it has to be withdrawn from the patients because the patients suffer from severe diarrhea. The reason is when they administer cisplatin, it goes to the liver and it will be metabolized into cisplatin diglucuronide. If you go back to your xenobiotic metabolism, every hydrophobic drug will be added, hydroxyl group, and then to that it will glucuronic acid will be added. That makes it facilitate their excretion into the bile or urine. Okay, in this case, the cisplatin will be glucuronidated and then delivered to the intestine via gallbladder, where the glucuronic acid will be eaten up by these bacteria which express glucuronidases. Okay, then the cisplatin gets reactivated. Then it kills all the mucosal epithelial cells because they have the turnover of only 2 to 4 days. Okay, then that causes mucositis. So that is the major killer for the cancer patients, like chemotherapy or radiation. They kill the patient because of the mucositis. So with this metabolic potential, they have the ability to promote or demote metabolic diseases. Okay. So they also generate lots of signaling molecules. Earlier, they thought that they are waste products, but now they are very important molecules in signaling. For example, short-chain fatty acids, which are generated in millimolar levels by digesting our plant-derived complex carbohydrates, that results in acetate, butyrate, and propionate, which can signal through G-protein coupled receptors, GPCRs. Okay, and similarly, the secondary bile acids, quinodeoxycholic acid and lithocholic acid, they signal via TGR5 and the farnesoid X receptor. And the indoles, the indole 3-carbinol, indole 3-acetate, they signal through aryl hydrocarbon receptors. I'm just giving these 3 examples because these are very well established metabolites which can signal the host receptors. Okay, I know this is the wrong time to talk before the lunch, but it is a fact. Gut microbiota is responsible for the odor of the feces, okay, or color of the feces. So you know the heme, right? Hemoglobin, it is a component of hemoglobin, which is cleaved into, converted into biliverdin by heme oxygenase. Biliverdin reductase convert into bilirubin. Bilirubin will be excreted into the duodenum via gallbladder. Then when it reaches the colon, it gets converted into urobilinogen. And urobilinogen further acted upon by bacterial oxidases gets converted into stercobilin. Okay, that impart the typical color of the feces. The reason I'm telling you is, if you see pale colored feces, that means you are anemic or something is wrong with your RBC or hematopoiesis. Okay, usually when you wiped out the bacteria or if you are severely anemic, the feces will be pale color. Okay. And also they are responsible for the odor of the feces. The reason I'm telling is, again, I worked with germ-free rats and germ-free animals. In fact, I did sensory analysis. They do not smell at all, okay? Because they do not have bacteria. And if they, in the conventional or germ-full mice, when the undigested food reaches the colon, they generate a very nasty smelling molecules, especially the methyl scatol and methyl indole. Those are the very noxious odoriferous chemicals. So in fact, this is published by my wife, and also I was also one of the author, which is highly cited, which shows that gut microbial products regulate gastrointestinal motility via toll-like receptor 4 signaling. Okay, we used germ-free mice. They have a lot of problem with their motility and also stool output. Okay, if you put them some LPS into the drinking water, we can correct their gastrointestinal motility. So the homeostasis of gut microbiota is very important. So general principles and theories apply. So demand and supply. Okay, example, if your diet do not contain enough fiber, so your bacteria go crazy because you are starving them. Okay, that could be the reason our cecum is almost like a rudimentary. It is in the rodents it is very big, but in the humans it is almost, you cannot see it because we are all eating now pizzas and all burgers which has no plant-derived material at all. Or in India our major food is rice, so it do not support any cecum or any okay. And use and disuse theory. So the cross feeding, this adds more complexity to the gut to study the gut microbiota, because there are so many bacteria belonging to different phyla and different classes, families. So they are interdependent. Okay. For example, if one bacteria generates butyrate, the other bacteria will utilize that butyrate and it generates acetate. Okay, it is all interlinked. If you deplete one species of bacteria, it will have a enormous change in the entire microbial ecosystem. Okay, and also prey and predator relationship. We do not know. There are some bacteriophages. So you know bacteriophages, right? Which kills bacteria. There are some viruses or bacteriophages in the ecosystem, but we do not know much about it. Okay. So diversity is the key for an ideal gut microbiota. The reason I'm giving this picture is when you have a low alpha diversity or low species richness, there will be a lot of competition. So that won't provide you very productive ecosystem, right? But when the ecosystem is like this, it will be very productive. So this ecosystem, the US population is one of the best example for this kind of high alpha diversity, high species richness, less competition, and very symbiotic in nature. So US population is the best example because the diverse population we see in the US, you can never see in any other country in the world. Okay, every country has their representation in US. How it is beneficial? The example, in this community, if everyone is meat eater, okay, there will be a lot of competition for the meat. Okay, you see in this community, there are some herbivores and all combination. So there won't be competition for only one thing. So that brings it, makes it very productive ecosystem. Some people eat plant-derived foods, plus some people they eat on meat. It won't make that much competitive ecosystem. You got it? So basically, your diversity is very key for the ideal microbiota, but there are some controversies, not controversies, kind of a contradictory. Reason is the infants who are on the formula food, their bacteria is diverse, but still they suffer from some metabolic diseases later in life. They are more susceptible to asthma, obesity, those kind of stuff. Okay, and also cesarean-born kids. They have a high diverse bacteria, but still they don't do well. Okay, gut microbiota is root cause for all diseases. Okay, even autism is linked to altered microbiota or microbiota dysbiosis. Okay, metabolic syndrome, systemic inflammation, obesity, diabetes, IBD. Okay, you name every disease is linked to gut microbiota. Okay, New York Times or National Public Radio in the US, almost every week there will be a story on gut microbiota. Nature, Science, Cell, at least 2-3 papers will be on gut microbiota. I stopped actually following the literature because it is too much to read all those papers. So microbiota dysbiosis is a widely used term. It is a catch-all term. Okay, it is very vaguely described in the literature. So basically the definition is an imbalance in taxonomic composition of the gut microbiota is called dysbiosis. So not all dysbiosis can cause a phenotype. Okay, some changes in the gut microbiota go you can unnoticed. So how the microbiota dysbiosis can result? Like diet, if you eat a lot of high salt, low fiber, high fat or food additives can cause changes in microbiota. Of course, antibiotics is known to cause dysbiosis. and certain drugs, and aberrant host innate immune responses or inflammation, especially in the inflammatory bowel diseases, they can cause microbiota dysbiosis. So microbiota dysbiosis is characterized by high volatility. That means morning you will have one composition, by evening they will have another type of composition. Low diversity, reduced species richness, loss of keystone taxa or commensals, bloom of pathobiomes, that means opportunistic pathogens. So you heard all last 2 days the pathogens. Now today you are learning about opportunistic pathogens which are present in our gut. Okay. So the proteobacteria is a microbial signature of gut microbiota dysbiosis. I told you E. coli is a minor community in a healthy person. Once he develops microbiota dysbiosis, the E. coli take over because it is a very resilient bacteria which can withstand heightened host innate immune responses. Okay, so in majority of studies, the microbiota dysbiosis, it is not very clear whether it is just an association or cause or consequence or an adaptation. Okay, only how you can do the cause or consequence by doing some fecal microbiota transplantation. You can assess whether the microbiota dysbiosis is cause for that phenotype. Okay, normal and cesarean delivery. So newborn intestines are sterile. Okay, again it is very, so now there is one paper in Nature Medicine, placenta has its own microbiome. And, but it was challenged by another paper, it is a contamination. Okay, it is, we do PCR, even there is a small contamination, you will get a signal. Okay, so you have to be very careful. And they slowly start colonizing bacteria. Usually what literature says is E. coli is the first one to go into the, for the colonization, where it utilizes all the oxygen and create a very nice atmosphere, like that is a anaerobic condition, and all the anaerobes will follow and they nicely settle in. So this initial colonization is very, very important. If it screws up, if it gets screwed up, then it will have a long-lasting consequences. Okay. And the C-section born babies have different bacterial species and a susceptibility to asthma and obesity. Okay, usually when a mother undergoes C-section, she may not be able to feed the baby. So obviously the caretakers take the baby, they will be feeding some infant formula. Okay, usually the babies get mother's bacteria when passing through the birth canal, and also when they start suckling mother's milk. Okay, skin and vaginal microbiota, those are the ones first they start colonizing. But in the cesarean delivery, it won't happen. The guts of C-section babies were dominated by opportunistic bacteria such as Enterococcus and Klebsiella, and the previous talk clearly indicated why Klebsiella is not a good bacteria to have, which circulate in the hospitals. So it is one of the very good example for nosocomial infections, which that means the infections of hospital origin. Compromised gut barrier or leaky guts. This is one of the very, very well accepted model for all the new age diseases like obesity, NAFLD, that is non-alcoholic fatty liver disease, and also type 2 diabetes. So good fences make good neighbors, right? If you have good fences, your neighbor, you will have a very peaceful with your neighbors. If they are not, then unnecessary problems comes. So it can leak, if there are leaky guts, it can cause metabolic infection. So I'm using the word metabolic, okay? It is not a true infection by a pathogen. So some leaking of some bacteria which are motile, they can go through the leaky guts. I told you that intestines are lined by a single layer of epithelia. Even if the 4 or 2 cells die, basically it is a hole in the intestine. So the fecal matter or bacteria or bacterial products can disseminate into systemic circulation. And these bacteria are commensals, okay, as long as they stay in the lumen. Once they go into the circulation, they are as bad as pathogens. Even though they will be killed by our innate immune cells, but sometimes it can lead to sepsis. So endotoxemia or metabolic infection, it is a very well accepted model. Endotoxemia is nothing but a leaking of a lipopolysaccharide. when you eat high fatty foods. So your liver makes lots of bile acids. So along with the fat, the dietary fat, even the LPS present in the lumen can be absorbed along in the as a lipid. Okay, and this can lead to low-grade chronic inflammation. It is not at all good because it is asymptomatic. That means you do not show any fever or any changes at the histological level. Silently, something bad going on in your body. Okay, you never notice it. Even there will be no increase in C-reactive protein or anything. So all of a sudden, when you go to something happens, right? If you get injured or anything, when you go to doctor, then doctor will say you have fatty liver or you are diabetic or something else. Okay, that is called low-grade or subclinical inflammation. So according to Hippocrates, all diseases begin in the gut. I agree to a large extent what he says. Okay, are we altering our gut microbiota? So yes, one of my favorite microbiologist, Marty Blazer in the NYU. So he surveyed many, lot of societies microbiota. This graph clearly shows that yes, US is almost completely changed because no one cook there. Everyone eat outside. Okay, so they will be changing their microbiota every day. One day they will go to Ethiopian restaurant, one day Indian restaurant, one day Chinese restaurant. So they will be exposed to so many varieties of whatever junk food, and finally they completely change their microbiota. Even this population with beginning modernization. So I put India into this because we never, in 1999 when I was leaving US, I never tasted a cheese. Even I don't know how it looks like. Okay, but now I see everywhere Domino's, Pizza Hut. Okay, so I don't know how our microbiota can handle all these new generation foods. Okay. Actually, declining microbial diversity in the modern societies, because I don't know, I can give so many reasons why the microbiota diversity is declining because of the strict hygienic conditions. Okay, even that is also not good. You can read a hygiene hypothesis. Okay, it's a there is a number of studies shows that if you keep your hands and everything clean all the time, you are very susceptible to a lot of allergic diseases or asthma type of thing. Okay, so tell your kids frequently go and play in the dirt. Okay, so they will get some good resistance. So in the US, otitis media, you know, otitis media means the ear infection. It is very common in the kids. Whoever send their kids to daycare, they have to go take leave for many days because the kids will get the ear infection. I have not seen any that kind of ear infection in India. Maybe it may happen here because here also daycare culture is coming. Okay, so something is related with the culture. This is another best example of we are changing our microbiota. This is again from the Martin Blazer lab. So disappearance of Helicobacter pylori and increasing esophageal adenocarcinoma. So you know that Helicobacter pylori is one of the most successful pathogen in the history or in the evolution. Okay, even it is also going away because of our drugs or foods or antibiotics. So Helicobacter is going down and the esophageal adenocarcinoma is increasing. So this is another example for we are changing our microbiota. So the bottom line is the host and the microbiota, they hold each other throughout their life, which starts at birth and ends with the body decomposition. You agree, right? So we can regard gut bacteria as the best frenemy forever. So my postdoc mentor, Andrew Govertz, still is my favorite. So he gives an example for frenemy is our in-laws. Okay, they are good or helpful at majority of times, but occasionally they can cause moderate to severe problems in the family. Okay, so that is the definition of frenemy according to my mentor, not from me. Okay, so to maintain such a huge number of bacteria, it is a Herculean task for the host to maintain within the lumen. Okay, so according to me, bioinformatics people may not agree. According to me, the bacteria numbers. Okay, I don't care who are there. It is more important is how many are there, where are there, whether they are confined to the lumen or they are approaching near the epithelia. That is not at all good. Encroachment. Okay. How many are there, where are there, what they are doing. That is mostly more important rather than just do 16S sequencing. Okay. People are already sick of looking at the sequences, because it doesn't tell anything. It is kind of a random sampling, like our exit polls. It may be true or it may not be true. Okay. So the gut-associated lymphatic tissue system, it expediently detect and clear pathogens, keep opportunities in check. This must be done while avoiding harm to beneficial microbes and the host tissues. Just imagine if out of zillions of bacteria, if you are infected with 10 or 100 salmonella, our immune system should be able to detect those pathogens. It is almost like finding a needle in the hay, right? Haystack. So gut microbiota helps in proper development of gut immune system. I feel that these bacteria, they are our inbuilt vaccine system. Okay. Otherwise, our immune system cannot develop, including the systemic circulation. I showed you nice neutropenic condition in the germ-free animals. So in other words, gut microbiota shapes and is shaped by the host innate immune system. So they regulate each other. There is a nice coordination between the gut microbiota and also our immune system. So I don't want to go through all this one. There are two types of immune system. One is innate immunity and the other is adaptive immunity. Innate immunity do not involve antibodies, whereas the adaptive immunity involves antibodies. Innate immunity is mostly generated by toll-like receptors, okay, whereas adaptive immunity is from the T cells, B cells, and their receptors. And the innate immunity is fast and non-specific, whereas adaptive immunity is slow but very specific, okay? For example, antibodies. Okay, so to maintain the homeostasis of those bacteria, we have several types of pathogen recognition receptors. Broadly, they can be classified into secreted form, that means they are present in the blood, or transmembrane or membrane-bound form, like toll-like receptors. And also there are some receptors in the cytosol. Those are called as cytosolic receptors. Okay. So again, this is a very broad field. I am only focusing on one receptor of my interest, that is TLR5, which stands for toll-like receptor 5. Why they are called as toll-like receptor 5? Because their amino acid sequence is very much homologous to a protein called toll in the Drosophila. That's why they are called as toll-like receptors. Okay, there are 10 receptors, 1 to 10. TLR5 is one among them, and it is one of the very well studied protein-protein interaction. We know that where the flagellin binds to TLR5 and which amino acids are required for this TLR5 flagellin interaction. So toll-like receptors are type 1 transmembrane receptors. They are evolutionarily highly conserved. They recognize specific microbial ligands, like TLR2 is for peptidoglycan, present in gram positive, whereas TLR4 for LPS in the gram negative. Okay, whereas TLR5 recognizes flagellin. Okay, I will tell you what is flagellin. It is my favorite molecule. It is magic bullet in my hands. Okay, so once they bind to their respective ligands, which transduces a signal via an adapter protein called MyD88, which results in the translocation of a pro-inflammatory transcription factor NF-kappa B into the nucleus, where it initiates the transcription of variety of pro-inflammatory genes, okay, like cytokines, TNF-alpha, IL-6, I mentioned only few. This pro-inflammatory response from the host or by the host is required for efficiently get rid of any pathogen going into the cell. Okay, and at the same time, this should happen in a controlled fashion. Otherwise, it can result in unnecessary inflammation to the host tissue, which can result in some auto-inflammatory diseases. Okay, you got it? Flagellin and TLR5. So what is flagellin? So if you look at the Campylobacter jejuni or Salmonella or E. coli, they are Clostridium difficile, they are all motile bacteria. How they are motile? Because they use a whip-shaped structure called flagella. Okay, flagella is a whip-shaped structure. It is made up of millions of flagellin monomers. Okay, all these flagellin monomers are covered in a sheath that constitute the flagella. Okay, flagella do not activate TLR5. Only the flagellin monomers can activate the toll-like receptor 5. Okay, I will be using TLR5 or T5 or toll-like receptor 5, all are same. Okay. Okay, when I started my postdoc career at Emory University, this is my favorite paper. This paper came out because my project goal was to study the TLR5 flagellin interaction in IBD. I forgot to tell, this is very important information. So people who are a non-immunologist, they think that all bacterial ligands are the same. Okay, they induce the same set of cytokines, both qualitatively and quantitatively. It is absolutely wrong. Okay, for example, toll-like receptor 4, which is predominantly expressed on immune cells like dendritic cells and macrophages, when they get activated with LPS, they secrete lots of TNF alpha. Okay, it is very pro-inflammatory. Okay, whereas toll-like receptor 5 is predominantly expressed on epithelial cells, including intestinal epithelial cells, lung epithelial cells, bladder epithelial cells, retinal pigment epithelial cells also. Okay, and epithelial cells react robustly to flagellin, but not to LPS. Okay, I don't want to go into the detail because there are many reasons epithelial cells are refractory to LPS. Okay, they do not respond. So when I joined, my goal was to decipher the role of TLR5 and flagellin in IBD. Okay, so this paper became a good rationale for me to propose a project to Crohn's and Colitis Foundation. I am here because of the Crohn's and Colitis Foundation, which is supporting me from 2006 till now. Okay, so bacterial flagellin is a dominant antigen in the Crohn's disease. So why you can ask question how some pathogens can evade evade our innate immunity. The Helicobacter, as I said, it is one of the most successful pathogen. It not only secretes urease to protect from the acid, but it also mutates its flagellin. Okay, you see, I don't know whether you can see it or not. So the flagellin is mutated in the Helicobacter when compared to E. coli. Okay, some amino acid residues. Then the TLR5 do not recognize Helicobacter pylori flagellin. And also the E. coli LPS has 6 fatty acids in the LPS, whereas only 4 in the Helicobacter LPS. That's how it evades innate immune system and became a very successful pathogen. So why I am repeatedly telling successful pathogen? Any pathogen will become successful if it do not kill the host. If a pathogen kills the host, even it will be eradicated, right? Slowly. For example, COVID-19, it is killing the people. So finally, it has to disappear. I don't know when, but it will. Okay. So when I started my career with the first verifying the loss of function of toll-like receptor 5 in a genetically engineered mouse, that is TLR5 or T5. I will be using interchangeably all these words. When we injected either E. coli flagellin, pure form, HPLC purified form, E. coli flagellin or salmonella flagellin into the TLR5 knockout mice or wild type mice, we call them as wild type mice. That means control mice. Okay, we have to use litter mates or cousin mates. We cannot use mice from our colony. Like for example, if I get our TLR5 knockout mice are from Japan. And we can't buy wild type mice from vendors and do the experiment. So basically we have to breed them and then we get the heterozygous mice, right? When you breed knockout and wild type, you get the heterozygous, again breed the heterozygous mice, then you get the wild type and knockout mice. Okay, that's what we have to do. Otherwise, they will have different microbiota if you buy the mice from vendors. Okay, so here you can see the TLR5 knockout mice, as expected, they did not respond to intraperitoneally administered flagellin. Like there is no IL-6 or KC or GCSF. So flagellin did not induce pro-inflammatory cytokine response in the TLR5 knockout mice. So based on this, we hypothesized that these mice would resist intestinal inflammation, right? Because the TLR5 is predominantly expressed on epithelial cells. So if there is no any pro-inflammatory cytokines, they will be protected from the inflammatory IBD or murine form of colitis. That was our hypothesis. My hypothesis never came true. Always I get opposite results. Okay. Instead of resistant to colitis, they develop spontaneous colitis. Okay. 10% of the mice, they develop rectal prolapse. It is a severe, irreversible form of colitis, but it doesn't result in mortality. But according to the animal protocol, we have to euthanize the mice because it is a lot of distress for the mice. Okay. And only 10% have this rectal prolapse, and 25 to 30% mice have typical or classical symptoms of colitis, that is colomegaly, shrunken cecum, and splenomegaly. Splenomegaly is common in IBD because of the anemia. Even humans, we develop splenomegaly. Okay, they also have increased bacteria in the TLR5 knockout mice, and they have increased colonic adherent bacteria. I told you they have to be confined to the lumen, but not to the epithelial cells. And they also have some easily detectable bacterial burden in the sterile organs like spleen and liver. Okay, we also characterize them. They are not pathogens. Okay, this is the result we published in JCI, and again, the spontaneous colitis development is microbiota dependent. We can prevent colitis by giving broad spectrum antibiotic streptomycin in the drinking water. They also have typical histological changes at the cellular level. Okay, am I good with time? 12:15, right? Okay. So we suspected that when we got these mice from Japan, they might have acquired some pathogen. Okay, we don't see them. We never saw them simply dying. So it is obviously very difficult to think that they have any deadly pathogen. So we thought that they may have some bacteria. They are triggering colitis in these mice. So to rule out that, we completely removed their bacteria. How? By embryonic transfer. So when the TLR5 knockout mice become pregnant, they by aseptically they transfer those embryos into surrogate mother. Okay, when the surrogate mother delivers those pups, the genetics will be from the original mother, whereas the bacteria will be from the surrogate mother. So that's how we change their microbiota. Or my mentor used to say, we Jacksonized these mice, because we got the mice from Jackson Laboratories, one of the world's largest animal supplier. So re-derived offsprings acquires microbiota of birth or recipient mother, but not genetic mother. Okay, then we will see what happens to their colitis. Okay, re-derivation reduced colitis. Almost there was no any rectal prolapse. Okay, 0%. But still, if you look at them carefully, they have colomegaly, that is increased colon weights, spleen weights, and increased cecal myeloperoxidase, which is an enzyme widely used for as an indicator of neutrophil infiltration. Okay, and also they have increased the general inflammatory marker serum lipocalin 2. So all these results suggest that the mice which do not have a severe form of colitis, they suffer from low-grade chronic inflammation. I hope you remember what I told you, like low-grade chronic inflammation is disorders. Okay, we'll see what happens to these mice. Okay, this serendipitous discovery. So we published paper in JCI. We don't know what to do next. Okay, our goal is to use these mice for multiple forms of colitis, but already they have spontaneous colitis, so we don't know what to do. So mice, if you breed the mice, you'll know. If you don't go to the animal house for 1 month, it will be crazy. Okay, they will be giving pups every 21 days. Okay, so we want to get rid of them. So one day I went down and they looked a little heavier for me when compared to their wild type mice. Okay, but definitely they are heavier, but not as severe as leptin knockout mice. Okay, so we didn't use any high fat diet or anything. This is with the regular lab show what we use for their maintenance. Okay. Then we thoroughly characterized this obesity phenotype in the TLR5 knockout mice, which both the male and females, they display obesity. They also suffer from hyperlipidemia. And also when we subjected them to magnetic resonance imaging, you can see increased fat distribution throughout their body, their abdominal fat pads, they are almost 2-fold higher than their wild type littermates. In this study, we use the littermates, okay? And that means the wild type mice and the TLR5 knockout mice are born to same mother. They are true littermates. That means they have similar microbiota, but still the TLR5 knockout mice cannot maintain their microbiota. And they also hypertensive. So this is our first study linking the microbiota to blood pressure. Okay, so collectively, all these results suggest that TLR5 knockout mice suffers from metabolic syndrome. So metabolic syndrome is a constellation of 5 different parameters. Okay, one is hyperglycemia, hyperinsulinemia, type 2 diabetes, and then hypertension, increased BMI. Among 5, if any 3 are there, so we can put that person into metabolic syndrome category. It is not a disease, it is a disorder. Okay, that's why they're called as a metabolic syndrome. Okay, any questions? And TLR5 knockout mice are insulin resistant. As I said, it is one of the major features of metabolic syndrome. So they are hyperinsulinemic, and when we subjected them to insulin sensitivity test, usually in the clinics, they won't do it for humans. In the humans, they do glucose tolerant test, not the insulin sensitivity test. Okay, you see the red one is TF5 knockout mice. When you give insulin, they are not inducing hypoglycemia as wild type mice. So that suggests that they are insulin resistant. Okay, so I don't like using high fat diet, but I had to go with the wind, right? So we put these mice on high fat diet. So basically they get 60% of the calories from the fat. It is the people call it as a Western diet or cafeterian diet. Just within 12 weeks, you see, this is the beta cells or histology showing the pancreas, and this cell is a beta cell, this one, okay? You see in the TLR5 knockout mice, when we fed them on high fat diet, they develop insulitis. That is immune cell infiltration into the beta cells. And also the liver is severely steatotic and also inflammatory. So with high fat diet, they develop severe metabolic syndrome, okay? Why they are developing obesity and other symptoms of metabolic syndrome? Because they are hyperphagic. So if you recollect what type 2 diabetes patients do, right? They will be hyperphagic. They have to eat all the time. That's what the TLR5 knockout mice are doing. They are eating only 10% food more. Okay. You may think that only just 10%. Imagine in 3 months period, that 10%, it adds a lot, right? That's why the TLR5 knockout mice almost 20% more body weight than their wild type littermates. And also first, so usually food intake is very tricky because I don't want to measure the food intake by putting 1 mouse per case, because they are social animals. That will, if you put them as 1 animal per case, that will cause a stress. So we put them 5 mice per case and then we measure their food intake. So there will be a lot of droppings will be there. It is not an accurate food intake measurement. So best way is measure their stool output. You cannot miss it. Put the mice in an empty case for 1 hour, then you will get the wet stool output. Okay, dry them, and then it's see the stool output is more in the T5 knockout mice. You may miss some food particles when they are eating, right? But stool output you never go wrong. And we thought that they may have diarrhea because more water content, but there is no water. It's not because of the diarrhea. So basically, the food intake nicely correlates with the increased stool output. And by calorie restriction, it greatly improves metabolic syndrome in the TLR5 knockout mice. So that means we balance the food intake in the wild type mice and TLR5 knockout mice. By doing so, we can prevent the metabolic syndrome. Okay, this is bioinformatics. Okay, this is one way, like 10 years back, people used to represent the microbiota sequencing like this. Here, each dot represents one bacterial microbiota. Okay, you see there is a little overlap between the wild type and non-colitic TLR5 knockout mice. I said some 10% of the mice do severe form of colitis. They are way dysbiotic. Okay, and they have reduced diversity, typical microbiota dysbiosis, increased volatility, but no change in major phyla. So Jeffrey Gordon in WashU, he is the pioneer in the microbiota studies. He showed that obese mice or leptin deficient mice, they have increased firmicutes to bacteroidetes ratio, but we did not see that kind of alteration in firmicutes and bacteroidetes. We saw only taxonomical alterations in their bacteria. That means there are 70 to 80 species changes in these wild type mice and obese TLR5 knockout mice. Okay, and then by spending $100,000, we generated TLR5 knockout mice into germ-free condition in the Taconic facility in New York, and then they sent us in a special container when they are 12 weeks old. You see no difference in the body weight, and their fat pads are comparable, and the spleen weights no change, and the colon weights are no change. So basically, when you remove the bacteria from these mice, no colitis, no metabolic syndrome. So that suggests that the metabolic syndrome and colitis is completely microbiota dependent. Okay. So this is my last slide before the lunch. So again, it is disgusting to before the lunch, okay? TLR5 deficient mice fecal bacteria is sufficient to induce metabolic syndrome in the germ-free wild-type mice. Okay, at that time we didn't have germ-free TLR5 knockout mice. So basically here we use the fancy word, even though it is a high school experiment, like simply gaussing feces from wild-type and TLR5 knockout mice, making them into paste, putting them into germ-free mice. That's all. But we use the word fecal microbiota transplantation. Okay. So then we measured their food intake. That means the germ-free mice which received fecal extract from either wild-type mice or TLR5 knockout mice. So you can see the mice which received TLR5 knockout fecal contents, they are hyperphagic, they gain more body weight, they are hyperglycemic, and they are also hyperinsulinemic, and the loss of glucose intolerance, more fat pad, and increased pro-inflammatory cytokines in their colon. So basically, just by fecal transplantation, we recapitulated metabolic syndrome in the germ-free mice. Okay. So all these results were published in Science in 2010. Metabolic syndrome and altered gut microbiota in mice lacking toll-like receptor 5. Okay. So you may ask, what is relevance? Right? Well, obviously, it has to be some translational potential, right? So with the help of Karan Molke at UNC Chapel Hill, she has a large collection of like 13,000 or 15,000 patients. And just we asked them to do the sequencing for the TLR5. So out of that, she identified 52 TLR5 knockout human beings. Okay, it is a very common 1 in 250 humans lacks TLR5 function due to the TLR5 stop mutation. Okay, so she found 52 TLR5 knockout human beings, and they also have similar phenotype as a TLR5 knockout mice. So okay, that's why I put them in the red, but to publish a human study, these numbers are too small, it seems. Okay, they need a rigorous statistical analysis. So this never published because whenever we send this manuscript, they say the number is too small. Then we lost our interest. Then you see they have these TLR knockout humans. They are hyperglycemic, large waist, high triglycerides, low HDL, hypertension. All the data is sent from Dr. Molke because they had the data already. We just asked her to sequence them and stratify them into wild type TLR5, heterozygous and homozygous TLR5. Okay, there is another study which Carl and Kerry may like it. Okay, these TLR5 knockout humans, they are very susceptible to Legionnaires' disease, Legionella pneumophilia. It is also flagellated bacteria, and they are very susceptible to this disease. It was published in Journal of Experimental Medicine, very highly respected journal in 2003. Okay, and now I will show you how we used flagellin. It is a potent activator of epithelial cells. I already told you. If you put salmonella on epithelial cells or flagellin on epithelial cells, or if you challenge the epithelial cells either with salmonella or flagellin, you get the same set of genes upregulated. Okay, live bacteria is equivalent to flagellin. So that's why I use the word, it is a dominant activator of epithelial cells. So one of my project which got funded, of course, very internal grant, I want to use flagellin as a antidote for coronavirus infection. Okay, I will show you why we are planning. So this is my crazy experiment, which was really gave a lot of ideas. Flagellin treatment protects against chemicals, bacteria, viruses, and radiation. Okay, it is all unexpected results. And also how flagellin protects, it prevents epithelial apoptosis. Okay, that's how it is very beneficial in the gut. Okay, apoptosis means cell death. Flagellin prevents it. Okay, once the epithelial cells are dead, I told you it will create a hole. Okay, so that can cause a lot of problems like leaky guts and all. And also, this is another follow-up paper. Flagellin protects gut mucosal tissue from radiation-induced mucositis. It is a big deal in the cancer clinics. And this is my favorite paper. Prevention and cure of rotavirus infection via TLR5 or NLRC4. These are the two receptors for flagellin, mediated production of IL-22 and IL-18. This is again in science. So basically, we can cure highly contagious murine rotavirus infection by a single dose of flagellin. If you give IP, even the mice will be completely free from any kind of infection. You cannot detect the viral RNA by quantitative RT-PCR also. Okay, so that's where I stop, and after lunch I will tell more stories about the TLR5 knockout mice. Okay, you want to have questions, you can ask me, or you can hold on to those questions. We can have the questions after finishing my second part of the talk. Okay, what do you want to do? If you have any questions, if you cannot resist, go and ask.
Now the session is open for questions. If you have any question, you can ask now. Like next presentation, you can ask after the presentation.
Yeah. Can you turn on your mic? I cannot hear.
Are there any set of data to show that in human beings, the TLR5 is completely different? It has a different set of expression in human beings. There are people who follow different various kinds of diet. Let us say if the person is completely off fat.
As I know, no one has looked at it. I think it is JSS should look at it. So that's why I'm stressing on that, because we cannot touch human beings. Okay, for us, forget it. I don't want to do any human research in US. Getting IRB approved, because I'm not an MD. They don't trust us. We cannot touch any humans there. Even I don't know why I have an IRB approval to get some serum from cholestatic patients. I don't know who they are. We don't know their name. We know only gender and their age. That's all. Very difficult to do all those things in the US. If you identify, yeah.
Here in India, now if you see all the advertisements, there are several kinds of diets. One is completely fat-free, one is this free, one is that free, whatever it is. So that will be, there are cohorts of studies, and to see TLR5, that will be a good study to show how it is correlating with studies.
That is good. I'm a nutrition, I'm biochemist, I'm a lipid biochemist. I don't believe all those things, diets, keto diet and all. Basically, doing a Atkins diet, protein-free diet, a carbo-free diet, we are screwing up ourselves. Okay? It never works. It has to be 1:1:1, either carbohydrate, fat, or protein. You cannot, how can you survive without protein diet? You need amino acids, right? To make your enzymes, proteins and all. Anyway, some people, you cannot change them, let them do whatever they want. Yes, yeah. Now keto diet, you know how dangerous it is, keto diet? They can go into ketoacidosis. Instead of that, do intermittent fasting. That's my recommendation. Huh? Yeah. Even keto beer also there. Okay, keto ice cream. Wow, that's... Any questions from the participants or students? Are hungry? You are like TLR5 knockout mice now.
Dr. Vijay, I have a few questions.
Sure.
It was a really nice presentation and informative presentation.
Thank you.
And we know, like, we as a cancer researcher, we do a lot of cancer research based on mice studies. Like, we use single genes and we focus mainly on xenograft and all those things. We don't focus on gut microbiota. What is your opinion on that?
You should do it.
You
should do it.
There are billions of papers, like cancer research papers.
Yeah.
Previously, we don't know, like, we didn't know about LPAs and all those things without focusing on gut microbiota. Maybe that will really.
Yes, it is already too late for India to for us to pick up these 16S now, but it is better late than never. So I don't know what you can do just with the 16S sequencing. I told you already. So you have to go beyond 16S sequencing. Okay, what they are doing there? Where are they? So you cannot link AV, like for example, anyway, I don't want to comment on that. I will go crazy. Yeah. Yes, it is good to do, but you don't have germ-free facility here in India. I don't think anyone has germ-free facility. So those are gold standard for microbiota studies. Like even if you take the feces from NAFLD, non-alcoholic fatty liver disease, give them to mice, they will develop NAFLD symptoms. It is amazing, right? So even I don't know how really they are, once they go into the gut, how their signaling and all, it completely changes.
And also one more thing, like in India now, we have a lot of diabetic people, diabetic and even metabolic syndrome patients. But when they go to clinics, they don't talk about gut microbiota and all those things. They just give.
It is better not to talk with doctors, Indian doctors, any knowledge, okay? They will scold you. By looking at the Google, you are, okay, you doctor or me doctor? That is the first thing they will ask, okay? I'm open to say that, okay? Even I had the same experience. They don't care. Whatever you say, they don't care. Okay, I think you didn't see the reason I put that funny, this one is American obesity, Indian obesity. I don't know whether you looked, you missed it, I think. So we are not like that, okay? But we are robust, okay? We have everything, okay? You see here, I think you didn't see it. I am not obese, but I am robust, means we have all types of problems. Like, believe it or not, like more than 30% of the Indian population is diabetic, okay? Like just if you discover any alternative to insulin, you will become a billionaire. Maybe I don't know other word after billionaire, so you can get that much of money.
And is there any link with TLR5 and the leptin?
Interest of time, I just focused on the insulin. Yes, they do have high levels of leptin. I don't know whether they are leptin resistance or not, because to do that, you have to inject leptin into the brain. So I didn't go to that level. I just stick to intestine. Okay.
Okay, then. Thank you.
Yeah.
So, sir, it's a wonderful presentation. So, very crystal clear slides, very easy to follow. It's a wonderful lecture. It's very enlightening as well. So, I have like a question saying, is the TLR5 knockout is sufficient enough to develop obesity, or do you require some kind of a challenge to see the obesity visible?
So your question is
just TLR5 knockout is sufficient enough to see the obesity, or does it require some kind of a challenge to see the visible phenotype?
Okay, that's very good question. I did not use any high-fat diet. I don't like that experiment at all, because people feed 16 weeks, 16 months that high-fat diet. It is waste of money. Okay. I'm sorry if I'm not offending anyone. This is why I am very excited about this phenotype is, it is regular diet. It is a maintenance diet, what they put in the animal house. Okay. And also my junior, after I left my postdoc mentor's lab, so they made epithelial TLR5 knockout mice. Okay, epithelial specific TLR5 knockout mice, they have the same phenotype. Okay, like a global TLR5 knockout mice.
Is there any link with absorption, food absorption and assimilation? Like TLR5 and food absorption?
In these mice?
Yes.
I will come in the after lunch. After eating, we will talk about absorption and digest, okay?
And I have the similar one. Another question is, alternatively, suppose if you make a transgenic TLR5 mice and challenge them with high-fat diet, what do you see? Do you see the reversal of it?
I don't, because they are highly, if you make transgenic means overexpression of TLR5, I don't think anything happens. No.
Okay. So in a clinical side, suppose if you have a TLR5 agonist, would that be helpful to kind of help in the treatment of obesity or diabetes?
No. No, that's not the showing all the beneficial effects of flagyellin. So, like a non-specific activation of epithelial cells by flagyellin can protect against many things. Not that that will protect against obesity. So that was my idea. Like if I don't have mouse model for coronavirus. Even I can 90% guarantee that your flagyellin injection can prevent coronavirus infection, because see, even coronavirus also targets respiratory or lung epithelial cells, right? So if we give flagyellin, we have data that the ACE2 expression, that is the receptor for coronavirus, it goes down. So my rationale is, by giving the flagyellin, the epithelial cells can resist the coronavirus infection. Okay, I don't think flagyellin administration or a TLR5 agonist can be used as a weapon for obesity. I don't think so.
Okay. And the thing is that the connection between knockout of TLR5 and the gut microbiota and the subsequent development of obesity, how that exact mechanism, the connection happens. Can you?
Yeah, I think, like, can I finish all the connections? Then I will tell you, like, because I still 3 more connections are coming.
Okay, yeah. Thank you very much, sir. Such a wonderful and enlightening presentation.
Actually target TLR to iris.
Yes, yeah. No, actually, there is one group, he is my friend only, his name is Ashok Kumar. Whatever I do with intestine, he will go and do it in the eye, okay? Like, I told him, yeah, Vijay, tell me what you do, I will do it in the eye. What I was telling. If you are okay, if you are not hungry, you can go on to questions.
One question.
Huh?
Yeah, I just want to make an observation, and I want to thank you for the wonderful presentation first. But I want to make an observation for the students here, because we've been talking about this all week. What's driven your work is questions. You have questions. You have curiosity. It drives you to think about it. It's not technology that's driving his research. It's his questions. And then he's actually going out and he's trying to determine using technology how to answer those questions. And I think this is the problem we've been having here. We've been having these discussions over and over again. This is a scientist that approaches the world with curiosity. And that's what you have to do. You have to hypothesize. You have to have compelling questions. I mean, wouldn't you agree? I mean, I think that's what we've been talking about this, which is there's this tendency to get some technology and just to process things without questions and without hypotheses. And I think this is a really important thing to notice about his work, that it was driven by his own curiosity. You know, and this is what should be driving your work. You know, you should be asking yourself questions. You know, you should be curious about everything you see. You know, that's how you get to the point where you make discoveries. So anyway, that's just an observation, and I thank you for allowing me to do that. So.
Thank you. Thank you. Yeah.
So could it be true the opposite, the vice versa, where you have people with diabetes and malfunction TLR?
Possible.
And does the TLR5 functioning affect the other two receptors?
No, I don't think so. Because TLR5 flagellin is unique. If you look at all the ligands for TLRs, they are not proteins. Only TLR5 is flagellin. That's why it is a Nature Immunology paper. They know which amino acids are required and where it binds, and which amino acids are required for inducing these cytokine response. It is very fast. If you give LPS, it takes 1 hour for the mice to go into distress. For flagellin, 15 minutes. The mice will be sick. Yeah. I love questions. Yes. Can you? Healthy fetal? Oh.
Fecal transplantation. She's asking about fecal transplantation.
By reversing, you say always it is easy to transfer bad characters. Okay, it's very difficult to reverse the bad characters. Okay, so that's what I will show you another cancer model. You will get the answer. So basically, it's easy to transfer bad bacteria. Okay, they can go and dominate. But if you think that good bacteria can go and dominate bad bacteria, I don't think even in our society it cannot possible, right? Yeah.
Okay, then we can go for lunch now. Sorry, lunch.
Thank you for insightful information, sir. Lunch has been organized in the guest house. I request everyone to join back for next session by 1 p.m.
Dr. Vijay will start now. If you have any questions, you can ask after his presentation.
Are you ready? Any questions before move on to the second part? Move on to the slides. Yeah. So we stopped here, right? So I discussed some of the beneficial functions of flagyellin against viruses and other things. So then again, I will move back to toll-like receptor 5 knockout mice. So this is only one summary slide I put. So what it shows is I told that TLR5 knockout mice has more bacteria. So obviously they generate more short chain fatty acids. Okay, SCFA stands for short chain fatty acids. They will be absorbed via enterohepatic circulation into the liver. So where they can participate in the hepatic lipogenesis. Okay, for example, they can by fatty acid elongase and synthase, they become stearic acid and palmitic acid. And there is a notorious lipogenic enzyme in the liver that is SCD1, steroyl-CoA desaturase 1. Okay, many pharmaceutical companies, they targeted SCD1 to prevent obesity like 15-20 years back, but they are failed. Okay, because of some serious side effects. Okay, with the help of SCD1, these saturated fatty acids, which are actually they are more lipotoxic. Okay, the SCD1 convert them into mono unsaturated fatty acids like oleic acid, palmitoleic acid. So why these fatty acids are a very good source for deposition into neutral lipids like triglycerides and cholesterol esters, and those levels will keep on increasing in the TLR5 knockout mice. And also, like all the roads leads to Rome, and every condition favors the hepatic lipogenesis in the TLR5 knockout mice. For example, they have inflammation, right? Like low-grade chronic inflammation. Inflammation can upregulate SCD1 expression. It is a very well established. And also, I told the TLR5 knockout mice are hyperinsulinemic. They have a lot of insulin, like type 2 diabetes patient, and insulin is a potent inducer of C18-1. Okay, so like that, the gut microbiota participate in hepatic lipogenesis, developing into fatty liver. Once the liver is loaded with lipids, that can kick in insulin resistance, and which spreads to systemic insulin resistance, and finally they develop hallmarks of metabolic syndrome. So in this paper, what we showed is we deleted SCD1 in the liver and prevented the metabolic syndrome. Okay. So remember, they have more short chain fatty acid generation activity in the gut because of the fermentation. So the question is related to what we observed in the previous publication. So question is, does dietary prebiotic fermentable fiber corrects or aggravates metabolic syndrome in the TLR5 knockout mice? There are two corrects or aggravates. That means I am asking two questions. Like a fermentable fiber, prebiotic fermentable fiber means, again, I am planning to increase the bacterial fermentation by giving a prebiotic fiber called inulin. You might have heard inulin. Do you know? It is a plant-derived polysaccharide. I will tell you more about inulin. So if you give inulin, it further increases short chain fatty acid generation, and it may increase metabolic syndrome. That is one possibility, right? And the second possibility is inulin is very well known as a bifidogenic factor. Okay, it can correct microbiota dysbiosis in the TLR5 knockout mice, and thereby it can prevent metabolic syndrome. So those are the two questions I am asking by intervening with their diet. Clear? Okay. So let me tell a little introduction on naturally occurring dietary fibers. So basically there are two types of fibers. Oh, I changed it. Okay. Insoluble fiber, it is a misleading. Basically, it is a non-fermentable fiber, cellulose. Like paper is a pure 100% cellulose. We cannot digest because our microbiota do not express cellulases. Okay, whereas the ruminants like cows, buffaloes, or goats, whatever, they can digest because their microbiota, they are enriched with the cellulase expression. Okay. And whereas the soluble fibers or fermentable fibers like inulin and pectin, they can, they are, even though they all look like a white amorphous powders, they are chemically very distinct. Okay, the cellulose is poly-D-glucose, whereas inulin is a polyfructosone, whereas pectin is a polygalacturonate. Okay, they not only differ in their structure and also their fermentation capacity. Cellulose has little or no access to fermentation, whereas these inulin and pectin can readily fermentable by gut bacteria, generating short chain fatty acids in millimolar levels like acetate, butyrate, and propionate. So Danone is one of the very popular yogurt in the US. So they supplement that yogurt with pectin, which is isolated from either apple peel or orange peel. Okay, very cheap source. And also, some of the grocery stores, they sell inulin in the purified form. Okay, and there is a brownies called Fiber One. Each brownie is loaded with 20 grams of inulin. Okay, pure inulin. Or it is in my view, it comes under a processed food. Okay, I'm giving you the clue already. Processed foods, good or bad, you keep imagining. Okay, recently, Food and Drug Administration, they approved to use inulin in the processed foods, including infant formula. Okay, so they went beyond what they know. Even they sprinkle inulin on the French fries and they claim that they are healthy. Okay, to that extent, the food industries are going because there is a huge miscommunication between the food companies and nutritionists. So food companies or food industries, they always think about getting a pure compound something, which should be very cheap and they can sell it in bulk. And the nutritionists, they also wanted pure compound. But Mother Nature do not like that, okay? We have to eat the whole foods. I will tell you why. Because this inulin is isolated from the chicory root or Jerusalem artichoke. Maybe you might not have seen the Jerusalem artichoke, but even onion and garlic also very rich in inulin. So, wow, since in the hands of nutritionists, inulin is a magic bullet. It cures everything. Okay. That's what all the studies, maybe there are more than 10,000 papers. So that's why I trusted inulin. Yes, if I put inulin, because of the Phylogenetic factor, it can correct microbiota dysbiosis and protect the TLR5 knockout mice from all the problems they are suffering. Okay, feeding inulin diet protects 40% of the TLR5 knockout mice from metabolic syndrome. Okay, this is the diet. I did not come up with the formula. So there is already a diet. It is an open source diet. It comes with 7.5% cellulose and 2.5% inulin. I simply asked the vendor to reverse the source of the fiber, reverse the composition of the fiber into 7.5% inulin and 2.5% cellulose. So why did I retain the 2.5% cellulose? Because to protect the animals from diarrhea, you need some roughage, okay, that can withhold water in the colon. If you give all the fermentable or soluble fiber, there is highly likely those mice will suffer from diarrhea because they don't have any water holding capacity. So total fiber is 10%. It is not like any higher levels or anything. If you order any commercially available diet, it comes with 6 to 10% of the fiber, usually completely cellulose. So we fed the mice for 6 months, and 40% of the mice, they become lean, like wild type littermates, but still 60% of the mice are obese. So we were happy that at least 40% of the mice were protected. By feeding, I will be using ICD, that is inulin containing diet. Okay, these are all metabolic parameters. So all these green ones are the mice which are protected from metabolic syndrome. Their body weight is comparable to wild type mice. Fat pad, blood glucose, and then insulin levels, they're all comparable to wild type mice in 40% of the TLR5 knockout mice. And what made me very excited about this is they have abnormally low level of triglycerides and cholesterol. Okay, so don't think that if anyone has 80 milligrams of cholesterol, it is healthy. Okay, something major problem they may have. Hypocholesterolemia is more dangerous than hypercholesterolemia, but no one knows about it. Okay, remember that. Okay, so I know that because my father is a physician, so always I used to check, like especially people suffering from protein malnutrition, kwashiorkor or marasmus, they suffer from hypocholesterolemia. Okay, I suspected that something wrong with that mice. That's why they are protected. Okay, so I want to disclose because inulin is a very popular food, supplemented fiber, and I don't want to brand it as a what is that? Untouchable person. Okay, what is that? The arapaya, something they call. Okay, anyway, so I am neither pro-fiber nor anti-fiber. Okay, and we just followed the unexpected observation. So this is because most of the talks last 2 days was very serious. I want to make something humorous. So I'm showing you some unexpected phenotype with my favorite actor, comedian actor, Johnny Lever. When we observed that phenotype, we were shocked. Okay, and also we were surprised. And when we tell this phenotype what we observed, sharing with our colleagues, they ridiculed us. They laughed at me. Okay, and they are enjoying whenever we say inulin is a magic food. You are saying that inulin is causing something bad. And finally, we reached a stage we were begging. Okay, please accept our result like that. Okay. So what we observed is when we were processing the serum sample, those 40% of the mice which are protected from obesity, they had a yellow color serum. Okay, all this success depends on how best quality serum you get. If you get any tinge of hemolysis, you cannot see this phenotype. Okay, so remember, if you become a researcher, so if you are working with mice, getting high quality serum without any hemolysis is very critical. Because if even in the clinics, the hemolyzed samples are not used for any of the studies because the hemoglobin will interfere with many of the assays. Luckily, people named me as a vampire when I was in Emory University because whoever want to bleed, they bring all their mice to my lab. So I bleed with retroorbital plexus, like eye bleeding. Okay, so I know that whenever the serum is yellow in color, because my father, a physician, I had an opportunity to see many of the jaundiced patients where their sclera and nails and all gets yellow in color. Okay, because of the, I will tell you. Then I suspected it could be bilirubin. Okay, having that much of visible bilirubin in the serum is not. It is a big problem. They have something big problem with their liver. And that too, it happens within 2 weeks after putting them on inulin diet. That is more surprising for me. Okay, that's why I put Johnny Lever expression. Exactly, we were in that kind of situation. We don't know because we thought everything will be protected, but they're turning into jaundice. Okay. So this could be cholestasis or impaired or no bile flow at all. So based on the bilirubin levels, we stratified the mice into low bilirubin, that means they're obese, and high bilirubin, they are non-obese. Okay, that means the obesity, they are protected from obesity at the expense of liver. Okay, then let me tell some like liver. Now I am moving from intestine into liver. So as you know, liver is a moonlight and an altruistic organ. Altruistic means it sacrifices many of the things to other organs. So it is a major metabolic organ. It is a central non-lymphoid organ, firewall and first hit organ. Whatever you do, even though intestine is getting it first exposed, but finally everything will go and accumulate in the liver. If you take acetaminophen or aspirin, finally it is the liver's job to send that chemical out by xenobiotic metabolism. It is also highest regenerative capacity. That means more prone to cancer. And immunotolerant organ, again it is immunosuppressive. Okay. In addition to many housekeeping functions, the liver has additional jobs like synthesizing bile, albumin, ketone body biosynthesis. Whenever you are starving, liver will make ketone bodies, but it cannot use those ketone bodies for itself. It synthesizes ketone bodies for other organs. Okay, that's why liver is a perfect altruistic organ. Okay. In addition to that, it has to metabolize all the chemicals we ingest, including alcohol. Okay, so then we because the jaundice or cholestasis is not just bilirubin. It will be associated with the bile acids also. Okay, because of the bile duct obstruction, there is no way for bile acids to go into the gallbladder. So simply they will be spill over into the circulation. That's how it increases almost 20-fold increase in the bile acids in the circulation. Actually, bile acids they do not have any job in the systemic circulation. Okay, that is condition called as cholemia. Okay, high bili TLR5 knockout mice are cholemic. This is happening only in the mice which are developing high bilirubin. So that perfectly matches what we are thinking, that these mice suffer from cholestasis. Okay, and also we measured the fecal bile acids, they are almost undetectable, some of the bile acids. That means there is some problem for the bile acids to go from the bile duct via gallbladder into the duodenum. Okay, so this is pure lipid biochemistry here. So bile acids are absolutely required for emulsification of the fats, right? Lipids. Without bile acids, we cannot absorb any fat, including fat-soluble vitamins. So we asked whether they have any problems of classical consequences of cholestasis. The first thing we looked at is fecal total lipids. That is my CFTRA expertise. It came into handy. So we extracted the fecal total lipid, and you can see almost they have 46% increase in the stool lipids, condition called steatorrhea. Okay. Then we asked whether because of their increased excretion of the fat, whether they suffer from essential fatty acid deficiency. Okay. 18-2 is one of the essential fatty acids. As expected, they have reduced liver linoleic acid level. So it perfectly matches. Yes, they have cholestasis and also they have consequences of cholestasis. We also looked at the fat-soluble vitamins. Serum retinol, that is vitamin A and vitamin D, they have 50% reduced circulating vitamin and vitamin D. Okay, because of the lack of bile acids or compromised delivery of bile acids into the gut lumen. Okay, this is a schematic representation of bile duct obstruction. So if the bile duct obstruction happens at the microbiliary capillaries happens, it's not a big problem. But if it happens at the major bile duct, okay, then the cholestasis will happen. Spillover of bile contents like bilirubin and bile acids. And this can happen during hepatitis B infection or C virus infection, or there is a human disease called primary sclerosing cholangitis, or even in the women, that is intrahepatic cholestasis of pregnancy, they become susceptible to cholestasis. And unfortunately, there is only one drug of choice to treat cholestasis, that is ursodeoxycholic acid. Okay, including for the gallstones, they use ursodeoxycholic acid only. So basically, it is use thorn to remove the thorn. So ursodeoxycholic acid is also a bile acid with this very minor structure. You can Google it and see, very close to deoxycholic acid. See, deoxycholic acid is very toxic. By converting that into urso, you can use it as a drug to treat cholemia or high elevated bile acids. So the high bilirubin mice, they also have classical transaminitis, that is ALT, alanine transaminase, aspartate transaminase, and alkaline phosphatase. These are routinely done in the clinical lab to assess the liver function. Okay, ALT. So unfortunately, there is a miscommunication. ALT is not a liver function test marker. It is a liver injury marker. Whenever there is an acute injury, the ALT level goes up. If the liver is already in the chronic state or damaged, okay, you don't see the increase in ALT. So it is a lot of times misleading. So we have another mechanism to identify this kind of cholemic condition. Just last week we got that patent. So basically, high bilirubin mice has the classical symptoms of all the liver damage. Okay. So this is another. So now you'll appreciate the Johnny liver expressions, right? So when we continued feeding a inulin-containing diet for 6 months, you see the mice which are obese, still their liver is severely steatotic. Usually the liver will be dark brown color. Once they start accumulating lipids, it becomes this kind of color, okay, very pale. And you see the mice which developed high bilirubin, every lobe of the liver become tumor. Okay, and we confirmed this is a type of liver cancer because I had never worked on any type of cancer. So we took the help of one of the board certified veterinary pathologist. She also confirmed, and also we confirmed this. They are tumors at the serological, immunological, histological, because we were scared. We cannot publish some result without confirming it, right? And also we are blaming one of the nutrition's favorite food, inulin. So we have to attack the problem from all directions. So you see here, this is the tumor. When the tumor is growing, tumor cells are growing, it pushes the healthy cells like into the corner. Then you get the tumor border. Okay, I also learned I never worked on any cancer. And also we did some cancer-specific staining like glypican-3, reticulin, pentachrome staining for collagen, Ka67, Irritable. So every possible histology we did and confirmed that they suffer from hepatocellular carcinoma, a type of deadly liver cancer. Okay. So this is a collagen staining, and this is a distorted reticulin staining. This is for PCNA. It stands for, I think Subbarav Lab will be expertise on this. Someone hijacking my talk or what? So this is a proliferating cellular nuclear antigen, which is a very good marker for cell proliferation, especially it will be very high during a cancer, okay? All these dark brown spots are positive for this PCNA. And also we measured circulating tumor cells. Those are CD45 negative EpCAM positive cells, okay? They are only detected in the mice with liver cancer. And this is the stages of liver cancer. So at 1 month, you don't see tumors, okay? Usually it never happens within 1 month. They have, but hepatitis, they have hepatitis. And also by 3 months, they develop steatohepatitis. Steato means lipid plus inflammation. By 6 months, they develop tumors. By 9 months, every liver tumor, every lobe of the liver become tumorigenic. And by 12 months, they die with the hepatic encephalopathy. And you never think that mice has this kind of liver, tumors in the liver. Almost no external phenotype. The liver, the mouse looks very healthy. There is no ruffled hair, no hunched back, and no symptoms at all. But finally, the mice die by 12 weeks. So that is because of the hepatic encephalopathy. I don't want to go in detail because the ammonia accumulates and it is very toxic. That's how the mice die. Then we asked whether this kind of cancer can be transferable, because co-housing, usually it is one of the strategy in the gut microbiota research. They co-house healthy mice and cancerous mice. By doing that, we can induce cancer in the wild type mice. But when you asked it right, we did not prevent cancer in the TLF anacot mice. So that again underscoring only the bad bacteria can be transferable much easier, or they may be very resilient, or they can survive much better than the bacteria from the healthy mice. Okay, this is a co-housing, because the strategy is they are coprophagic, they eat the feces, that's how they share their microbiota. And we also did cross-fostering. So my postdoc, my graduate student, literally he lived in the animal house. Okay, he coordinated the breeding of 4 or 5 cages of mice, and whenever the mice deliver, he switched the mothers. So that means wild type mice is feeding TLF knockout mice pups, and TLF knockout its mom, that dam, we call it a dam, is feeding the wild type pups. So I told you, like, initial colonization is very, very critical. Okay, if it is messed up, that will have a long-lasting consequences. By doing so, we induced liver cancer in the wild-type mice, 2 out of 12 mice. But again, the liver tumors is not as aggressive as in TLF knockout mice. And finally, we asked, this is also one of the standard assay you have to do in the gut microbiota research, ablating the microbiota by giving broad spectrum antibiotic. So we use ampicillin and neomycin in drinking water for 6 months. Okay, so there is no tumors in the antibiotic given TLF knockout mice. That is not enough actually to publish in high standard journal. You have to use germ-free mice. So fortunately, my postdoc mentor, he had a TLF knockout mice in his germ-free mice facility. So we requested him, and he generously agreed to do the experiment for us. And you see, the germ-free TLF knockout mice failed to develop liver cancer. And whereas the conventional, we did only for 3 months, unfortunately, because it is very, very risky and painful to do 6 months experiment in the isolators. Okay, anytime contamination may happen. If it happens, again you have to start over. So because we have to give irradiated inulin containing diet, same diet, but it will be sterile. Okay, but anytime contamination may happen. That's why we terminated that experiment at 3 months. And so we relied on the bilirubin. The germ-free TLF knockout mice, none of the mice become hyperbilirubinemic. So I know few of them may go into research, but you should know how you should conduct any phenotype or microbiota dependent phenotype. So gut microbiota dependent phenotypes are highly variable. Okay, in the same university, people get one phenotype. If they move those mice into another building, the phenotype will be gone. Okay, so that it depends on strain and use of litter meds. It is a must. You cannot use your knockout mice from your colony, buy wild type mice from a vendor. You should not do that experiment at all. So basically, whatever is published now, we have to reconcile. So it is very difficult to believe those papers now. And also diet. So diet is vendor, whether it is a regular diet or autoclave diet, whether it is lab chow or purified diet. And also these vendors, they go with the seasonal variations in their food components. Whenever something is cheaper, they prefer for that. But the next season, if it becomes expensive, then they use something else. So those things also happens. And water. So it is not just regular water. Some people give chlorinated water, and if you buy mice from Jackson Laboratories, they will be always on the acidified water, okay, with hydrochloric acid. So that will prevent some growth of some pathogenic organisms. And bedding material, this one was really bummer for us. We never thought that bedding material is, we don't have to care, but it really matters, okay? And also the nestlets, whether you are giving cotton nestlets or paper nestlets is also important. This is all my personal experience I am sharing with you. And also frequency of changing cages. It is not easy, okay, just to go and put some food and water and after the exercise, it is not like that, okay? You have to monitor the mice regularly and you cannot change the cages every day. If you change that one, it is a stress for the mice. They eat more and they become obese. Okay, because they are coprophagic, right? Why they are coprophagic? Not for fun. They want to recycle some of the nutrients. For example, I told bacteria can be a source for vitamin K or biotin or vitamin B12. How do they get? Because in the colon, these vitamins are not absorbed. So when they eat their feces, so then they can be absorbed. So if you prevent that coprophagy by changing the case every day, then they have to eat fresh food, right? They have to eat more fresh food to compensate the loss of recycling nutrient, critical nutrients. And also the personal and the animal caretakers, to what extent they contribute to this kind of phenotype, I don't know. For example, if a caretaker, if he sneezes, when he is changing the cages, all his nasal bacteria can go into the mice. So okay, that is, I'm exaggerating the situation. That's why even those caretakers also have to cover their mouth with a mask. Okay, this is one of the paper they describe very well. So what are all the control experiments you have to do? It has to be meticulous. Defining the impact of host genotypes on microbiota composition requires meticulous control of experimental variables. Okay. So when I decided to move from Pennsylvania State University to University of Toledo, that was my great concern. After moving there, because this paper was not published, after moving to University of Toledo, if the phenotype is gone, I will be jobless. Okay, because I spent 4 years already on these mice. When I go to Toledo, if they don't develop liver cancer, it will be disastrous. Okay, so luckily the liver cancer became aggressive when I moved to University of Toledo. So no one can identify them as liver. Okay. See, they become fidget spinner. In the US, the kids, they love it. They're playing like that. Okay, it is a heart shaped, and you see, this is it exactly looks like an embryo, right? Liver. I know. I cannot identify that it is a liver. Okay, that means it is an aggravated liver cancer when I moved to University of Toledo. Okay, then finally, we also thoroughly focused on some of the microbial products like fecal LPS and immunoreactivity LPS, fecal flagellin and immunoreactivity to flagellin. This is my brainchild experiment where we fractionated the E. coli proteins on the SDS space and probed with either wild type serum, or all are immune fed only, and wild type serum or TLR5 knockout mice without liver cancer or TLR5 knockout mice with liver cancer. You see the more bands in the mice which develop liver cancer. That means serum as antibodies to E. coli proteins, suggesting that there are some lot of microbial products are going into the circulation in these TLR5 knockout mice. Okay. So, and we also did a quantitative RT-PCR for the total bacterial load. They have almost threefold increase in the total bacterial load. So this is the way now they represent microbiota composition. Do anyone understand? Cladogram. Okay. This is all the microbiota field is taken over by bioinformatics because they are very smart. They can generate all these. Yeah, I don't understand this, honestly. Okay. But now I understand. So basically what it says is the Clostridia, they have, when you feed a fermentable fiber, obviously the fermentable bacteria are increasing. The Clostridia is one of the notorious bacteria phyla which can ferment the fiber. And also they have ability to convert our primary bile acids into secondary bile acids. And also they have enriched with the proteobacteria which are under opportunistic pathogens, especially E. coli, which also have LPS and flagellin. So the bottom line is that TLR5 knockout mice with liver cancer, they have reduced species richness and alpha diversity in the liver cancer mice. So this is, they call it as cladogram. I didn't show you another LEFSE analysis. So where it shows some meta-transcriptomics. This is metagenome, okay? Meta-transcriptomics, what are the genes changed in the bacteria? So I'm not, because I have some concern, that's why I'm not showing that. So finally, the inulin is a fermentable fiber. So it's obviously we have to measure the short chain fatty acids in the cecum, which is an anaerobic bioreactor. So we used the GCMS. So we didn't see much difference in the acetate or propionate, whether in the cellulose or inulin, but in the inulin fed mice, they have more butyrate. Okay, it is a very potent proliferating agent, butyric acid in our body. There are no metabolic pathways where we can make butyric acid. Okay, there may some butyryl-CoA comes in some fatty acid biosynthesis pathway. Other than that, you never ever get butyric acid in any of the pathway. Okay, so inulin-fed mice has more butyrate. So then we targeted fermentation by contacting one agricultural scientist, where he is trying to inhibit fermentation in the horses, because in the Kentucky Derby, you know, so which is a big deal there. Like they have dedicated scientists to work on the horses. So they developed laminitis. So to prevent that, he discovered that giving them beta acids or hops, they used in the beer manufacturing to extend the life of beer, okay? Even in India also. So we use the humulus and lupulus in the drinking water at a very low 20 ppm, that is parts per million in drinking water. So by preventing fermentation by this beta acids, the mice were protected from liver cancer. And also their bilirubin levels are also gone down. So we also targeted them with metronidazole. So recent literature indicate that metronidazole completely kills butyrate producers. Okay, so just metronidazole alone can prevent the liver cancer in the TLR phenocot mice. And this is one of the cool experiments. So here we intervened with their diet at the early stages. I said within 2 weeks, the mice develop hyperbilirubinemia. Once they become jaundice, we switch their diet. Those mice got 100% cellulose without any inulin in the diet. So by switching their diet or switching their fibers from fermentable to non-fermentable fiber, those mice were protected from liver cancer, and also their bilirubin levels are gone down by giving cellulose instead of inulin. So this is how the gut microbiota is involved in generating secondary bile acids. So taurocolate is the major bile acid in the mice, whereas in the humans it is glycocolate. So this bile salt hydrolase is present in the Clostridia species. Okay, so basically they are not playing there. They use taurocholic acid as an energy source, because they all in the colon, they don't get much carbon source, because we absorb everything in the intestine, right? So every carbon they will make use of it. That's why they express 3000 types of glucuronidases. So the bile salt hydrolase, it cleaves the taurine and it uses for their energy purpose. Then you get the cholic acid. This is the primary bile acid. Okay, and again this primary bile acid can be dehydrogenated or dehydroxylated, okay, by epimerization. And again, Clostridia scindens and Clostridia sardellii, they are known to catalyze these reactions. And finally, you get potent detergent and hepatocarcinogen deoxycholic acid. So Clostridia cluster, they express bile salt hydrolase, and also we measured their fecal bile salt hydrolase. Okay, they have strikingly elevated bile salt hydrolase activity in their feces, suggesting that majority of bacteria, they have the ability to generate the secondary toxic bile acid. And we profiled individual bile acids in the serum. See, you see the every primary, secondary bile acids are elevated, and the same bile acids are almost significantly reduced or undetectable in the feces. And this is the major bile acid which can cause severe injury. So we also targeted just gram-positive bacteria, because Clostridia belongs to gram-positive. Okay, vancomycin. So vancomycin also prevents liver cancer, and you see vancomycin treated mice we did not detect any deoxycholic acid or lithocholic acid, but there is no change in the bile acid level. Even after giving antibiotic, they keep synthesizing bile acids. That makes sense. Why should antibiotic inhibit bile acid biosynthesis, right? That's why this is a good readout. Vancomycin is working because there is no bacteria to generate secondary bile acids. So we also targeted cholestyramine. Is one of the widely used treatment for the hypercholesterolemia. Basically, the people with high cholesterol level, they take grams of cholestyramine. It won't be absorbed. It is basically a resin that binds luminal bile acids and preventing their enterohepatic circulation, because 95% of the bile acids are reabsorbed in the ileum. Okay, if you prevent that, then more cholesterol will be degraded into bile acids. So the net effect will be reduced blood cholesterol levels. That's how the cholestyramine works. So by doing that, if you give cholestyramine, the mice are protected from liver cancer, but still the liver is not healthy. They have some kind of fibrosis. So I will end with the mechanism of why these mice develop liver cancer with fermentable fiber. So first we looked at the apoptosis. So the mice with high bilirubin, this is at 4 weeks. They have massive cell death at the portal triad area. The green cells are dying cells. Okay, and also those in the same liver sample, they have abnormal proliferation. So that means there is abnormal cell death and abnormal cell proliferation. So what could be what could happen in these situations? There is a rapid cell death, rapid proliferation. Can we guess what could happen in this situation? During inflammatory condition, there is a rapid death, rapid proliferation. High chances for DNA, high chances for
apoptosis.
Errors, yes. So whenever there is this kind of things happening, there will be a high chance for errors or DNA mutations. Okay. So we also at the early stage we asked for what kind of immune cells are causing hepatitis. Okay, because there should be some immune cell infiltration to cause the inflammation. So we screened all the immune cells. We found that neutrophils are the one which are going at the early stages. So neutrophils are notorious in generating reactive oxygen species because they have a pro-oxidizing enzyme myeloperoxidase, and also they have notoriously active proteolytic enzyme elastase. It can chew up any protein comes on its way. Okay, and also the neutrophils are so a good source for NADPH oxidase. Okay, and generating reactive oxygen species. These reactive oxygen species also they can react with any biomolecules. Okay, and especially the polyunsaturated fatty acids, when they were attacked by reactive oxygen species, the final end product is malondialdehyde. So it has 2 aldehyde groups. It can react with any amino group either in the protein or DNA, forming a Schiff's base. Okay, finally resulting in the DNA damage marker 8-hydroxydeoxyguanosine. As so when we measured their levels in the serum, they have high and also increased 8-hydroxydeoxyguanosine excretion in the urine. Okay, so what we are now focusing on is the bile acids derived from the gut microbiota, butyrate derived from the gut microbiota, and we are bringing bilirubin also into the equation. Okay, because bilirubin is a potent antioxidant. Earlier it was thought that it is a waste product, but it is not. It is a very good antioxidant. So we think that liver cancer is fueled by the deadly 3 Bs in the TLR phenocot mice. So I don't want to go into the detail because this is bile acids are killing the cells. Butyrate is proliferating the cells under the when there is a severe oxidizing environment, and bilirubin is an antioxidant. Okay, it is a complete mess in these mice. So we are still working on it. So finally, the result indicate that these mice develop liver cancer because of the severe immunosuppression. Okay. So I hope I convinced you that the germ-free TLR5 knockout mice they develop colitis. No, TLR5 knockout mice develop colitis, metabolic syndrome, fatty liver, and liver cancer, which are all absent in the germ-free TLR5 knockout mice or mice given broad spectrum antibiotic. So finally, we were successful publishing this paper, and our feeling was like this. Okay, it was really 5 years mental torture. Okay, conclusion. So despite the beneficial aspects of prebiotic refined fermentable fibers, the random or rampant enrichment in the processed foods should be approached with a great caution, as it may increase the risk of liver cancer. So everything pure is not good. Okay, if you eat the whole foods, for example, this is where I love to share with all of you in any of the seminars. So if you eat the chicory root, it not only has lots of inulin, but it has a small quantities of the inhibitor for fermentation. Okay, difructose anhydride. It inhibits the fermentation. Just imagine if you eat whole chicory root, okay, or whole foods, you are eating fermentable fiber, and also there is a small chemical or some small amount of inhibitors that balances out. But if you eat pure thing, you are missing that inhibitor for fermentation. So the fermentation go unchecked. This is a very good example for why we should depend on whole foods instead of processed foods or pure foods. Okay. So that's why we think that the refined fibers need to be redefined. So we are what we eat, that is the old one, right? But now we are what they eat. They means what? Huh? Gut microbiota, yeah. So I give all the credit to my team members because they really sustained my torture for last 5 years. And of course, they all got their benefit after this publication. And I was supported from NIH, NIDDK, and NCI, and also Crohn's and Colitis Foundation of America. And finally, I want to share my take it as funny, okay? I like this. So modern biological research, how we generate hypotheses, okay? It should not be the case. So according to Sidney Brenner, not me, don't quote me, okay? Sidney Brenner, we are drowning in a sea of data, thirsting for knowledge. Yes, absolutely, that's fine, no, nothing wrong with that. But the approach is very wrong, okay? Most biology today is low input, high throughput, no output. You agree with me? Yes. So we are becoming lazy. Already by using Google, our brains started shrinking. Okay? I hope it won't become vestigial organ. No, okay, because we hate to remember anything. Everything Google, because we have cell phone. We are going in the wrong direction. You have to increase your memory, then the neuronal network will keep increasing. If you decrease it, neuronal network will keep going down, and finally brain will shrink. Now we have 1 kg, maybe after 100 years, it may become 70 grams or I don't know. We all have microcephaly. You agree with me? So don't use the omics approaches to derive an hypothesis. So reviewers comment that you are doing a fishing expedition. I am very harsh on this, okay? I agree. That's what people think. That means you don't know what to do. You will do something, you get 30,000 gene or 30,000 proteins, either proteomics or lipidomics or whatever. And then you pick the top gene, which is highly expressed, and you pick the low gene which is down-regulated, and you do the experiment. That is, we don't need PhD for that. Any high school kid can do that. Agree? So, and also it is very expensive. And I am not undermining, this is a great technology, but we do not have the capacity to mine them. You have to be perfect in all metabolic pathways, all proteins, all enzymes, all metabolites. Then you can do something out of it. Otherwise, the data, raw data will be sitting in your computers, maybe for centuries, nothing happens. And this is also another one, I loved it. Of course, it's not my own idea. I copied from someone else. If you want, I can share with you. So biological version of mechanemerous fallacy. So measure whatever can be easily measured. That's what we are doing now, okay? Disregard that which cannot be measured easily. And presume that which cannot be measured easily is not that important. Presume that which cannot be measured easily does not exist at all in the system. So focus, strategic, tactical, and management efforts from number one. So measure whatever can be measured easily and make your story and publish and get promotions and other things, okay? So with that, I thank you for your patience.
Now session is open for questions. If you have any questions, you can ask.
Vijay, I have a lot more questions.
Yeah.
So why only liver? Why not other organs?
Huh?
Why cancer was there on liver, not in other organs? Did you look at it?
Yeah, we tested, there is no any metastasis of the nearby organs like lungs or kidneys, or usually kidneys are resistant to cancer. We didn't see any, even though they have the circulating tumor cells, we did not, maybe it may require more time to go into bone or other organs.
So it may be related to bile acids, sir. Bile acid is directly related to
GI also will have a lot of bile acids.
Yeah.
Right? And proliferation rate of GI is much, much bigger than the liver, though the metabolism will be less here. So is that the reason? Could be that you don't see much?
No, actually inulin is doing its function perfectly. Okay, colon is absolutely fine. Even though I said the TLF knockout mice had a history of developing spontaneous colitis. That's why we were curiously looked at the colon in the entire Swiss roll. There is no any kind of minor histological changes. Okay, they do not have. They have some splenomegaly that we attribute it to liver cancer. So we think that all the short chain fatty acids going there, and also the spillover of, we also actually the perfect way is to measure the liver bile acids. Yes, they do have very high. We spend so much money because each mice we have to take one piece of liver, right? So it is impossible to take the same lobe where we get the similar levels of bile acids, but that didn't happen. One lobe has less, one lobe has more. Finally, all the data points were random. They didn't fall into like a grouped one swap we get in the serum sample. So I think the three Bs are playing a major role.
The next question was, so only 30 or 40% of mice are getting tumor. Is it that to start with the gut microbiota was different in these mice? You think like that?
Yes,
or
yeah.
To start
from the day zero, when they are on lab chow, before putting them on ICD, they have increased bacterial fermenters, Clostridia.
So it's more of reactivation, or it was from exogenous source, you think?
No, it's the same bacteria. It is further booming in the gut because of the giving the fermentable fiber. Inulin.
No, but it's happening only in the 40% mice.
Yes, yeah.
So you think the basal level of this bacteria was different to start with? And what could be the signal for this carcinogenesis? I know you put these pictures, but what is the ligands which have secreted from the Clostridia bacteria that is going into the bloodstream and causing, or is it just the intestinal porosis more?
No, I don't think they are even as good as you can, we can call them as opportunistic pathogen like E. coli. Okay, the Clostridia, they are very good fermenters, obviously, because they have food is there, inulin. Okay, they are fermenting, and they also have equipped with the bile salt hydrolase, so they can deconjugate the primary bile acid. And there are other Clostridia species which further convert them into deoxyfolic acid. I don't think it is because of any toxins they are releasing.
But you see that, the E. coli antibodies,
Yeah.
antibodies against E. coli was
Yeah,
more.
it is a consequence of liver cancer. We try to detect some bacterial RNA and bacterial DNA in the liver. Again, it is we are picking some wrong lobes. Okay, even if you pick the same lobe in every mice, the bacteria may not be there in one lobe. So that's why we didn't get very conclusive answer for the bacterial translocation into the liver.
Thank you. Very good it was.
Okay.
And also I have a few questions. And bitrate. Bitrate is a well-known anti-cancer compound. Many people already published a lot of papers. Even I used to treat bitrate to breast cancer cells. They used to die with bitrate. But here, bitrate is a proliferation compound, like it is supporting proliferation.
So here, in your case, you are supplementing with only butyrate. You see the other conditions here, they are cholemic. Not all conditions, butyrate can act as an anti-proliferative or whatever. Here, the other metabolites are different. They have increased bilirubin because bilirubin has to go into the duodenum. That is not happening. It is an antioxidant, and they have increased bile acids and also neutrophil infiltration. But in your case, you might not have all these additional features to have the butyrate to show the negative effects. Right? I know everything was controversial. That's why I was very scared whether this.
Even one more thing, like this one, what is that? Bile acids. Obitocolic acid is approved by NIH for liver diseases, but these bile acids are causing cancer.
Yeah, we had that idea to supplement obitocolic acid in their diet, but it is very expensive, and also. Reviewers are not, if you propose anything like that, it is already known, it is routine. So I don't, but that's why it is still at the proposal state only.
And one more thing, like UPenn result, that liver cancer, the image looks different compared to Toledo result. Toledo result looks like somewhat, what is the abnormality with the liver generation.
Yeah.
Like maybe stem cell related problem.
Yeah, that I don't know. I'm happy that the phenotype is reproducible. I'm still at the stage only.
But phenotype looks completely different. Like in UPenn result, it looks like exact cancer liver. But Toledo result looks like a metabolic abnormality, that liver deformation.
Yes, that's why I put the word reprogramming of the liver morphology. So, but still they are cancerous. Some liver atrophy is also happening, like the shrinkage of liver. So in the human liver cancer, it is very asymptomatic, okay, and also highly heterogeneous. Some people develop jaundice, some people do not. Some people, the liver becomes enlarged, some people liver gets shrunk. Okay, that's why it is called as deadly. Asymptomatic is also not good. Any disease, that is the whole problem, right? In the cancer, you cannot detect at the early stages. Only you will come to know when they are in fourth stage. It is already beyond any clinician control. Nothing will work. So they give high dose of radiation or high dose of chemotherapy. Finally, it results in mucositis. They die because of the mucositis and neutropenia, not because of the cancer. Yeah. Any more questions? Yeah. Yeah. Yes. Bifidobacterium is also increased. Yeah, because I don't know what is Bifidobacterium it secretes, so that's why you have to play smart here. So I know Clostridium, they are fermenters, they make short-chain fatty acids, they make secondary bile acids. If they put the Bifidobacterium, then people like you who are working on Bifidobacterium, what it is doing, I don't know.
Again, sir, once again, such a wonderful and enlightening presentation. Actually, it gave us abundant information about the bad effect of using refined fibers.
Yeah. And not only in any refined one is not good.
So just wondering, sir, did you make an attempt to see, that in the last presentation you said 1 in 250 people have some TLR5 defects or knockout, or maybe not be there TLR5. So did you make an attempt to see that if such people have consumed processed food for a longer period of time, have any chance of increased frequency of getting liver cancer?
Yeah, it is a wonderful idea, but if you think in the daily life, we eat different types of fiber. No one will eat inulin now, right? So very difficult to know how much fermentable fiber they are eating, how much non-fermentable fiber eating. So it is very difficult to do that kind of human studies. At least if we come to know that if in the liver cancer incidence is higher in the TLR5 knockout humans, that itself is a great thing. As a fiber and all, it is secondary.
So that the inulin alone, as your the omics or the cladogram showed that increased population of Clostridium bacteria. So what about the impact on other bacterium as well? Because you said that Clostridium population increased and all this, the problems started from there, leading to the liver cancer.
What about the impact on other bacteria?
Yes.
Yeah, I told like they are all interdependent bacteria species, and they have cross feeding also because of the increased Clostridium, increasing butyrate. So other bacteria may be thriving or other bacteria may be suppressing. So that's why we get all that microbial dysbiosis and also increased bacterial load. I don't care who are there or that for me bacterial load is important, and where are they encroaching the epithelia or just remaining in the lumen, or they are completely altered metabolic activity. Those are more important than just looking at the 16S sequencing.
Okay. Thank you, sir. Any other questions from participants? From east side or west side, please ask some questions.
Yes, no, everything is clear. Yeah. Huh? No. It will cure. I will bet. Okay, it will cure. Only the certain fraction of the mice, some inulin is not working. We already know, but I cannot disclose it now. Why that only 40% of the mice they develop? I don't think inulin will, definitely inulin is a wonderful food, but not for everyone. Okay, not all fibers are made equal. Some people are vulnerable to this kind of problems. And I forgot to tell you, like inflammatory bowel disease patients, like Crohn's disease and ulcerative colitis people, they should not eat any fermentable fiber foods, period. Okay? That they classified as FODMAPs. I forgot the full form, fructose, oligo, something like that. So they have to abstain from any kind of fermentable fiber foods. 90% of them will develop IBD flares or constipation or severe abdominal pain. It is well-known fact. Yeah, they can withstand cellulose diets, but not the fermentable fiber. So fermentation, again, it promotes bacterial growth and their metabolites. So you can...
no way of having probiotics with anti-fiber.
See, show me which probiotic, what is that, longitudinal study or cross-sectional study or whatever. Did anyone have a conclusive answer? Any probiotics or any, for that matter, any antioxidant? No, it is all personalized. There is no such thing that one size fits all. So you go and do what you want and you find out what is good for you, stick to that. We don't, I don't think anyone can make any food which is universally beneficial for the people across the globe. I hope you all agree with me. I don't think it is impossible to make maybe some things like banana, those kind of things, but making a formulation for everyone, I don't think it is. We are nowhere close to that.
Even they say that the gut microbiota of individuals differs from each individual. So obviously that the concept of personalized things are coming up. So its variation is too high. So you can't have, as sir said, cannot have a one size fit for all. Any more questions?
Thank you, sir.
Yeah.
Thank you, sir, for the wonderful talk. Now I would like to request Dr. Rajesh Kumar to hand over the mementos as a token of appreciation.
Okay.
Now we'll have a tea break for 10 minutes and later we'll gather for the next session as a valedictory function followed by the oral presentation.
Good afternoon and welcome back all. We have now gathered for the valedictory function for our guests. I welcome our principal, Dr. Basavan Gaudappa. He is here today with us. I sincerely thank all our distinguished speakers for the brainstorming session since the last 2 days. We deeply acknowledge them for... Good afternoon and welcome back all. We have now gathered for the valedictory function for our guests. We deeply acknowledge them for rendering valuable insights into the principles and practices of molecular epidemiology of infectious diseases. So it is now time to extend our heartfelt gratitude to our esteemed guests for their valuable time at JSS. I request Dr. Suma, our Vice Principal, is not here. Okay, I request our Principal, Dr. Basavan Gaudappa, to hand over the memento to Dr. Poornima Madivannan as a token of gratitude. I request Sir to hand over the memento to Dr. Karl Krupp. I request Dr. Subrao to hand over the memento to Dr. Matam Vijay Kumar, sir. I request Dr. Prashant, sir, to hand over the memento to Dr. Kerry Cooper. I also take this opportunity to thank Dr. Lee Riley for gracing the special occasion with his esteemed presence online. Thank you, Dr. Lee. So with this, we have come to the end of the program. So we'll next proceed with the oral presentations. Of Dr. Lee, I request Dr. Poornima to receive the token of appreciation. Photo session with them. Photo session with them. So it one followed by oral presentation. We now have a photo session with our guests. So followed by that, we'll have the oral presentations.
Before that, I would like to take this opportunity to thank all of you for taking your time off and putting a lot of efforts, traveling and spending every minute for the last 3 days in enriching the knowledge of our participants here in this proposed project. I'm sure Purnima was telling they have utilized every minute of our the effortness or efficiency. And Krupp was visibly tired and he said, you have made him totally getting exhausted, but he enjoyed all the sessions because they were all good, and your feedback is the one which is keeping them still active and agile in spite of 3 days effortful work here and in spite of having traveled and jet lag. Thank you all. Thank all of you for real pain taken and conducting a very, very fruitful, meaningful, effective workshop here, INDO-US workshop. Any research, if it has a tag of translational impact, it will always be respected, meaningful, and more fruitful. So hopefully all of you who have participated will have learned a lot from these experts. And any doubt, any clarification is there, I'm sure they will be happy to get back to you in a mode of communication that you can establish with them. Thank you all for actively participating. Hope this will be giving you an impact and more impetus to do more research. Thank you.
We request some feedback from the participants so that our speakers know how you feel about it. From outside JSS.
I request all the participants who listen to the conference.
Just one feedback. Any one person outside JSS can volunteer to just give a gist of how we went on for this.
So they have another 4 days of workshop. So I think they still.
Hello.
Yeah.
Very good afternoon to everyone. I'm from Dr. D.Y. Patil Medical College, Pune. First of all, I would like to thanks to the organizing committee as well as the JSS Medical College for organizing such an informative workshop. I think all the speakers, they are not good, they're extremely good. The information they shared with us, no, it's a fact, it's a fact. The information they shared, I think it's a lifelong for us, because every topics, according to this level, they covered, and most importantly, they taught us how to write the manuscript as well as how to write something to sanction a grant. It's very important for the early researcher, I think, in future. So that's why I'm really thankful for the organizing committee and the GSS Medical College, Mysore. Thank you, sir.
So before that, I request Dr. Subrao sir to hand over our token of appreciation to our principal and Prashant sir.
स्विच कौन सा अभी फिलहाल बंद कर दो स्विच कौन सा फिलहाल स्विच नो सर बटन सपोर्ट आवाज आ जाएगी ना कौन सा बटन दिस वन
Dr. Mathan Vijay Kumar, Dr. Carrie Cooper, Dr. Prasanna Kumar, and Dr. Ravindra Kumar. And I request Dr. Shweta, Assistant Professor, JSS Medical College, and Dr. Devanand, Assistant Professor, Department of Biochemistry, to coordinate this session. And instruction for the students: each student will have 10 minutes for the presentation, and you hear a bell at 8 minutes, followed by 2 minutes to wind up the presentation, and later it is open for the discussion. Now I would like to request Amitesh Datta to start the presentation.
Shall I start, sir? Pointer. Table. So, very good afternoon to everyone. The title for my today's presentation: Characterization of Acinetobacter calpaciticus Acinetobacter baumannii complex (ACV complex), a potential nemesis for an intensivist. Presented by Amitesh Datta, Dr. D.Y. Patil Medical College, Pune, India. I'm going to present my topic under the following headlines. First is background, which will be included the research gap, objectives, methodology, result and discussion. It will be included the conclusion, future work, and references. The abbreviation I used here: ACV complex, Acinetobacter calpaciticus Acinetobacter baumannii complex, ASBL, extended spectrum beta lactamyces, MBL, metal beta lactamyces, HAI, hospital acquired infection, ICU, hospital intensive care units. So background. Genus Acinetobacter consists of a short pleomorphic non-motile gram-negative Lactobacilli. ACV complex collectively includes Acinetobacter baumannii, Acinetobacter pt, Acinetobacter nosocomialis, Acinetobacter C40, Acinetobacter discerniae, and Acinetobacter calpaciticus. It can survive under varying environmental conditions for long duration, thereby causing an outbreak, endemic infection, as well as hospital-acquired infection. ACV complex, once considered as opportunistic organism, has emerged as an important nosocomial pathogen, especially in hospital intensive care unit. The increasing role of ACV complex in the establishment of nosocomial infection is a worldwide concern, and its antibiotic resistance pattern during recent years had made this infection difficult to treat. Therefore, having information regarding the prevalence, distribution, changing trends of epidemiology, risk and virulence factors associated with infections, antibiotic susceptibility pattern, resistant genes of Acinetobacter species are crucial. So we planned the study to isolate and identify the ACV complex as it turned from opportunistic organism to important nosocomial pathogens. To determine the antibiotic susceptibility pattern, which is continuously changing from time to time and place to places, thus institutional antibiogram is very important to know. To detect the antibiotic resistance determinant, as there is a significant differences in the behavior and spread of multi-drug resistance Acinetobacter species recovered from various geographical locations. This is the objective of my study: isolation and identification of Acinetobacter calpaciticus Acinetobacter baumannii complex (ACB complex) from clinical samples, determination of antibiotic susceptibility pattern of ACV complex, phenotypic detection of extended spectrum beta lactamyces (ESBL), AMPC beta lactamyces, and metal beta lactamyces from ACV complex. That is the methodology. Study type: it was an observational and cross-sectional study. The sampling method, we used the purposive sampling techniques. The inclusion criteria: the suspected patient for ACV complex infection. The exclusion criteria was the samples from the patient with hospitalization less than 48 hours. The study setup: the study was conducted in Department of Microbiology, Dr. D.Y. Patil Medical College, Pune. The study duration: it was September 2021 to Feb 2022. The sample size: the total 74 isolates were included in the study. The sample size, it was calculated. The sample size, it was calculated by considering the prevalence of the organism with the confidence interval and margin of error by using the WinPIV statistical software. That is the methodology. The identification is done by standard phenotypic microbiological procedure. As for example, the colony morphology on culture media, ground stain, positive catalyst test, negative oxidase test, the absence of motility, and the standard biochemical reaction. Again, the isolate, it was confirmed, the confirmation of identification done by automatic Vitek 2 compact system of biomerics. The antibiotic susceptibility pattern was determined by disk diffusion method and Vitek 2 AST curve as per latest CLSI guidelines. TSBL, AMPC beta lactamyces and metal beta lactamyces production was detected by double disc synergy test and combined disk diffusion test. So that was the result and discussion. Out of 74 isolates, 76% was obtained from ICU. And respiratory specimen yielded the maximum number, followed by blood specimen. The majority of the strains were highly resistant to most of the antimicrobial agents. All strains are susceptible to tigecycline and cholesterol. 61, that means 82% isolates were found to be multi-drug resistance MDR. The 3, 5 and 64, 86% isolates were screened positive for ESBL, AMPC and MBL respectively. The conclusion, the increasing Acinetobacter infection with high resistance pattern was observed in the study. In absence of new antibiotics in the pipeline and the limited therapeutic option, the prudent use of antibiotic, continuous surveillance and strict infection control practices could limit the emergence and spread of organism, especially multi-drug resistance Acinetobacter Bowmani infection. The future plan, in future I have planned for the genetic study. So as for example, the genotypic characterization of SCV complex by detecting the following gene, the RACAN BALA-OXA21 gene for genotypic confirmation, the BALA-OXA23-like gene, BALA-NDM1, TEM, CTXM, SHV and IMP gene as antibiotic resistant determinant. And we have planned for the virulence factor detection by this detection of the biofilm formation as well as CNF1, CSGA, CV, AC, and IUTN, as well as the plan for the sequencing and rest of the process. Thank you so much. So references of my.
Thank you, Amitesh Datta, for the presentation. Now it is open for the discussion.
Yeah, what is the prevalence of Acinetobacter infections in India?
What is the?
What's the prevalence?
What is the prevalence? It is
prevalence in Indian population.
It is 5 to 9%.
Why do you see? I mean, I don't really see the actual results that you are present. I only see the sentence wise. I mean, like a conclusion. This much percent was positive or negative. So, where is the actual results? I mean, you did a lot of process, but I wanted to see the result. I mean, what you presented was like a conclusion, like a summary of your result findings, not the experimental result. Can you understand what I say? Like in the last presentation, sir demonstrated, you know, there is a bar diagram. This is the percentage. This is the percentage. This is the result of this double diffusion assay, like that. But I see only the summary of your result.
So the Acinetobacter infection is increasing gradually. First, second thing, the strain were
increasing gradually. Where is the result? Where is the result there?
Sir, it was my part of my study. Still, based compared to at my place compared to my previous study.
In the simple words, I wanted to see the bar diagrams or some kind of a results from the methods that you have used.
Yes, sir.
Not like a summary of the results.
Right.
You got the point?
There you go.
Thank you.
It was like.
So, so what was the question that you're trying to answer when you went into doing all this? I mean, you said that in India it's 5 to 6%. What was the question? Why were you looking specifically for this in this setting and all that? What was the original question that you were looking to
Do you want to
answer?
know research question?
Yeah.
So what is the goal of your study?
So actually, I just want to know what is the reason behind it. The patient who is taking admission in the hospital, especially intensive care unit, or especially I can say the patient with intubation history, why they are getting very frequently the Acinetobacter infection. What is the reason of it? What is the source of infection? What is the route of transmission? What is the portal of entry? What is the mechanism of action behind it? What is the antibiotic susceptibility pattern? Why the resistance pattern is increasing? What is the drug resistance mechanism behind it? That was my research question.
So just as notes, stay that in the beginning so that everybody knows. I mean, that's a great research question. That's a long research question. You got a lot to work on, but putting that up right away so that we all know, okay, that's what he's going for would be good, you know, in your presentation. Does that make sense? So state that at the beginning when you're giving your introduction, you go through your Acinetobacter, say, this is what I want to look at. Because we know all this, this is where I'm going. Okay?
Thanks.
And if this bacteria causes any disease?
Yes, sir.
If this bacteria causes any disease?
I not get. Yeah, definitely, sir.
So what type of disease?
Acinetobacter. Basically, it is causing the hospital-acquired infection. Most of the time, it is related to the pneumonia.
Okay.
As well as you can find out the wound infection, other bacterial infection is also related. Most of the time, what I am getting the incidence of respiratory samples most of the time, and it is from ICU.
Okay.
And I told it is most of the time what I noticed after admission of 48 hours or 3 to 4 days, especially the history of intubation. Patient is getting the Acinetobacter infection. In my setup, it is more than 175 ICU beds we have in our hospital, and it's very frequently we're getting the Acinetobacter infection that is related to hospital.
Does this bacteria produces any toxins?
So still now this virulence factor detection did not do. Phenotypic and rest of the antibiotics I'm doing. In future, we have planned to detect the virulence factor and
okay.
Actually, in a presentation, you should, as Sir said, you should clearly state your objective, slowly explain the methodology, okay, and the answer, results, what you got it, and the conclusion, okay. It seems you are very fast. I mean, and you know, the clarity wise was a little bit lacking. This is what I found it, but otherwise, it is a good presentation. Thank you.
Yeah.
Thank you. Next, I would like to invite Krishna Karthik, MBS, for the presentation.
Can you, I can't read any text. Can you put that video on the corner? Can you put the video on the corner? Because it, from here, I can't see anything. The text. I know it is not your mistake. They put too small letters.
To go. A very good afternoon to the respected panel members, colleagues, and my dear friends. Myself, Krishna Karthik, tutor and research scholar from the Department of Microbiology, presenting my abstract on genetic diversity of Mycobacterium tuberculosis in Mysuru, their drug susceptibility profile and geospatial distribution. So tuberculosis is known for a disease of antiquity, and it is a disease of public health concern. So the magnitude varies across the setting depending on the socioeconomic cost, HIV burden, availability and performance of the healthcare system. So globally, approximately 10 million people has developed tuberculosis in the year 2020, and India accounted for around 26% of the total TB cases. So TB report from India has shown variations in the trends and case notification rates by the region. So the government of India and its partners like NTEP have taken significant steps to prevent, diagnose and treat tuberculosis. So comparing the share in the global burden, the knowledge about the genetic diversity of the strains prevailing in the India is inadequate. So the study of genetic diversity of MTB is important for effectively controlling the emerging drug-resistant strains, which enables us to understand the transmission dynamics. So Mysore, which is a city in the Karnataka state of South India, is well known for the section of tourists as well as migratory people from other parts of the world and India. So hence it would be interesting for us to know about the circulating genotypes and of MTB in this geographical area. So the objectives of our study is to determine the predominant lineages circulating in this region, to determine the drug susceptibility profile, and to know the geospatial distribution of the mycobacterial lineages through GIS mapping. So this is an exploratory study which is conducted in the Department of Microbiology, JSS Medical College and Hospital for a period of 3 years. So all the clinical samples that we have received in the designated microscopy center in the Department of Microbiology with the clinical suspicion of pulmonary tuberculosis and extrapulmonary tuberculosis were included for the study. So the specimens were processed for the culture, smear microscopy culture, and it is simultaneously subjected to solid drug susceptibility testing for the first-line anti-tubercular drugs, followed by DNA extraction and oligotyping. So all the bands that were analyzed using Situate2 database to know the lineage. An attempt was also made to understand the transmission dynamics by spatial mapping of these lineages in the GIS mapping. So moving on to the results, a total of 354 clinical subjects were involved in the study, of which 211 has yielded the growth of mycobacteria. So on oligotyping, we have found like 21.8% of the isolates were orphans. So followed by East Asian and East African and Indian lineages, H1, Cas, Delhi, Lamb, T4, Cas, and Manu, Beijing and Manu, and bovine and Cameroon were the other lineages which we have found in this study. So this is the frequency and the lineage distribution what we have found in our study. So conventional drug susceptibility testing was performed, and 83.4% of our isolates were pan-sensitive to all the 4 first-line anti-tubercular drugs. 5.7% were streptomycin monoresistant, 4.3% were multi-drug resistant, 3.8% were isoniazid monoresistant, 1.9% were polydrug resistant, and 0.9% were rifampicin resistant. So of the 9 MDR TB strains, what we have isolated, 3 has belonged to the lineage of EAI, which is followed by 2 from the Beijing lineages, 1 from each of Lamb. So most of the orphans in our study, which were sensitive to all the first-line anti-tubercular drugs, but 3 of them were streptomycin monoresistant, and 1 isolate was resistant to isoniazid as well as polydrug resistant. So 22 isolates that were monoresistant to isoniazid as well as rifampicin belong to Cas, EAI, and Lamb, and other 4 were from the orphans. So all the lineages, like we have extracted the demographic data from the hospital information system, and all the lineages were point mapped using ArcGIS software, and the point density and the hotspot regions in the Mysore district was determined. So genetic diversity was much observed in Mysuru taluk than other 6 taluks. So combination of this GIS mapping and oligotyping lineages identified 2 hotspot regions among 7 taluks in the Mysore district with potential TB transmission. One being the Mysuru rural and other Nanjangud taluk with a wide range of genetic diversity. So this is how we have done the GIS mapping, which showed the potential TB transmission in the Mysuru taluk as well as Nanjangud. So coming to the conclusion and implications, the current study was aimed in isolation, identification of MTB, and to understand the frequency of drug resistance of MTB in this study setting. So it is also aimed to know the predominant lineages circulating in and around this geographical area from the tertiary care hospital, which is situated in Mysuru. So this study highlights the importance of oligotyping, which is used as a tool for the molecular typing of MTB isolates. And it is the first kind of study which is being conducted in this region. So using molecular techniques such as oligotyping can help us in studying the transmission dynamics of Mycobacterium tuberculosis. So results of this research has revealed there exists a genetic diversity and several lineages are being circulating in this region. So this may be linked to the ethnicity of the population residing and their migration values. So these results has formed a baseline data of the circulating lineages. So this study also has several other operational research programmatic implications that can be implemented to design the policies and new strategies under the national program. So this can be implemented in a setting where the TB burden is high to moderate. So though India having the highest TB burden and looking into the national program currently, there is no strategy or the policy that is based on molecular epidemiology. Hence, there is a dire need in adapting the newer technologies and innovative technologies. So the recommendations of this study, a multicentric study with a larger sample size will really help us in determining the prevalence of drug-resistant tuberculosis. It will also help us to know the predominant lineages that are circulating in and around Mysuru. So the combination of other molecular methods such as whole genome sequencing will help us to know which is the feasible method and economic method in knowing the molecular epidemiology. So particular age group like when it is compared with the adults or pediatrics and patients with the comorbidities such as HIV, hypertension, diabetics, smokers or alcoholics can be further selected, and further analysis of the genetic diversity and its association with the severity of the infection can be studied. Thank you.
Thank you, Krishna Karthik, for the presentation, and the session is open for the discussion.
So I have a couple of questions. One, how does this genetic diversity here in Mysore compare to like other parts of India, other parts of the world? And two, the antibiotic resistance that you found, how does that compare to what's seen in other parts of India, other parts of the world?
Sir, actually, according to the literature, it states that southern India has particular lineages and northern India has particular lineages. So in the Mysuru, in our study, like we had found like there is a distribution of several other lineages when it is compared with the other studies. This is because like Mysuru is a place where the tourists come and migrate. So there are several other lineages that are circulating in this area. So according to the literature, actually it is EAI lineage has to be predominantly being in the South India, but we have distribution of EAI as well as caste, which is most commonly seen in Northern India.
And what about the antibiotic resistance? Just the general, like the percentages that are resistant.
So majority,
like Northern India and such.
Majority of our isolates, like something around 83.4% were pan-sensitive to almost all the anti-tubercular drugs. So the resistance is correlating with the other studies also.
Thank you.
Yeah, I have a question. So where are the figures?
Sorry, sir.
Where are the figures? Did you try, or put it other way? Did you try to put your data in the form of figures? You are simply reading all the text what you wrote. Actually, you don't any. You can give us the slides and we can read on our own, right? So you should try to put your data in the form of figures.
Yes, sir.
And also, whatever the text you put, it is a suggestion. It is not a question for everyone. Okay. Minimize your text and try to speak your own words instead of reading the entire slide for us. We can read on our own, right? And also, if someone is asking question, listen first. Before they complete the question, how can you answer? You don't know what they are going to ask, right? I also did this doing the same mistake. I learned. That's why I'm telling everyone. Listen the question first. Okay.
Is there any link with air pollution and severity about TB?
Sir, that was not made in our study, sir, which is we wanted to do in the future.
Because you showed that map, no? If you see that Mysore city, you see more severity.
Yes, sir.
Compared to Nanjangooda and the other towns. That's why I asked.
Sir, we have done a previous study where we have taken the smear positive cases, which have been notified in the whole Mysuru district. So that study and our study was correlating with each other. So we also has got like Mysore rural has maximum number of cases, which is followed by Nanjangud Taluk as well as T Narsipura. So in our study, we had found like hotspot regions in Mysore Taluk as well as Nanjangud Taluks.
Even Nanjangud has more air pollution. Maybe you can check that. Okay, then thank you.
Yeah. So out of, I think, 354 subjects, I remember around 4.3% were MDR, multi-drug resistant isolates. Where were they mapped?
Sorry, sir?
From where were those samples were mapped?
Sir, actually, 2 of them were belonging to the Mysore rural only, and 2 of them were belonging from Nanjangud Taluk. So we couldn't exactly show like the drug resistant patterns in the map, sir, because we tried to concentrate the map, the lineages. So it is a bit difficult for us to show the drug resistant patterns in the map. So if we are taking only the drug resistant strains and we are mapping, probably we can show it.
Okay. So in a more of like a theoretical questions, what's the main reason for development of multi-drug resistance from TB?
Sir, it is because of interruption with the anti-tubercular therapy. So if the patient is not taking the anti-tubercular therapy properly, that can be a major cause. So apart from that, any other mutations or contact with the MDR TB persons can also be a major cause of acquiring multi-drug resistant tuberculosis.
Audience?
In Mysore, we are not sure, but like we haven't concentrated much of EPTB in our study because coming to the microbiology, we received majority of the pulmonary tuberculosis samples, which are smear positive. So initially we concentrated on smear positive. Later on, we also has to include the extrapulmonary tuberculosis.
Okay,
thank you. Thank you, sir.
Thank you all. Now I would like to invite Deepali Saibun for the presentation. So
Good afternoon, everyone. My name is Dipali, PhD scholar under the supervision Dr. Mukesh Nandavi and College Delhi Pharmaceutical Science and Research University. My topic is evaluation of anti-cancer activity of Artemisia vulgaris stem hydroalcoholic extract and its fraction. These are the content of my study. Introduction: cancer is a group of disease characterized by a proliferation, differentiation, growth of abnormal invading normal tissues, organ, eventually spread to all part of the body. It is a multi-factor, multifaceted and multi-mechanistic disease requiring a multi-dimensional methodology for its treatment, control and aversion. An abnormal growth of tissue resulting from uncontrolled progressive proliferation generates a mass of cell is called tumor. A mass can be self-limiting, which does not invade neighboring tissue and hence poses no danger to life, called benign tumors. Tumor continues to grow, breaches the basement membrane and continues into the circulatory system whereby these cancer cells migrate to new sites in the body. This process is called metastasis and such tumor called malignant. Types of cancer: breast cancer, lung cancer and liver cancer. Breast cancer is a heterogeneous and complex disease, being the most common cancer in women worldwide. It is also the second main cause of death from cancer among women globally. And lung cancer is one of the most malignant tumor, highly invasive and common in both men and women. Various factors such as radon gas, asbestos, air pollution, exposures and chronic infections can contribute to lung carcinogenesis. Liver cancer or hepatocell carcinoma is the sixth most common cancer across the world. Approximately 24 people for every 1 lakh individuals were influenced by hepatocell carcinoma cell in a year. Causes of cancer basically tobacco, smoke, air pollution, radiation, virus, occupational factors, diet. These are the major causes of cancer. Worldwide scenario of cancers represent the, according to International Agency for Research on Cancer, reported that cancer burden rises to 18.1 million new cases and 9.6 million cancer deaths in 2018. And according to WHO in 2018, reported that every 5 men and 1 of every 6 women develop malignant tumor growth during their lifetime, and 1 of every 8 men and 1 of every 11 women die from the disease. In 2019, according to American Society Cancer Society, reported that 6,06,880 Americans will die from cancer in 2019, corresponding to almost 1,700 deaths per day, in which largest number of deaths are from various cancer. Breast malignant growth has position #1 disease among Indian females, according to ICMR. Pathophysiology represents the carcinogenesis, in which the transformation of cancer cell from a normal cell can occur due to 1 or more mutation in its DNA. It has basically three processes, in which initiation, promotion, and progression. Initiation is the first step, in which the normal cells are exposed to carcinogen, and promotion where the environmental factors favor the growth of mutated cell, and in progression is final stage of neoplastic growth, involves further genetic changes leading to increased cell proliferation. Activation of protoncogenes and oncogenes. Protoncogenes might be typical genes that normally control cell division, apoptosis, and differentiation, but when they are converted to oncogenes by the viral or carcinogen action, they tend to induce malignant change. These are the cancer treatment which can be provided by surgery, chemotherapy, radiation therapy, and hormonal therapy. This is the plant which I have chosen for the study, that is anti-cancer activity. I have chosen this plant because it has DNA damage marker properties, and also having a cell cycle arrest on various cancer cell line like MCF7, HeLa5, FR2, A549. Rationale of the study is aerial parts, including stem of the Artemisia vulgaris have been reported to be useful in tumor. No scientific evidence justifying its anti-cancer or anti-tumor properties is available till date. Therefore, the present study was undertaken to evaluate anti-cancer activity of Artemisia vulgaris hydroalcoholic extract and its fraction. These are the objectives in which I have selected the plant, collected, authenticated, preparation, its extract and its fraction, then phytochemical screening, and in vitro studies on these cancer cell lines, that is MCF7, A549, FR2, and HEBG2. Methodology, I have represented that the authentication of the plant was done by Dr. K.C. Bhatt, that is scientific principal scientist in ICAR Pusa, and prepared its hydroalcoholic extract by using its oxalate apparatus in the solvent ratio 30:70, and then prepared its fraction by using this scheme, in which I have prepared its 4 fraction, that is anhexane, chloroform, ethyl acetate, and methanol. This is the chart. And then in vitro anti-cancer activity have been performed in these cell lines, that is MCF7, FR2, A549, and HEBG2. Cell viability assay I have used for the in vitro studies, that is cell viability assay was assessed by MTT. Principle of this study based on mitochondrial dehydrogenase enzyme present in the viable cell, which convert yellow water soluble substrate into MTT, dark blue formazine crystal, which is insoluble in water. The amount of formazine product is directly proportional to viable cell number. And then preparation of reagents by using a literature, and preparation of cell culture. results, percentage yield of extract and its fraction. I have prepared first I have prepared hydroalcohol extract, then it's prepared its fraction, and this is the results in which quantity of the powdered stem taken for extraction, that is 320 gram, and quantity of hydroalcoholic extract obtained 10.38 gram, and percentage yield would be 3.24%. This is the and fraction of hydroalcoholic extract of Artemisia vulgaris, fractions, extract weight and its percentage yield, that would be calculated from the extract. And phytochemical screening, I have concluded. This is the hydroalcoholic stem extract, and I have concluded by phytochemical screening that hydroalcoholic extract contain the more phytochemicals for phytoconstituent that would be responsible for anti-cancer activities. These are the fractions. These are the results of in vitro anti-cancer cell lines. I have concluded, I have used 4 concentration, that is 0.1, 1, 10 and 100 micromole. And after the study, I have found that 10 micromole in hydroalcohol extract of Artemisia vulgaris represent the more inhibition on cancer cell lines. Conclusion, done. Conclusion, the study was carried out to evaluate anti-cancer activity of extract and various fractions of Artemisia vulgaris stem extract MCF7, FR2, A549, HepG2 by MTT assay. Various phytoconstituents such as carbohydrates, saponins, alkaloids, flavonoid, minerals, terpenoids are found to be in the plant extract and its fraction. The hydroalcohol extract showed the maximum IC50 value in all cancer cell line by MTT. The effective IC50 value was found to be in breast cancer cell line, that is 18.32. And these findings suggest that the reduction observed in the viable cells with the following treatment is due to cell death. In conclusion, the present observation provide preliminary data exposing hydroalcohol extract have potent cytotoxic activity against these cancer cell lines. Future outcomes are, these calls for further studies on the active components animal model for proper assessment of their chemotherapeutic properties, possible development as promising anti-cancer drug. We can further work on molecular docking studies like ligand-based drug design, where in silico studies of each compound of plant will be performed for cancer targets, designing of Artemisia vulgaris formulation, last investigation for in vivo. Thank you.
Thank you, Dipali, for the presentation, and the session is open for the discussion.
You have used 4 different cell lines, no?
Yes,
3 or
sir.
4. What is the reason why you
because extensive literature already reported on MCF7, HepG2, so that's why I've chosen 4 cancer cell lines.
But these are different types of cancer cell lines. You know that?
Yes, sir.
Do you know what?
FR2 and MCF7 are breast cancer cell line, and HepG2 is liver, and A549 is lung cancer cell line.
And you have mentioned that animal
Yes,
models.
I have mentioned that.
What is that animal model? In future plan, you have mentioned animal model. No?
Yeah, there are various animal models for in vivo studies like subcutaneous tumor model or subcutaneous malignant tumor models, and that's it.
What are the other in vitro assays you will do in future?
Sorry, sir.
In vitro assays, you did
In
only
vitro,
MTT assay.
in vitro, I can perform in vitro by SRB, dye exclusion, fluorescence assay, depending on the ability of institute.
That is sufficient to show that.
No. There are so many tests like. And on the basis of PhD, then I can explore more or more. Yeah.
Okay, thank you.
A comment and a question. So the comment, that was a lot of information you had in there. In the future, you might want to summarize it a little bit as you have it on your slides, and then just kind of, you could still speak it. what did you use as your controls for the cytotoxicity? Besides the compound that you're adding, what did you use to confirm, a negative control and your positive control for that?
Sir, I can't understand.
No, negative control and positive control, what are the controls you used for your MTTSA?
For MTTSA, MTTSA basically a cell viability assay.
No, that
in
we
which
know, but control, he is asking about controls, positive control and
control, yeah, positive control, that is streptomycin.
Okay.
Yeah.
And what was the negative control?
And negative control,
did you have like uninoculated cells to make sure that you weren't getting like natural cytotoxicity and things?
I think you didn't get the question. A known compound you need to use along with your these extracts. Known anti-cancer compound, like already known compounds.
Any other anti-cancer drug which is having therapeutic activity against the breast cancer or lung cancer, sir, asking for.
Okay. Basically, you are asking for a drug which can be used to cure cancer.
In your experiment, you need to use that drug along with your extracts.
Yeah.
Any other drug which has like anti-cancer activity you would like to use in your
in
study?
term of natural. Yeah. Yeah, like paclitaxel, vincristine, and vinca alkaloids. These are the anti-cancer which can be obtained from the natural products.
Okay.
See, what is the guarantee that your plant extract doesn't have any effect on the normal cell? Did you use the normal cells to see whether your extract has any impact on the cell viability on normal cells? That is the negative control you should have been used. Did you use that?
Sorry, sir.
You didn't get it.
No.
What is the guarantee or that your extract will not have any cytotoxic effect on normal cells, not cancerous cells? Do you know what is normal cell and a cancerous cell?
Yes, sir.
So did you use normal, did you treat those normal cells with your extract, and did you do that cytotoxicity assay there?
Sir, I have used a cancer cell line.
Do you have any literature that can suggest it will not impact the other cell line and it will not harm the severe cell line?
No, sir.
Does this plant is edible?
Sorry.
Does this plant is edible plant?
Yes, sir. I have planned a work. Plan. Huh?
Thank you.
No, sir. No.
Thank you. Thank you.
Okay, thank you.
Thank you,
Thank you all. Next, I would like to invite the participant, Bhavana.
So, good afternoon, one and all present here. I would like to take this opportunity to thank the organizing committee for giving me such a big opportunity to present my work in front of such eminent speakers. And myself, Bhavna, I'm pursuing my PhD under the guidance of Dr. Ashwini P., Department of Microbiology, JSS AHER. Okay. Klebsiella pneumoniae is one of the gram-negative bacteria that is found in the gut of the normal individual, healthy individual, and Coming to immunocompromised individual, Klebsiella pneumoniae can infect lungs, liver, as well as urinary tract. Multi-drug resistant pneumonia has almost conquered, we can see the entire world map here. The countries which are represented in blue indicate the affected, these countries are affected by multiple drug resistant Klebsiella pneumoniae. And these are the virulence factors posed by Klebsiella pneumonia in order to spread pathogenicity. So among these virulence factors, Fimbriae is one of the important virulence factor because it helps the organism in the formation of biofilm. So antibiotics and its resistance. So penicillin, cephalosporin, carbapenems, ceftazidime, and piperacillin are the antibiotics used against Klebsiella pneumoniae. But resistance gained by the Klebsiella pneumoniae against these drugs has resulted in the research of new drugs. So beta lactamases are a class of enzymes that bacteria has. These enzymes break the beta-lactam ring that is present in the antibiotics, and this is the mechanism behind antibiotic resistance. So, as I said, biofilm formation is one of the important virulence factor in Klebsiella pneumoniae. Biofilm formation is mediated by C-di-GMP. It activates transcriptional activator MRKH that drives the biofilm formation in Klebsiella pneumoniae. And in my work, I have targeted particularly MRKH protein. So we all know that medicinal plants, plants have a lot of secondary metabolites in it, and we can use these plants as medicine. So among all the plants I have chosen Ruta graveolens. It has a lot of medicinal properties based on the phytochemicals present in the plant. So it has almost 120 natural compounds in it, and it has antioxidant, anti-inflammatory, antimicrobial, and antiviral activity according to the literature. So these are my objectives. Coming to the first objective, I have chosen HS11286 Klebsiella strain as a reference and chosen KP-HS0098 protein in HS11286 Klebsiella pneumoniae strain to study, preliminary studies using in silico approach. So the sequence of the protein sequence in HS11280 strain was taken for homology modeling, and it was validated using Ramachandran plot, where the Ramachandran favored region, the amino acids fell in almost in favored regions. That is 96.57% of the amino acid present were in the favorable regions. So we took the protein for further studies. That is, we did molecular docking studies against this protein using rutin and two standard drugs, that is piperacillin and ceftazidime. Among all the four, rutin, that is the major phytocompound present in the Ruta graveolens, shows better binding affinity, that is -8.4. And we can see that dark green circles depict the conventional hydrogen bonding. So based on this results, we chose Klebsiella U25 strain for further studies, that is for in vitro studies as well. I have carried out in silico studies as well based on the previous data. So protein structure validation was done using Ramachandran plot, as I said earlier, and 96.57% of the amino acids were found in the favorable region,
And 5K EC was the protein that was responsible for biofilm formation in Klebsiella pneumoniae. So that protein was taken for validation and for molecular docking purpose as well. So even here we got that we could see that rutin, as I said earlier, this phytocompound showed better binding affinity, that is 8.9. So through this study, compared to that of the standard drug, that is piperacillin and ceftazidime, rutin showed better binding affinity. So by this, I could conclude that my plant had potential antimicrobial as well as anti-biofilm activity. Further, to check the antimicrobial activity of the plant, we did in vitro assays, that is antimicrobial assay and anti-biofilm assay. Coming to the result of antimicrobial assay, the first image is of the standard drug and the second one is of the crude extract, that is ethanolic crude extract was ethanolic crude extract of rutagraveolens was taken, and we could see that in 50 μg per ml concentration, we had zone of inhibition in the crude extract. Coming to the anti-biofilm activity result, A and B images, those are the images of cells which are untreated, and C and D are the images of the cells which are treated with the standard drug, that is piperacillin and ceftazidime. We could see a small amount of disruption on of the biofilm, whereas E and F, it is a SEM image where the crude plant extract showed better disruption of biofilm compared to that of the standard. So coming to the conclusion, rutagraveolens had better anti-biofilm activity as well as antimicrobial activity. So it can be developed into a novel drug. So further aspects, actually we have carried out the experiment from the crude extract. So further, we need to isolate and identify the particular phytocompound from the plant, and then in vitro assays need to be tested, that is genomic level. I need to identify the genes that are responsible for the virulence factor, that is biofilm formation, and check for the molecular mechanism what is going on. So these are the future aspects of my study. So these are the references, and thank you.
Thank you, Bhavna, for the presentation. Now it is open for the discussion.
So which microorganism produces penicillin?
Sorry, sir, which organism?
Which microorganism produces penicillin?
Penicillium, penicillium notate.
Penicillin, what?
Notate.
No, how do you get penicillin? Which microorganism produces penicillin?
Penicillin.
Huh?
Penicillium.
Huh?
Penicillium.
What is that microorganism? Is it bacteria, virus, or what?
It's a bacteria.
Okay.
What is the Ramachandran plot?
Ramachandran plot, the torsion angle of the amino acid, the rotation of the torsion angle can be checked using Ramachandran plot, that is phi or psi.
Psi what? You know who is Ramachandran?
Yes, sir.
Who is it?
G.N. Ramachandran.
You are not confident. You need to be confident. And also, what is biofilm?
Sir, when bacteria is in a culture, when the bacteria grows after reaching a certain threshold number, these bacteria start to communicate with each other, like using quorum sensing. So using this quorum sensing method, they colonize, and external membrane-like thing is formed around the colonies of bacteria. So this is biofilm.
How the transcription factor is linked to biofilm? You did this or docking and all those things, no?
Yes, sir.
How does this transcription factor is linked to biofilm formation?
Sure, I didn't get the question.
You did the docking studies, no?
Yes, sir.
What is that protein?
Biofilm forming protein.
What is that protein? Like
5K-EC.
Is it a transcription factor?
Transcriptional activator. Yes, sir.
How it linked to biofilm formation? It induces some genes?
Yes, sir.
What are those genes?
Sir, I need to go through, like I've done the work based on the protein level. So I need to study the genomics of behind the biofilm formation.
And also you showed that biofilm picture. Where did you took that picture? Did you do here?
Sir?
Where did you do that picture? Like
SEM analysis.
Yes, the biofilm picture. Did you experiment here in lab?
Experiment I have done in the lab, sir.
Which microscope you used to take that?
Sir, SEM images was sent outside with the help of my guide. For imaging, I sent it. It's as the SEM, microscope was not available in the department. I sent it for outside for the analysis. For the images.
Okay.
So in your presentation, you said that the probable back-to component present in your extract could be because of the routine because in your docking studies, you are done with the routine and the transcription activator, yes, whatever you're saying it. So, but when you go to the assays, like biofilm assays and other assay that you have shown it, did you use routine there?
No, sir, that's what is my future aspect. Actually, I need to isolate the different phytocompounds present in the plant and then need to check the activity of routine in vitro.
But you in your first part of your result, you have done the docking studies with the routine and other, yes, protein, right? So why you didn't include routine in your while doing that assays, biofilm assays and other assays? You have used the antibiotic and you have used the negative control, but yes, what is the control for the phytochemical compound?
Sir, for in silico assay, I just took the phytochemicals based on the literature. So further, I need to isolate the different phytocompounds and then again check for the activity. So that is my, in future, I need to do this, sir. So.
Okay.
Okay.
Thank you.
Bhavna, I have a question for you. Why is the problem you have tackled worth tackling?
Sorry?
Why is the problem you have tackled is worth tackling?
Problem that I have taken is worth tackling.
Yeah.
Yes.
What?
Because biofilm
like same work has already done in the past, na? So what is the novelty in your work?
Novelty.
Number of literature is available on the Google Scholar and PubMed and everywhere you can see. Then why did you choose this?
We get a lot of papers based on the biofilm activity or anti-biofilm activity.
Biofilm formation, in this field, the work is going on from the last 40 to 50 years, yes, isn't it? And the biofilm formation plays a very important role in urinary tract infection, isn't it?
Yes.
So why did you choose only this? I just want to know something.
Okay, can I answer?
And yes.
Can I?
Yes, you can say.
Okay. Klebsiella pneumoniae, the problem, it's an old one, but MDR, multiple drug resistant, it is emerging now. And biofilm formation and anti-biofilm, a lot of papers are there, as you said. But my point is in the point of my plant, that is Rutagraveolens, using this plant, I didn't find, according to me, the plant was novel. It's not that Klebsiella is
novel means you have identified the activity of biofilm formation of this plant. Novel means what? You have developed the activity for this plant for biofilm formation.
Yeah, for this plant. That is.
So for this plant, no work has done in the past.
What is the common name of this plant?
Rutagraveolens.
The common way.
Nagdali.
Sarpagandhi.
Yes.
Sarpagandhi.
Yes. No, sir.
Not Sarpagandhi.
No. I think I have the image of the plant, if I can show. This is the plant, sir.
Plant. What is the common name of this plant? Actually, I'm not familiar with this.
It is a local plant here in around Mysore city. Even in Karnataka also, we find this all over Karnataka. So we call it as Nagdali. So this is the common
actually,
name.
I was asking something from you that explains something new. So that because of your research, we did not know before, and now we know. Because these guys and like sir and all the participants and all the international guests, they are like more and more knowledgeable, isn't it? So please tell us something different that you are doing in your research.
Yeah, but
because they are like
I know, but I have now I have started my work and
whatever she has done, she has shown it. What else you want? She has not done it. What else you can show?
The work has already done many times, I think.
And also one take is suggestion because this is a common plant and also
No, sir, I'm
many
not
people
from the-
have done already this antimicrobial activity from this plant.
No, actually, sir, I want to know something. Actually, my expertise in diabetes. I'm working on diabetes, sir. No, I'm not from the infectious. Like
I have I'm stepping into this. I mean, PhD right now. Recently, have joined. So this is my preliminary work. So the work need to be done. Antioxidant assays need to be done. Anti-inflammatory, anti-diabetic. If you want, you can do it. So I mean, sorry. If I'm rude, I'm sorry. I'm sorry.
Good answer.
Sorry, if I'm rude, I'm really sorry.
So please give us some suggestion and advice. Like we are entering in this area.
No,
I'm
she
doing-
will give later, like after presentation, she will give. Just we can stop now.
Actually, I'm not satisfied with the question. Okay, thank you.
Thank you.
I have one question. Already in market, so many antibiotic drugs are there. So you are saying that your plant extract will be a good novel drug. So have you checked, means compare with previously already FDA approved drugs with your means proposing drugs, means the plants?
Yeah, as I said, piperacillin and ceftazidime I have tried with. It showed better. I mean,
how you compare? Means is there any result? How you compare?
I had antimicrobial assay results were displayed. Like depending on the zone of inhibition.
No, I'm not taking. I'm talking that in market, so many antibiotic drugs are there.
Yeah.
So they are also working in a good way. So you are telling that your plant extract will be a good drug in future. Means so how you are telling, in what basis you are telling that your plant extract will be a good drug? Means already so many drugs are there.
Can I answer? So in my study, I compared the drugs. I mean, like crude extract with piperacillin and ceftazidime. I agree with you. There are a lot of drugs available in the market, but I need to check. But this can be an alternative for antibiotics because we are doing this in our in order to combat antimicrobial drug resistance. We are doing the study. So alternative to antibiotics, we can use plant extract or like that. That was my approach.
That's good, but already a lot of antibiotic it's showing good results. So why will choose for as an alternative? Means if it shows a good result, it's an excellent result, then we'll go for that. Okay, it will be alternative pathway, means alternative drugs. But a lot of antibiotics are showing good results. So why will choose for alternative drugs?
If all the antibiotics showed better results, then I think MDR strains wouldn't have come up. I mean,
Bhavana, we can stop here. We can go to next presentation.
Now I would like to invite Priyanka for the presentation.
Good afternoon all. Myself Priyanka. I'm a research scholar from Department of Pathology, JSS Medical College. Today I'm going to discuss on correlation of clinicopathological factors with estrogen receptor expression status in breast cancer patients. Introduction. What is cancer? We all know what is cancer. Cancer is a disease in which abnormal cells divide uncontrollably and destroy body tissues. We know many types of cancer like lung cancer, stomach cancer, pancreatic cancer, so many cancers. Among them, breast cancer is the major disease of concern in women. According to Globalization 2020 worldwide fact sheet, estimated new cases are 22,61,419, that is 11.7% of all cancer cases, and estimated deaths are 6,84,996, that is 6.9% of all cancer deaths. In India, estimated new cases are 1,78,361, that is 13.5% of all cancer cases. Estimated death cases are 90,408, that is 10.6% of all cancer deaths. Survival of breast cancer patient depends on early detection and accurate prognosis. We all know that breast cancer is the #1 position in India, in developing countries like India. Survival of the patient is possible with early detection and timely and appropriate treatment. Prognosis of the breast cancer can be monitored by using clinical conditions and pathological condition and molecular markers. Estrogen receptors is overexpressed in breast cancer. Expression of estrogen receptor influence the management of malignancy in breast cancer patient. Estrogen is an important mitogenic exerting its activity by binding to its estrogen receptor. ER express is reported in 50 to 80% of breast cancer. Endocrine treatment are used in estrogen and estrogen analogs. Targeted inhibition of ER retards tumor growth. Therapeutic hormones like tamoxifen competitively block ER, thus antagonizing transcriptional activation of gene required for tumor growth. Existing therapies target ER in breast cancer, hormonal therapy and chemotherapy. Based on ER expression that is positive or negative, which tells when we do IHC, that is immunohistochemistry, based on that report we are going to suggest that whether it is hormonal therapy or the patient go for chemotherapy. ER is the potential marker for predicting the prognosis and treatment response in breast cancer. It is a routine practice in evaluating all invasive breast cancer for estrogen receptor status. It helps in predicting the prognosis and responsiveness to endocrine treatment. Endocrine treatment is nothing but hormone treatment. Immunohistochemistry is the standard method as IHC has been approved to be equal and in fact superior in predictive tamoxifen response in the clinical relevant manner. Tamoxifen is not used as a single component, it is used with a combination of the treatment. Tamoxifen will come under hormone therapy. Levels as low as 1% positive staining of the carcinoma cells are associated with significant clinical response to hormone response. Prognostic role. ER positive tumors have better prognosis than those which are negative. Like estrogen positive will go for hormone therapy and estrogen negative will go for chemotherapy. That is also will be in the combination of many of the drugs. Treatment, prevent or delay recurrence in ER positive cancers. ER serves as good biomarkers for treatment decision to make a better drug combinations. Like if in case we are giving chemotherapy or immunotherapy and the patient is still have a recurrence or a high level of biomarkers, then we will go for the combination of drug. We will change the physicians will change the drug combinations. ER negative tumors do not benefit from endocrine therapy. This is what I said. Adjuvant tamoxifen reduces ipsilateral and contralateral cancers. IHC is the best suitable method for diagnosis cancer. In brief, the main important are as follows: benign and in situ lesions of the breast versus invasive cancers. Diagnosis of benign breast lesions, it will mimic as malignant. They will be benign, but they will be mimicking as malignant cells. In this case, IHC will give a clear picture of cancer cells. Identification of specific histological subtypes like lobular or ductal, we can able to classify them. Identification between the subtypes of breast carcinoma, fatigue disease of the nipples versus superficial spreading of melanoma and Bowen's disease. Diagnosis of intra and extra mammary metastasis. Prognostic and predictive application can be we can say by seeing the reports of PR, that is a prognostic estrogen receptor, progesterone receptor, and HER2/neu. IHC is widely used in the diagnosis of breast cancer. Mostly we are using in the histopathology, HND sections, and also IHC sections for predicting and prognostic values to see the values of that. Primary objective: to correlate clinico-pathological factors with ER status by IHC and HND, that is in amatoxylin and eosin stains. This is the hypothesis. Clinico-pathological parameters may be assessed by ER status of the breast cancer cases. Study methods: it is a cross-sectional study. We have done the samples are collected in Department of Pathology. Study period from Jan 2021 to February 2022, that is 14 months. In JSS hospital, a total of 97 breast carcinoma cases with all clinico-pathological parameters and hormonal status mentioned in the studies. We have collected modified radical mastectomy, lumpectomy specimen and core biopsy. They are examined grossly according to the standard guidelines, and specimen are fixed in 10% buffered formalin, and it is processed in histokinete, that is tissue processing. And we have taken 4 to 5 micron section, were cut using microtome by using paraffin embedded block and stained with HND and also IHC. Histological grade was assessed according to Nottingham modification of the Bloom-Richardson system. Representative sections with tumor and adjacent normal breast tissues were processed for immunohistochemical staining with controls. We have collected all demographical data and IHC reports and also histopathological examination reports. Result were analyzed to know the correlation of ER status and clinico-pathological factors in the patients. These are the clinico-pathological factors we have collected: age, size of tumor, and Bloom-Richardson score grade, and lymph node involvement, lymphovascular invasion, that is present or absent, and estrogen receptor status. Results. Here we have distributed age distribution can be seen according to 10 years of gap between 20 to 29%. 2% of the population are under 20 to 29, and 14% are falling between 30 to 39 years, and 22% are falling between 40 to 49 years, and 33% are falling between 50 to 59 years, and 19% are falling between 60 to 69 years, and 8% in 70 to 79, and 2% in 80 to 89 years. More breast cancer cases were observed in the age group of 50 to 59 years. This is a distribution according to the tumor size: less than 2 centimeter, 2 to 4 centimeter, and tumor size more than 4 centimeter. We can see here 37.36% falls in the group of less than 2 centimeter, and 37.36% in 2 to 4 centimeter, and more than 4 centimeter are 29.3 centimeter. Lymph node metastatic status, you can see involved 47% are involved and 51% involved. Just one second. Lymphovascular invasion, that is 46% and 54% are not seen. 50% of breast cancer are in grade 2. This is ER status. 44.55% we can see in ER positive cases, and ER negative are 42.43 cases. This is a picture of IHC. This is a picture of IHC positive. It is showing ER positive score 8 in low power, and this is CA ER control, and this is HND section, how it look like. These are the cancer cells. This is the table what I have shown in the pie chart. Discussion. Prognosis is related to clinical parameter, histological type, grade, lymph node metastasis. Estrogen receptor have with increased importance influenced the management of this malignancy. Age distribution, this and all we have already studied in the result part. A limitation, we could not able to consider core biopsy specimen to rule out lymphovascular invasion because the core biopsy will be very thin tissue, and IHC could not be run for some tissue fixed paraffin blocks which were taken by the patients for further investigation in a government setup or some other setups. IHC is only qualitative test, whether it is there, it is present or absent. We can't able to see quantity of the ER positive or ER negative cases. Conclusion, IHC has an increasingly important prognostic role in determination of factors that affect clinicopathological features. This is a large series of IHC profile of carcinoma breast as well as their correlation with clinicopathological parameters in Indian population. Correlation was observed between ER expression, age, tumor size, Bloom-Richard score, lymphovascular invasion, and lymph node metastasis. Correlation of ER status and clinicopathological factors in breast cancer patient give an effective plan of treatment, access better prognostic markers, provides quality care in cancer patients. Further study, I need to collect still more demographical details of the patient and also PR and HER2 new proliferation rate like Ki67, P53 values and all. I need to collect, then to evaluate MMP9 and IL-8 biomarker. These are my reference.
Thank you. Thank you, Priyanka, for the presentation. Now the session is open for the discussion.
What is the novelty of your study? This information we know, like ER positive, HER2 positive, everything we know.
Sir, this is my first objective of my PhD research work.
But at least you need to tell something new, no? This already known.
Yes. But it will be helpful for correlating with MMP9 and IL-8, matrix metalloproteinase 8 and IL interleukin 8.
What is the relation between those MMPs with this one?
Sir, the MMP9 is basically, it is a protein enzyme. These are the hormone status. These are the hormone status. They will be, we are conducting it in EHC and also in ELISA to estimate qualitative and quantitative, both in serum, saliva and tissue and also in urine. That is,
what is the relation with this PR and MMP9 or IL-18, IL-8 or IL-18?
It will, ER positive will give a hint that can be, MMP9 values can be increased in these cases.
Okay.
What is IL-8?
Interleukin 8.
I know that.
What is the function of IL-8?
Very good answer. I know.
It is a chemokine.
Okay, what does it do?
Sir, they are already present inside the body. Like they will be increased. They will be increased when the tumor cells, when the tumor cell start growing, the IL-8 will increase.
When inside the cell or outside the cell?
Outside the cell. And it releases to the bloodstreams.
Okay, then what happens?
Don't.
Okay.
Do you have any question?
So you said you're just starting out on your PhD on this. So what are some, and you've got this nice preliminary data. What are some questions you'd like to answer with moving forward with this? So that kind of jumps back to the question of the novelty, but just you thinking, what kind of questions would you like to ask based on what you've found so far and you've seen?
Sir, please.
Just any question that's based on your data here that you have. What kind of questions would you like to ask? Let's just say I came to you and I said, here's $1 million. You can do anything you want based on what you've done. What kind of question would you like to ask? There's no wrong answer. You can say whatever. Just think about what, don't get nervous. You're like worried that I've got something. Just.
Suppose if you have any grant from DST or DBT, what you will do that fund with your data, with your
based on what you've found so far,
with your samples.
Sir, I will conduct experiments.
What type of experiment?
ELISA.
What are your objectives?
Sir, correlation of MMP9 and IL-8 in breast cancer, we are seeing in all the types of body fluids. Majorly, we will get tissue only once in a patient, but we are correlating before surgery and after surgery. What will be the MMP9 value before surgery of the patient? This is a primary biomarkers which we can identify in the basic level of the cancer. Like we can in India, none of the patient I have seen from 1.5 years, they have come for stage 1. They are either in stage 2 or 3, 4. So if we are implementing this biomarkers, they are very sensitive biomarkers. At least we can able to pick it up very fast, and also we can go for chemotherapy or any of the treatments.
That you need to inform in your presentation. Like you need to take sample from normal people and also like a cancer patient with a stage-wise grade, like stage 1, stage 2, stage 3, like that. You need to answer that.
Okay.
What happens in normal patient serum, like cancer patient serum?
Sir, actually, ER will help in the modification of the drugs. The same way, MMP9 and IL-8 also can give a hint for a physician to rule out and monitor the treatment efficacy in the breast cancer patients. And according to the value of MMP9 and IL-8, there can be they can modulate, like they can increase the dosage or decrease the dosage. And also it helps in a recurrence, importantly. Recurrence of breast cancer we are seeing, so it will help in that cancer also.
Okay then, thank you.
Thank you, sir. Thank you so much.
I have a question, ma'am. Can I ask? I want to ask one question, ma'am.
Yes.
What are the features of MET to EMT in the development of breast cancer?
Sorry, can you repeat?
EMT. Do you know about the EMT?
No.
Epithelial to mesenchymal transition.
Sorry.
What are the feature in the development of breast cancer?
No, I have to read still more.
Oh, thank you.
Thank you, Priyanka. We have concluded with all the presentation. Results will be announced on the last day of workshop. I thank all the presenters, judges, and coordinator of this section. We appreciate for taking out time for the session. Now I take this opportunity to thank the coordinator of this session. I request Dr. Prasanna Kumar to hand over mementos as token of appreciation to the coordinators.
I mean, they don't know if it's something what like initially they won't have interested in before. At that time, for that it is very difficult to choose the item and they simply start and later they working. That is the thing. ಅವ್ರು ಕೊಡೋದು